yeah, yes, like the Americanization of it has happened, but we've always had a version of it, like so.
Right. So are you going to introduce me in some way?
Yeah, I'm going to say you did physics first.
I did.
Yeah, I did maths. Well, I'm still a mathematician.
Okay.
Employment, not by research.
Right.
And then
yeah, it was a long time ago in my physics, I have to say.
Yeah. No, I did it. I did it all the way to my PhD in maths, and then I went to neuroscience for my postdoc.
Right.
Sorry to cut you in. We are live on YouTube, and I'll go off screen, and we'll start in like 8 minutes the webinar. If you have anything, I'll be in the chat.
Yeah.
So
and I'll put those papers, David, and things like that. If there's anything else, just let me know that you want me to include. So.
Okay, he's gone. So how does it work then if we're live already?
So it's streaming on YouTube in a few channels there. And then what happens is attendees come in here at around 3. So I think it'd be diehard people who are already on the Room channel on YouTube. So they have 7 different YouTube channels streaming.
I see.
So it continually takes in the feed from Zoom.
And we can't see any of the audience, presumably. Do we know how many there are in the audience?
We will by the end, hopefully. And we also know there's Q&As, which that's my job as well, is at the end.
Oh, okay. To protect me.
Yes, to phrase them nice. You'll see them as well. There's a Q&A button, and they upvote them and down. So actually, not the first question, but the one that's most people want to hear the most about comes up.
Okay.
And
so
there's also
is that in the chat part of it? I mean, I'm not going to be able to monitor the chat while I'm giving a talk.
I do quite a lot of teaching, so I now can split my focus to 4 different places at the same time. And so I can monitor chat, questions, and images. But your job is just your talk, and then I ask you questions that's been asked by the audience.
Sure.
But you can also, at the end, you can see the questions, and if there's a question that's particularly,
one that I can answer, yeah.
Yeah, that floats your boat more, I was going to say. I think it's funny. I'm more on the behavioral EEG side of research.
Right. Yeah, I saw that. Yeah.
But I do a lot of stuff to do with in Parkinson's, where we look at resources, how much resource it takes to, and those. The word's very hard, but automatic resources get taken away. So therefore, how much computational power they can use and how it has to be distributed in a different fashion to make equivalent basic movement and decisions. I'm higher scale, but I'm going to be particularly interested in yours because I don't think that low. I think in columns and big parts of brains and button presses as opposed to,
Yeah, this is to some extent a side part of my lab. A lot of my work is on brain energy use and how it's generated and how it's allocated by mechanisms controlling blood flow.
Yeah, and that brain energy use is it's subtle. It's hard. It's a hard question.
Yeah.
As things go, I need to keep an eye on the time. Oh, we've lost
another 5 minutes, I think.
Yeah. Oh, it's like that. How to distribute resources, how to do it appropriately, and that's a harder question, maybe.
Right. So do you have any idea how many people attend this meeting normally? It seems very put together very late. It's impressive that you can put a meeting together in a few weeks like that, really.
In theory, there are 900 speakers, so 80 keynotes like yourself,
right?
850 short talks or interactive talks. And then there are other kind of sessions. And then on top of that, I think they said it was like another 2,000 people. So about 3,000 people.
Oh, well, I doubt that they'll all show up for this one. But,
no, but what I found is, because I worked with them on the Neuromatch Academy.
Yeah.
What I found is the talks, when they stay online, are the ones that get picked up. And that's where people watch them more and more. So that's actually kind of where you get your second, third, fourth, you know.
So is it recorded and left online for a while?
Mm-hmm.
Okay.
Forever in theory.
Oh, really? Okay.
And so, and that's done very well as well, you know. But as you say, it's a very short amount of time. Like I'm only very peripherally involved. I helped with the first two in the summer school, and I you know,
right.
It's actually great because I couldn't travel anyway. So
yeah.
For me.
Yeah. How's the COVID situation in Dublin now?
Well, we're in full lockdown.
Oh, really? Okay.
Yeah, we're up to what we call level 5. So 5 kilometers, most shops are closed. And I've been 100%, my university won't let me on campus at the moment.
Oh, really?
I'm mainly computational, so that's grand.
Right.
You know, I have some experimental colleagues, and that's very difficult.
Right.
But teaching maths online,
that's
why primary teaches. It's difficult. But yeah, London, things are calmer now, right? At the moment, anyway.
Well, it's ratcheted up a level in terms of severity, but you know, I can still do whatever I want to do, really, apart from go out to the pub or go and eat in a restaurant or go on holiday.
No, I have the only person right now in our bubble is my dad, and that's it. So the only person I physically see is him.
Yeah.
My daughter.
It is very weird.
Yeah. Yeah, it's on like the schools. My daughter's only 6, so
yeah,
the school staying open is important for her and us.
Yeah, I'm sure.
But she's off. You might even, I hope you don't, but she might hear in the background because this is a week off for her. She's painting in the room beside us.
No worries.
Shall we start if you're ready?
Sure.
Okay, great.
So, it is my pleasure to introduce David Atwell. He did his undergrad in physics. Let me get my notes. In Oxford, and then he did a PhD with Julian Jackson. Went to Berkeley for a postdoc and moved back to England to UCL, where his lab is based. And today we'll be talking about the regulation of neuronal propagation delays by node of Ranvier morphology, receptors, and ion channels. So thanks so much, David.
Thank you, John, for the introduction. So this is an oligodendrocyte in the cortex, myelinating axons. What I'm going to talk about today is how the propagation delay for neuronally transmitted information is determined by the node of Ranvier in myelinated axons. So I'm going to start out by reviewing myelinated axons and reminding you that conduction speed is a crucial determinant of neural circuit function, and think about how to tune it. I'll then, in the first third of my talk about morphological aspects of node of Ranvier function and how that can be used to tune conduction speed. So I'll look at variability of node and internode length and push the idea that node length is a tunable variable which can adjust conduction speed, and that to do this is particularly energetically favorable compared to putting more myelin on an axon. Then in the last 2/3 of the lecture, I'll turn to the properties of the electrical properties of the nodal membrane and ask the question whether myelinated axons are simply transmission lines of fixed latency. I'll show you that adenosine receptors and IH channels are expressed at sites of action potential generation, show you that adenosine modulates somatic excitability and decreases the conduction speed of myelinated axons. And then I'll look at where adenosine comes from in the brain and show you that uncaging calcium in astrocytes releases ATP, and this release regulates excitability and conduction speed. So let's start with the kind of theoretical problem. Here's the human brain. You need to send information across it from one bit of gray matter through the white matter to another bit of gray matter. And because this takes a long time, about 300 milliseconds for an unmyelinated axon, evolution has come up with the idea of wrapping the axon with lots of membranes of myelin to decrease the capacitance. And this increases the conduction speed by about a factor of 60. And at the same time, this myelin creates these nodes of Ranvier where the voltage-gated sodium channels are located to propagate the action potential. Now, why might we want to tune action potential speed? Well, if you want to send information like this across the brain, it might need to synergize with information coming from another part of the brain, because when there's coincident excitation of neurons, when synaptic currents arrive at the same time, that tends to facilitate the production of more action potentials. And because of the uncertain length of these pathways, you may therefore need a mechanism to tune conduction speed to get the timing right. Because conduction speed, and hence conduction speed, is important for neural circuits to function correctly. And indeed, theoretical work shows that if you mess around with conduction speed, then you mess up neuronal network oscillations, which may be important for cognition. So how is speed tuned? There are various ways you can do this, and this is particularly important if you want to coordinate time-critical physical tasks like juggling or playing the piano. A broadly known way of doing this is to increase the amount of myelin around an axon. This decreases the capacitance, and that will increase the speed. Another way to do it is to play with the internode length. If you increase the internode length, then a larger fraction of the axon is myelinated, so the capacitance is lower and the speed increases. But on the other hand, there's less passive signal transmission between the nodes the longer the internode is, and so that will decrease the speed. There are two opposing effects there. And finally, and much less well known, you can tune the node length. If you increase the node length, that means less of the axon is myelinated, so the capacitance will go up. And in addition, the resistance for current flow from the sodium channels here through the internode will increase, so that will decrease the speed. But if you increase node length by inserting more of the same kind of membrane, you'll be putting more sodium channels in, and that will increase speed. So I'm going to look at these various factors in the first third of my talk. And then in the last 2/3, I'll look at how tweaking the node electrical properties can adjust conduction speed. So let's start out looking at variability of node and internode length. If you label voltage-gated sodium channels, in this case in the cortex, you can see the length of the node here between the Casper cell adhesion molecules in the paranodes on each side, which glue the myelin onto the axon. And if you look either in the optic nerve or in the cortex, as here, you find that just looking pretty much anywhere, there are lots of different node lengths. Here's a short one, here's a long one in the optic nerve, and the same in the cortex. I've stacked some different length ones up here. And not only is the mean node length different in these two areas, in the optic nerve it's a micron, in the cortex it's about 1.5 microns, but the spread is different as well. So you can see there's a range of optic node lengths here in the optic nerve. It's broader in the cortex. So the node of Ranvier length varies between and within regions, but it doesn't correlate in any way with node diameter. So it's an independent variable from that. However, it does correlate with the number of sodium channels present in the node. So if you integrate the immunohistochemistry signal here over the whole node, you find the signal you get is proportional to node length, rather roughly. And so it's as though the same membrane is inserted with the same density of sodium channels, same number per unit area, and the longer the node, the more sodium channels you've got, and this will be important in a second. Now, what if we look along a single axon? Here's one axon which we fill with a dye, and we can then, this is in the corpus callosum, we can then focus in and look at individual nodes of Ranvier in that axon. And when we do that, we make a remarkable finding, which is along one axon, the node lengths are much more similar than they are between axons. So this is a roughly 1 micron node length axon. This is a roughly 1.5 micron length axon, node length axon. And this is a 2.5 micron node length axon. And another way of looking at that is to plot each of these curves is for one axon. This is the probability distribution in half micron bins of node lengths. Each one is relatively narrow, much narrower than the overall distribution of node lengths in the white matter tract. And that's expressed here in this coefficient of variation. Yet node length varies, has no correlation with internode length. So these seem to be two independently tunable parameters. This raises the question of how axons coordinate their node length at different positions along the axon, and we have no idea how this happens at present. Now, what does it mean? We've investigated this using computer modeling, using a model which is freely available at the Atwell Lab GitHub site, and you can download and run these models from there. And what we're interested in is how the conduction speed will vary as a function of node length if you adjust the node length. Now, this is the observed range of node lengths, in this case in the optic nerve. It's basically the same story for the cortex. So as we increase the node length going through this observed range, what's going to happen to the conduction speed? And I'm going to start out by looking at a situation where we have a constant number of sodium channels in the node, so that we add membrane but not sodium channels to make it longer. When we do that, less of the axon is myelinated, so the capacitance is higher. And in addition, this resistance for current flow along from the sodium channels here into the internode is increased. So both of those factors will decrease the conduction speed. So this dashed curve here for constant channel number just shows a continual decrease. However, I showed you earlier that the number of sodium channels isn't constant. It increases with the node length, implying that you're putting in membrane with the same sodium channel density here to make it longer. And in that case, as you go to shorter node lengths, you've got less sodium channels, and that decreases the conduction velocity down here. The decrease up the top end is the same, but you decrease the number of sodium channels down here, and that means that you get an optimum to this graph of speed against node length, which is somewhere around here. So changes of conduction speed in this, of node length in this observed range are predicted to significantly affect the conduction speed and could be used to tune the arrival time of action potentials. What about the internode length? If you patch clamp oligodendrocytes, the cells that make myelin, and fill them with dye, then you find here at P12, but also in older animals where the internode has finished growing, you find that there are differences in internode length between different brain regions. This is the corpus callosum, this is the cerebellum. And we think this reflects the development of the internode. We've done experiments where we image growing oligodendrocytes in zebrafish, in which the oligodendrocytes express a dye, so we can see the myelin sheaths growing here. And if we have a calcium sensing dye in those sheaths, we can see calcium transients, which occur every so often. And it turns out that the growth of the sheath over a 5-hour period is proportional to the rate of those calcium transients. So the more calcium transients you have, the more the sheath gets longer. Indeed, if you stimulate the axons electrically, which increases the rate of calcium transients, then you get a longer sheath. And if you block action potentials with TTX, you get a shorter sheath. And if you use patch clamping to put a calcium buffer bacter into the developing sheaths, then you prevent them elongating. So we think the eventual length of the sheath probably reflects the developmental process which is controlled by calcium transients in the developing oligodendroglia. Now, how does internode length variation affect conduction speed? This is like the graph I showed you earlier for node length, but now I'm plotting speed as a function of internode length. And you can see I've drawn it for 3 different node lengths. They're all broadly the same. All have got an optimum, and there's a maximum speed for each. As you initially increase the internode length, the speed gets faster because more of the axon is myelinated, and so the capacitance is lower. But as the internode length gets longer, eventually less current reaches the next node, and it takes longer to initiate the action potential at the next node. So altering internode length, like altering node length, will also tune conduction speed. And it's interesting that the mean length, typically you find the mean length is longer than the internode length that will give you the maximum speed. And we believe this is an energetic constraint. It turns out this saves energy. If you quantify this as the amount of sodium which comes into the axon on each action potential, you find that for each 1% that you increase the node length, internode length beyond this value which would give you the maximum speed, you reduce energy consumption by 2 or 3%. Now, let's just think briefly about the energetics of tuning conduction speed. I've suggested you could change the number of myelin wraps, or you could change the node length. And it turns out that changing node length is an energetically cool way to do this, because if you work out the amount of membrane you need to add 1 wrap of myelin to a typical corpus callosum axon, that gives you about a 10% speed change. You need to add about 1,000 square microns of membrane to each internode. In contrast, you can produce that 10% speed change by just manipulating 1 square micron of membrane at the node. So changing the node is more energetically efficient and probably also faster to tune the speed of an axon. Okay, I want to turn now to the electrical properties of the nodal membrane and show you that adenosine receptors control those and modulate conduction speed. Now, are white matter conduction delays modulated by chemical agents? We're familiar with all the neurotransmitters and metabolically derived molecules which can modulate synaptic function and neuronal excitability. Does this also happen in the white matter? Is it possible that there are systematic changes in conduction speed? I'm going to try to persuade you that adenosine is a crucial modulator of propagation speed. And we started out by using antibodies to adenosine receptors to look at whether there are sensors of this metabolic derivative in axons. We found no A1 receptors, but we do find A2a, we do find A2a receptors in the axon initial segment, for example. There's a soma just here. This is the axon initial segment where the action potential starts. If you label for adenosine 2a receptors, you see there are lots here. They go all the way down to the start of the region where myelination starts. This is antibody to myelin basic protein. And if you plot the density of sodium channels in green and of adenosine receptors in red, you can see there's a good overlap in the axon initial segment. And it's the same at the node of Ranvier. This is the sodium channels bracketed by Caspar labeling at a node of Ranvier. And if you label for adenosine receptors, you see there at the node as well, and their density matches that of sodium channels. Indeed, about 85 or 90% of AISs and nodes express A2a receptors. And interestingly, A2a is a receptor which raises the cyclic AMP concentration within cells. It's GS coupled. And there's an ion channel, IH channels or HCN2 subunits, which are gated by cyclic AMP and open to generate an inward current. And these are classically what form the hyperpolarization activated channels in the heart, for example, and in neurons. And about 50 to 60% of the AISs and nodes express these. And if you measure the length of the AIS, it comes out as pretty similar no matter which of these 3 markers you use. So can we look at what these adenosine receptors do? We can patch clamp layer 5 pyramidal neurons and puff adenosine. We've put a red dye, Alexa 594, into the cell here, and we then puff adenosine or an analog CGS, which activates A2a receptors. There's a red dye in this pipette as well, so we can see where it goes. We find that evokes a depolarization of the cell, about 10 millivolts in this case. If you puff ACSF, nothing happens, but adenosine or the A2a agonist both produce this depolarization. Now, what effect is this going to have on the firing of the cell? We're just puffing it on the axon initial segment. If we inject a current into the soma, which evokes a low current, which evokes a steady rate of firing, then this depolarization produced by the adenosine brings the cell closer to threshold, and so you increase the firing rate. Whereas if you're already injecting a high current into the cell and you have a high firing rate, applying adenosine decreases the rate of firing because you get a depolarizing block because you inactivate the sodium channels. So if you plot the firing rate against injected current, there's an increase in firing rate at low currents and a decrease at high currents, and that's just quantified here to show that it's statistically significant. So that's the AIS. What exactly is happening here to produce this depolarization? Well, if you do negative going voltage steps, you find that in these cells, there's a slowly activating inward current, which is this IH, hyperpolarization activated current, mediated by HCN channels. When you puff on adenosine or this agonist, you find this current gets larger. And carrying out a Hodgkin-Huxley kind of analysis using the tails, which are recorded in this little square box here, you can plot the activation curve of the channels. They're activated as you go more negative. The black dots are the control situation. The red dots are with the adenosine present. And you find there's a shift of this curve to the right. It's significant. And that means that more of these channels get activated at the resting potential, for example. And this is produced by cyclic AMP, because if you put cyclic AMP into the neuron from the patch pipette, then you have already got this rightward shift of the activation curve. And when you then apply adenosine, you get no further change. I should have said that these currents are blocked by the IH blocker ZD7288. So at the AIS, adenosine modulates excitability by raising cyclic AMP and increasing IH. It turns out it's the same at the node of Ranvier, and that's really what I'm focusing on now. So Jonathan Lesmie, a very talented postdoc in my lab, developed this preparation where he can patch clamp a layer 5 pyramidal cell, put a dye in the axon to be able to monitor where the nodes are by looking for branch points. Branches only go off where the nodes are. This is in a mouse we got from Diane Sherman and Peter Brophy, where there's GFP in the paranodes, so we can also see the nodes that way. And he then patch clamps the end of the axon where it's been severed by the brain slicing process, and there's a bleb which he can patch. And then afterwards, he can do immunohistochemistry to label for adenosine receptors, shown in blue here, producing this purple color, and at the node. So he then waits for a somatic spike to occur and records the resulting spike when it reaches the end of the axon. And then by doing these phase plane plots, we have some arbitrary criteria to define when the start of the action potential is, for example, shown by these dashed lines. And you can see that if you compare these two at the bottom, which are recorded at the bleb here, when you puff the adenosine, there's a significant increase in the delay till when the action potential arrives. So this delay is large and significant, and we can then estimate how that turns into conduction speed by making some assumptions, first of all, about where the action potential is initiated within the AIS, either in the middle or at the end. And we also need to assume something about the forward speed of propagation of the action potential versus the rate at which it goes backwards into the soma. And it turns out the extreme values you get by playing around with all of that are that it's either 2 or 3 times faster going forwards than it is going backwards. And that's these two plots here. No matter what you assume, you get a large change of conduction speed, about 2-fold on this estimate. And I'll validate that with some computations later. So this is a computational model where we use that same model that I showed earlier to simulate a soma with an AIS, then 3 internodes, and then the bleb. And the active membranes are just the AIS and the 3 nodes here. And we can then simulate the action potential spreading through these different compartments. And if we inject, first of all, we assume there's no IH present at all. just have standard Hodgkin-Huxley kind of currents. If we inject half a nanoamp here at the soma to get a nice rate of spiking, if we add IH, implying we're puffing adenosine just at the AIS, then that converts it to a situation where we get a depolarization of 5 millivolts or something. We now only get one spike because we've got a depolarizing block. So we're at the high end of that curve I showed you earlier. And in fact, if you plot a graph, putting the adenosine activated IH just into the AIS, then you get this blue curve here. So the more IH you have, the lower you push the rate of firing. If you put it just into the nodes, then you get no effect on the firing rate. And that's because the nodes are relatively electrically isolated from the AIS. So if you look at the resting potential at the soma, as you add IH at the AIS, it gets more and more depolarized, but putting it in the nodes has much less effect. If we look at this graph on the right, you can then do the experiment, the theoretical experiment, the other way around by putting IH just into the nodes. If you put it just into the nodes, you get a profound decrease in conduction speed, whereas if you put it just in the AIS, then you only get a much smaller decrease of conduction speed. And finally, if we simulate an extended axon, not a little stubby axon like this, but if we forget about the soma and the bleb and just have an infinitely long axon going across the corpus callosum, as we put more IH into the nodes, we find we can roughly halve the conduction speed like this. So I've shown you then that adenosine decreases the conduction speed, and the last question to answer then is, where might the adenosine come from? Adenosine is classically produced when energy levels get low in the brain. For example, as you're heading towards sleep, adenosine builds up. And I want to show you that the adenosine probably comes from astrocytes. Now, there's a long literature on astrocytes releasing ATP from Stan Cater's work in cultured astrocytes 20 years ago. There's been a lot of work by many people since, including Phil Hayden and his colleagues, which have shown that ATP is released when calcium rises in astrocytes. And that is then converted extracellularly to adenosine. And a recent paper by Anne Schaefer has shown that at least one of the enzymes doing this is present on microglia. So it's a real glial collaboration going on here. And that then can act on synapses to decrease glutamate release or modulate excitability. And in fact, as ATP is released and adenosine builds up as you head towards sleep, this is thought to promote sleep. So this is all in the gray matter. What about myelinated axons in the gray and white matter? If we label for astrocytes by labeling for GFAP, astrocytes, of course, are not just in the gray matter. GFAP actually labels white matter axons better. White matter astrocytes better. And we can then use the preparation that I talked about earlier, where we patch a layer 5 neuron, fill it with dye, and then look afterwards with immunohistochemistry to see where astrocytes are in relation to the path of this axon. And for both of these, for this axon here, if we focus in on particular regions, you can see there's an intimate association of astrocyte processes with the myelinated axon in both of these regions. And this is also true in the white matter itself. Astrocyte processes run in the same direction of the axons. And if you patch clamp oligodendrocytes and fill them with the dye and then label for GFAP, you again find that the oligodendrocyte processes forming the myelin sheaths are quite intimately associated with astrocytes. So does it matter? Well, to look at this, what we've done is to patch clamp astrocytes, in this case with the calcium dye ROD2 inside. And we also have NPEGTA, which is a caged form of calcium. So it's calcium bound to EGTA, which can be released by light. And we detect internal ATP with quinacrine, which labels vesicles of containing ATP. I should say the ROD2 doesn't go into all the small processes of the astrocyte, and it may be these ones which seem to be somewhat detached from the astrocyte process are in finer processes that the ROD2 hasn't gone into. When we uncage astrocyte calcium, and I'll show you a movie of this in a minute, most of these vesicles disappear because they're released into the extracellular space. You can see the number of vesicles is roughly halved. If you do this without the calcium cage in the astrocyte, there's no change in the number. And if you look in adjacent areas away from the astrocyte, there's also no change in these little green blobs. Now, is this ATP being released? We can assess this using luciferin luciferase, which generates a fluorescent light when ATP is present. And here we've just got a dish of this stuff, and we puff some ATP into it to show you get a light out of it, but you don't if you just apply saline. If we then put the cage calcium into the astrocyte and uncage it with our 2-photon laser, you get a big signal due to ATP release. And although there is a bit of an artifact here, you don't get this happening if you don't include the cage calcium in the astrocyte, as you can see from these mean data on the right. So we believe that when calcium goes up in astrocytes, it releases ATP. And this can happen during neuronal activity. Here's an astrocyte around a layer 5 pyramidal neuron. This is its dendrite, this is its axon, this is a blow up of this area. If I play the movie, you'll see that when it says spikes, now there's a gradual rise of calcium both around the dendrite and around the axon. And it turns out that if you just evoke spikes for a second, and they're going at about 30 hertz, then you get a delayed rise in calcium around the dendrites, because of substances released from the dendrites affecting the astrocyte. But if you stimulate for a longer period, then you get a rise around the axon as well as the dendrites. So this establishes that it is possible to get a rise of astrocyte calcium around the axon. And in this case, we now replace the neuronal activity by uncaging again to show that we can raise calcium in these processes here around the axon of this pyramidal cell here. And if we do this whilst recording the membrane potential is depolarized. This is blocked by the IH blocker that I told you about earlier. It's blocked by dialyzing in a blocker of this is the A2a blocker. It's blocked by dialyzing in a blocker of those IH channels, and nothing happens if there's no calcium cage in the astrocyte. So this is just like applying adenosine, and we assume that's what the calcium uncaging is doing. And if you inject current steps into the neuron at the same time, it produces an increase of excitability at low firing rates and a decrease at high firing rates, just like we saw with adenosine. And if you block A2a receptors, that abolishes this effect. And if you block IH channels, that abolishes the effect as well. So we think that uncaging calcium in astrocytes releases adenosine, which has the same effects on the AIS as I showed you for applying adenosine. And it's the same for the node of Ranvier. Again, we do use this stubby axon preparation filled with a dye, and we uncage calcium in an astrocyte here. And later on, we can do immunocytochemistry to show that the astrocyte extends around the nodes of Ranvier. And again, we find a decrease of speed of the axon, just like we saw when we were applying adenosine itself. So to sum all of that up, when neuron activity increases, that increases calcium in astrocytes, and this can spread to the processes near the axon. I already showed you that the morphology of the node of Ranvier can be used to change the speed of action potentials going along these axons. But in addition, release of ATP and conversion to adenosine activates A2a receptors, which raise cyclic AMP. They activate an inward current through these IH channels, and that changes the conduction velocity, as I've shown you today. So the change of conduction speed may disrupt information processing by altering synchrony of arrival of action potentials. It may disrupt generation of oscillations, which depend on timed communication between different parts of the brain, and it may contribute to functional disconnection as sleep approaches. Thank you for listening. These are the people who did the work, and I'm happy to take questions.
Since I'm the only person you can hear, Dave, just 75 people were watching this. So we have a few questions. Well, I'm actually, since I'm the host, I'm going to ask a question. What kind of time scale are we talking about for the changing of the length of the node? Like, I think you showed a plot there, like minutes or longer. Like,
I think we don't have data yet on how it might change in a let's tune the action potential speed kind of situation, but we are doing parallel experiments during ischemia. If you go to ischemia, the node of Ranvier gets longer, and we have quite good understanding of the mechanisms here, which are quite complex, and that takes about half an hour to occur. So it's not that dissimilar to LTP, if you like. LTP, you get a rapid change and then it stabilizes over an hour or so.
You know, like it's really phenomenal work from like clamping to the modeling as well. So the first question came in from Matteo Mancini. He states, a probably naive question. Although energetically expensive, is it possible to modulate the axonal diameter of a myelinated axon?
It is possible. In fact, when axons get myelinated, well, first of all, they need to be above a certain diameter in the first place to get myelinated. And there's a modulation of the diameter at the node. Often the diameter at the node is different to the diameter along the internode. So that's certainly possible. But there are lots of competing constraints here. You want to reduce energy use. You also want to have, for time critical information, you want the action potential to go as quickly as possible. So that means you want to have a large axon and you want to have the capacitance reducing effects of myelin around it. So somehow evolution has to optimize all of these things at the same time as not having enormous axons which fill up the entire central nervous system with volume.
And so, like, kind of, for instance, for example, like MS, it's a myelination disease, and would you say they both have issue with elongation of myelination, or is it like, I know it's decay of it, but can you speak to any of that?
You mean for remyelination?
Yeah, remyelination, or
after you lose oligodendrocytes, then typically people with MS go through several phases of remyelination. New oligodendrocyte precursor cells migrate into the region, and there's been a bit of a controversy about they get there, but they don't seem to develop into myelinating oligodendrocytes so well. And the question is whether there's a differentiation block there. Some of them early on do form new myelin sheaths. Those sheaths have different properties to the original ones. They're thinner. There'll be all sorts of adaptational changes going on. And there's also a big influence of microglia in all of this now, which, as is becoming increasingly understood. But to go back to the original question, it obviously is possible to change the diameter of axons. The question is, what are the constraints you're operating under? You don't want to get them too big. And the classic example is the optic nerve, where there just isn't enough space to have enough axons coming from the photoreceptors to represent all of them in the brain.
So the next question comes in from Daniel Gardner. In artificial neural networks, they have training versus production modes. So could astrocytes, ATP, and they're saying switch brains from action when awake to a recall or a replay during sleep. So is it a transition to a different state of encoding, maybe? No?
Well, certainly, I mean, lots of people think that sleep is a period where you refine all your synapses, renormalize the total amount of synaptic excitation so that neurons don't become hyperexcitable. I'm sure that astrocytes change what they're doing during sleep as well, but it's so early days for this kind of plasticity in the white matter that most of it is fixated on whether more myelination occurs. And it's clear that that does occur during learning. And a whole range of motor tasks have now been shown to depend on increase in myelination and production of new myelin by oligodendrocytes. So this is the work of Heidi Johansen-Berg and Bill Richardson, for example. But very little is known about changes in the node of Ranvier in this scenario.
Yeah, not so much, but in a lot of my other work, I do multi-sensory work where the, you know, two areas are communicating, and so therefore the timing and timing change is so important. So I would never have considered this thing. So next question from Barack Perlmuther. It's more a comment. Oh, reading as you go is difficult. Seymour Cray cited two aspects of computer design philosophy: remove heat and ensure that all signals that are supposed to arrive somewhere at the same time do indeed arrive at the same time. So a bit like what I'm saying with the multi-sensory world. So Barack suggests that you had more evidence that as we push to the limit of the brain, that our brain and computers might do the same thing, that we control the speed of information arriving. And this astrocytes. So it's really more a comment, as he says, but do you think it, you know, I think it seems very useful, but have you looked at it? You're looking at clamped neurons. Have you looked at it in like network of neurons yet?
We haven't done any modeling of networks. And the question really is how far you get away from experimental reality. I'm a physicist originally, so I like modeling, but you've then got to sell it to our experimental colleagues. And unless it's really firmly based on experimental reality, I think they will just ignore it. That's the problem. So I think that the aspect we want to look at next is sleep. It's known that there's a buildup of extracellular adenosine during sleep. I can't see why that wouldn't occur in the white matter as well as the gray matter. And so miraculously, it seems there is no data in the literature just looking at the conduction velocity of central tracts as sleep onset occurs. You would think this would have been done decades ago, but we can't find any papers on it. Hopefully, among your viewers, there will be people who will write in and say, I don't know the literature, and point out a relevant paper for me. But I mean, there is a clear prediction here, which is that the conduction speed, let's say across the corpus callosum or even in the optic nerve, should slow down as you move towards sleep and adenosine builds up. And that would kind of mess up the timing of circuits which depend critically on timing. Not all circuits do depend critically on timing, but lots do.
Yeah, no, a lot does. Yeah, I know nothing about sleep, but that amazes me. People haven't looked at, and I don't see anyone in all capitals giving out, so it seems that no one's disagreeing with you.
Okay.
So Eric Andrew Kirk asked, would the size, time duration of the depolarization wave matter with how many nodes and sodium channels summate under each wave? And is this the effect of conduction velocity from the adenosine independent from the wave size?
This sounds as though it's a physicist asking the question. The action potential is a kind of standardized length normally, unless things are going badly wrong. So yeah. It's about a millisecond long and 100 millivolts tall. It actually, when it propagates, it extends over many nodes of Ranvier, if you work it out. So for a typical conduction speed, let's say you pick 10 meters a second. I can't do the maths online, but you'll find that it extends over many nodes of Ranvier simultaneously. I'm not sure whether that answers their question.
Yeah, but actually, it's always a question I have as someone who only looks at papers with spikes. There is spike size differences, even in the simulations you showed there, that, you know, it's very subtle, but very few, I've never got a decent answer to see, does that matter? I know classically we think about naught and ones, but, you know, there was a paper just recently suggesting that, of course, interspike type timing is as important, if not more important, for some encoding than rate. You know,
I think probably the duration of spikes might matter more than the amplitude, because the amplitude is what you're ultimately interested in is neurotransmitter release at the synapse at the end of the axon. And the size of most spikes is going to sweep through the activation range of the voltage-gated calcium channels. But the duration for which the calcium channels are open will have an effect on the amount of transmitter released, and hence integration of information at the next synapse.
Yeah,
I'm sure someone can produce a model where it does matter.
Yeah, I can. Yeah, definitely a mathematician can produce a model where it has an effect, but that doesn't mean it does. So a question from Maria Ponria. Are you aware of other receptors or channels present in the nodes?
We're not at present, but there probably are others. And basically, I mean, based on this work, anything which raises the cyclic A level, so any G protein coupled receptor linked to cyclic AMP, to adenylate cyclase, is going to have the same effect probably. It would be interesting, for example, to look at amine transmitters, which you can actually image fluorescently, and see how their location correlates with node position.
Yeah, there's more. It's just to jump around the Q&As. Oh, did I ask this? Oh, most of the modeling of ion channel affects localized channels to the node. What channels are in the internode, and do these conduct? Do these alter conduction velocity? So
yeah, I mean, in the model which you can download from.
I put the link to the GitHub.
Our GitHub site. You can change the ion channels everywhere. Normally, we just assume in the internode itself, they're just potassium channels. It's known that there are channels under the junctions where the paranode forms in the region further away from the node than that, the so-called juxtaparanode. There are voltage-gated potassium channels. And under some circumstances, they may be able to get activated. It depends on the current path from the node through the tiny extracellular space to that region under the myelin. So certainly in pathological conditions, they can get activated, and that tends to decrease firing. So
the last question is by Ablofazi Zamanir, sorry. Does the research result in having a delay map of the human brain? At least a rough map.
The answer is no.
Well, it's a perfect way to have a succinct answer at the very
at a very reduced. I like this last question actually. From
good. Of course.
Seti. It used to be thought that, people drink coffee and they wake up, and the dogma for years and years was that that's because it blocks A1 receptors, which are presynaptic adenosine receptors which suppress glutamate release. And for all I know, maybe most people still believe that's the fact, but there is a paper in Nature Neuroscience, which you can find via PubMed, where they just do knockout experiments, where they knock out A1 or A2 receptors. They actually claim that the alerting effects of caffeine are mediated by A2 receptors. It's a very short paper. I'm not sure how well controlled it was, but that's what they suggested. So I've shown you today that adenosine acting on A2a receptors can affect the excitability of neurons, and therefore it could explain the effects of caffeine. I didn't know caffeine was beneficial in MS, but it would modulate propagation of the action potential as well as the excitability of neurons. Yeah.
But that's perfect timing. And I'd like to thank David again for a fascinating talk of just really impressive work spanning everything. And thanks again.
So hello, everyone. I welcome everybody to this session of Neuromatch conference. We have an amazing lineup of speakers in this session, and we are going to learn a lot about motor control from songbirds to human. I just remind everybody that this is an interactive talk session, so speakers have 5 minutes to introduce their work, and then we have plenty of time to ask many questions. So I hope we will have a fruitful discussion. So feel free to ask your questions in the Q&A panel, and remember to upvote your favorite questions so they would be asked first. And you can also use the chat to, let's say, to to share your thoughts and relevant information about the talks and seminars. So with that, and for the sake of time, I'm going to quickly introduce our first speaker of this session, which is Javier Las Ortiz. Javier is a PhD student in the Laboratory of Dynamical Systems in the Institute of Physics in Buenos Aires. And I think we're going to hear about different coding mechanisms in the nervous system of songbirds for the generation of bird songs. The stage is yours, Javier.
Thank you very much. Let me share the screen. And let me. Perfect. Well, thanks for the introduction. And I'm going to talk to you about a recent publication I've done with Cecilia Herbert, Gabriel Midlin, and Ana Amador, who is my PhD advisor. Well, as it was said in the introduction, there are, well, I'm going to talk about neural models under discussion. And in Birdsong, there are two neural models under discussion for the activity of a nucleus called HVC. While that is under discussion, the activity of the nucleus while the bird is singing. HVC is a nucleus that is involved in song production, song learning, and it has also auditory inputs. And for explaining both models, I'm going to use this figure. In the bottom, there is a pentagram that shows is a representation of the birdsong. Well, in one hand, the clock hypothesis proposed that HVC, the HVC RA projecting neurons, these ones, will burst every 10 milliseconds while the bird is singing. And on the other hand, the gesture transition hypothesis proposed that HVC RA projecting neurons will burst at significant motor instances while the bird is singing. That significant motor instances are called gesture transition extrema, or also GTE. Usually, few neurons can be identified in free-moving singing birds. So the red boxes here represents the activity of those neurons. And the white boxes in the two panels down here represents the predictive activity of both models. Well, the most popular animal models in birdsong are zebrafinches and canaries. Zebrafinches songs the song of zebrafinches is a repetition of one motif. A motif is a sequence of different syllables. Here I show one motif that is repeated 3 times, and this makes the zebrafinch song. Canaries song is a repetition of different phrases. And a phrase in canaries is a repetition of one syllable, always the same syllable. You can see it more clearly here in phrase 4. The phrase 4 is a 3 repetition of a whistle syllable. Here the whistle syllable is sung 3 times. So we calculate syllable duration, constant frequencies, syllable duration, and constant frequency segment duration for each syllables of the bird's own song. Each point represents one syllable. The scatter points are orange if the syllables contains a frequency segment duration larger than a threshold of 120 milliseconds. We found that canaries have a group of syllables different from the rest, called whistles. These syllables take the greatest values in the syllable duration and constant frequency segment duration. And there are no syllables with this characteristic in zebrafinches. Well, GT, or gesture trajectory extrema, indicates significant motor instances in the song, like maximum of air sac pressure or tension of the syrinx muscle. The syrinx is the organ analog to the vocal cords in humans. And well, GTs are identified automatically by software developed in the lab previously. For a better comparison, we generate pseudo-motif for canary songs. A pseudo-motif takes one syllable repetition for each phrase to generate a song similar to the zebra finch motif. We calculate the distances between consecutive GTE for 20 canaries in red and for 20 zebra finch in blue. We found that canaries have distances between consecutive GTEs greater than 100 milliseconds. Moreover, the syllables that have those GTEs were all whistle syllables, this kind of syllables. Well, what we conclude with this analysis that zebra finches GTs are distributed regularly along the bird's own song and very close one another. The mean distance between consecutive GTE is 15 milliseconds. This characteristic makes difficult to differentiate between the two models proposed for neural activity in HBC. In canaries, the canaries have in their repertoire simple syllables called whistles. These syllables have few GTs separated one each other for more than 100 milliseconds. This characteristic makes this species a promising animal model to differentiate between the two neural models. Well, you may ask what actually happens in the brain. Well, stay tuned because Ceci Herbert will tell you that soon. Thank you, and well, that's the talk.
Okay, thank you very much, Javier. We went a little bit over time, but it was good. I think 5 minutes is very, very hard to fit in. And now it's time for questions. Thank you very much again for the quick introduction to the topic. So I think our first question is from Conrad, and the question is, I usually work on humans. To which level can similar methods be used to analyze human or maybe rodents data? So let's say this way that you find these, what you call GTEs, I guess, right? Yeah.
Yes. Well, in rodent data, I really don't know because I'm not familiar with that kind of data. But in humans, I think, humans in the lab, we studied humans a long time ago, and we found that, to make the vowels, I can't remember the word. La frecuencia fundamental, the fundamental frequency, is the same for almost all the vowels. And the difference is only how you put your mouth and how you filter that frequencies with your mouth. So perhaps it could be analogous to GTE, the position of the tongue and the rest of the mouth, the teeth, to try to classify different and significant motor gestures. Yes, a motor gesture in human may be the position of the tongue and the teeth to make a vowel, for example. That's what I'm thinking. It could be a good analogy of how you can translate from bird song to human.
So let's see if I continue a little bit on that, elaborate on that question. Has there been any effort in also suggesting some other dynamical models that could, let's say, reproduce the vocal system of humans, like similar to the model that you have used and your lab has been suggesting for birdsong production?
In humans, well, that's not my specialty. So can you repeat the question? If there are more models of what? Of try to find just motor gestures?
The gestures, exactly. Yeah.
Humans. No, I know that the position of the, as I said, the position of the tongue and the teeth and the whole mouth could be classified for the vowels. In Spanish, A, B, C, A, B, C, A, E, O, sorry, I, O. But I don't know more than that. Yes, I'm specialized in Bergson. Sorry.
Okay, no problem. So I'm going to move to the next question from Kelvin Jones, and the question is: do the models? Well, actually, I have to also say, if the questioners would like to come on the screen and ask their questions lively, they can put on a screen at the end of their questions, and we will invite them to ask the question themselves. So I will read this question. Kelvin Jones asked: do the models apply for listening as well as song production? Does this make different predictions?
Well, this is a good question. This nucleus HVC, that we are studying, is a nucleus that has activity when the bird is singing, but also it has activity when the bird is listening to his own song, not every song, his own song. But the activity only can be recorded or is elicited when the bird is sleeping or is anesthetized. So there are experiments. We have made experiments in the lab. We made experiments in the lab with a bird that is anesthetized or sleeping, and we put a playback of their own song. And it is known in birdsong that the activity when the bird is sleeping and hearing his own song or singing his own song is almost the same. So we usually use that model of anesthetized bird for studying the dynamics of this nucleus that is HVC.
Okay, great. And maybe I move to the next question, which is from Alexei Krotov. I don't know if Alexei wants to ask live his question, or I read the question. Okay, I'm going to read the question. So he's thanking you for the talk, and he's asking if you can generalize your findings over other motor actions, such as body coordination, object picking, or transportation.
Well, we will love that. We will love that there are GTEs for other instructions, but we don't know. We don't know, and we are trying to understand GTEs here in birdsong. So I think it's an open field, and that we can ask a lot of questions with this approach. So it's an open field. I think it could be generalized. It has to be done. And
related, I think you briefly mentioned that. And so let's say in what you suggested in your talk is that basically the canaries are a nice model to be able to disentangle two different coding schemes. But you're not going to talk about any of whether actually which of the coding schemes might be our answer yet. Yeah, I think that's what we're going to hear from Cecilia. Is that correct?
Yes, that's correct. Because I made an study to try to figure which of the two models,
animal models,
or canary, are the best to answer this question. To try to see which are the neural calls underlying for HVC nucleus. Yes.
I'm going to ask you the last question. Are there any other models beyond these two models? That was also asked by Conrad before.
No, for HVC, the two models under discussion are the clock model, or also known as fire chain model, or the GT model. Yes, they are the both under discussion for this nucleus. Yes, only the two of them. The classical model is the fire chain model of the clock model. This model of GT is relatively new. It was proposed by my PhD advisor, Ana Amador, in 2013 in an article that she published in Nature in 2013.
I will post that also in the chat later if the audience want to dig into that paper. Thank you very much, Javier, and thank you very much, the audience, for all the interesting questions. I think with that, well, I will, for the moment, I will ask for the round of applause for the great talk. And we will move to our next speaker, which is from the same lab as Javier briefly also introduced, is Cecilia Herbert. She's a PhD student in the lab of Gabriel Mindlin at the Institute of Physics in Buenos Aires. And she will tell us now about actually these coding schemes, I think, that Javier introduced that could be studied in the Canary song. So stage is yours, Cecilia.
Thank you. Thank you for that nice presentation. So can you see my presentation? Fine. Great.
Yeah,
so yeah, I'm going to continue to tell you about the work that Javier just described. Given that behavior in canaries is better suited to test the gesture transition hypothesis, I set out to test the predictions of a neural model also developed in our lab using electrophysiology. And this is work I did with Santiago Boari and Gabriel Mindlin and Ana Amador. So here's an example of canary song. This is a spectrogram of the song. So that's a brief snippet of what the animals sing during the day. And you might see the long whistle syllables that Javi just described and the short syllables here. And these are the repetitions of syllables which make up phrases that make up the song. So this is another animal as the spectrogram and the associated air sac pressure required to produce these syllables. So the long whistles are typically produced with this pressure pattern called a P0 pattern, and other short syllables have this other pattern, P1 pattern. So this pulsatile expiratory gestures. And this is work, this categorization was done by our lab, and it will help us to group these syllables together. That's why I'm showing it to you now. The other element of this is the all-sign song system. We're looking at HBC, but this is a system in which the motor pathway is shown here in black and in green. And the neural model I'm telling you about was developed in our lab. It follows this circular architecture, so it's called the circular model, and it joins HBC with RA with the expiratory areas in charge of generating these pressure pulses that I mentioned, and an initiating area that gives rise to the instructions going to this circuit. This is a rate model, so we're going to be looking at summation of activity of individual models as activity over time. And the model was able to reproduce experimental air sac pressures that I showed you just now. So this is a robust neural model that produces the behavior that we're looking at. And I'm going to tell you about the predictions in HBC in a little while. But to test this model, I had to develop and record electrophysiologically in nucleus HBC. And this is the device that we used. It's just a micro drive driving tetrodes into the brain, so micro wire tetrodes. And we constructed it in-house since we had to make it ultra lightweight for implanting it on the bird's skull and compact. And using tetrodes also fabricated in-house made that it was customizable and low cost. So here's a bottom view of the tetrodes. And this allowed us to perform chronic neural recordings for around 7 weeks in most animals. So with this device, we recorded activity during song. Here you can see for a P0 phrase, the example sound pressure, the spectrogram associated with the song with the long whistles, and example activity for one neuron, an isolated neuron firing bursts correlated with behavior. These bursts are firing locked to syllabic rate, so one per syllable. And you can see this is the case in P1 syllables as well. So a different neuron, a different animal firing locked to syllabic rate in another kind of phrase. So these basic neurons are the ones that we grouped activity across birds. And I'm going to be comparing that to the predictions of the circular model, which for P0 syllables involve an initiating area having 2 kicks of activity and two sparse pulses in HVC during the start of the syllable. If you notice, this is the pressure pattern, remember, for the P0 syllables, and this is the long whistle part. And there we don't have an associated activity in HVC. So this reminds of the gesture transition model that Javier was, hypothesis that Javier was telling you about. So no bursts in HVC here, predicted. And this is the recorded in activity in HVC during song in 7 neurons for 5 different animals. And this is a raster plot of the activity of single neurons. And here you can see an example pressure pulse. So the summation of this activity is shown here in green. And you can see that there are pulses of activity in HVC at the start of the syllable, as I was saying. We couldn't find for this amount of neurons and birds any firing during the whistle. And there we did find something that was not in our model, which could help future iterations of the model, which was some sort of activity for the termination of the syllable.
Cecilia, sorry to interrupt, but we need to wrap up. Thank you.
Wrap up. Perfect. Yeah. So we found the same this to be in P1 phrases, and in conclusion, our experimental recordings support the circular model. So thank you for listening.
All right. Thank you very much. Very interesting results. So I think we have our first question. And the question is from Conrad, and he's asking, well, I don't know if Conrad wants to come live and ask the question. I think so, right, Shant? Okay, otherwise I will read the question out. Okay. So the question is, in a lot of mammalian cortical cells, there is firing to many other things. Are these neurons totally silent when the bird does not sing? Or are they really specifically tuned to bird songs and not doing anything else?
During production, they're actually hard to pick up. Like these projecting neurons fire very sparsely, and you might have seen that I don't have tons of neurons, and I did tons of recordings. So they're hard to pick out, and they do they start firing basically during specific phases of the behavior of the bird's production of song. We're not recording them outside of any other behavior. There is HVC does serve as a hub for other kinds of activity, but as far as we know, like they are starting to fire basically locked to song production. And they do, as Javier said, in some animals they do, in some species they do have this mirror activity, and they fire while the bird is listening to their own song. And in canaries, we're looking into that at the moment. So maybe next conference.
Actually, I mean, related to what you just mentioned, that they are quite sparse, and it's technically also hard to record from these neurons. Do you try, for example, as you said, because you know they're responsive to the playback of their own song as well? Do you try such tricks to be able actually to increase your yield of recordings, like playback their song, so maybe it would be more active, so you could have more neurons?
Well,
let's say,
yeah, no, this one is like an unbiased recording. Like we're just stimulating the animal to sing in many ways, doing some playback, but just like to get them to sing, not necessarily looking for a response. And in canaries, it's still not, I mean, we're looking at the moment, it doesn't seem to be the case that the same neurons that respond to the auditory stimuli respond to the I mean, are active during song production. So we're trying to figure that out at the moment. Some of the neurons I found do have that, but others don't. So yeah, it's not as reliable as a mechanism for fishing neurons. I mean, I think the way to go there would be some sort of imaging, you know, to get some a wider field of view, but then you lose the time resolution. So yeah, it's hard.
Thank you. And or neuropixels.
Well, yeah, I mean, it's, yeah, of course.
Yeah, promoting now.
But I wouldn't, I mean, I don't know, would you implant chronically for 7 weeks with a neuropixel?
Well, I think there are actually, I think even in this conference, we are going to hear about some people that have implanted and they're recording chronically with neuropixels probes as well. Nice. Yeah, and which,
we don't have the resources at the moment to carry out.
Right. Okay. And we have the next question from Kelvin Jones, and he's asking if it is possible to apply local cooling in the circuit and test predictions of your circular model.
Yes, it seems that that you have been reading the literature from the lab. I mean, it is true that in HPC that we have done some local cooling, and we see we see the predictions of the circular model testing there as well. Yeah.
Okay. So you have done this experiments?
Yeah, no, I mean, not my experiments that they were done previous, and I think they were during auditory, not production, but yeah, that has been.
And then when you were speaking about this projection, you call them projection neurons. Is it, so what was, because I'm not very familiar with the field, is it like, so you kind of dissociate between the projection targets, like whether they are innervating different downstream areas, or just as, so somehow you're labeling these cells, or no, you're just recording from every neuron there?
No, technically that would be brilliant to actually do some antidromic stimulation to figure out who they are. Up to now, we characterize them based on the electrophysiological behavior. So projection neurons in these nuclei, in HVC rather, are these phasic firing ones. So that's the characterization. So the results I showed were of phasic firing neurons, and those are putative projection neurons. Exactly. Yeah.
Okay. So and again, I'm gonna make a kind of a loop to the first question. And since normally, like in the mammalian cortex, when we have these premotor areas, or at least speaking of cortex, sometimes they are responsible for different, let's say, motor outputs, not necessarily one body part. Like in this case, okay, we're talking about complex motor behavior, bird song generation. But is there any studies or anything that suggests also the implication of these neurons in other motor control or any other type of movements of the birds or so on?
Not as far as I know, but it is hard to it's like which population of neurons, like it's just a general thing at the moment, right? The whole like projection neurons, or so maybe there's something comes up if we can get to see like different subcircuits within HPC that are currently not described. I don't know.
Right. For example, in the rodent field, there's I would say a tendency to like more and more sophisticated ways, try with like high speed filming and so on and so forth, try to really quantify different behaviors, like even like the small, very like small movements of their facial movements or like forelimbs and hind limbs and so on. And we see a lot of correlations of those type of movements during like task performance and so on in a lot of these premotor cortices. So I was wondering if one has also tried to really quantify such behaviors and see if there's any correlation between the activity of the HPC and such behaviors. But yeah. Very interesting. Thank you very much, Cecilia. I think now we can move to our next speaker. So we're going to move from the songbird. Before that, again, I'm going to ask for a round of applause for Cecilia for a great talk and answering the questions. Thank you very much.
Thank you for having me.
Thank you. And so with that, I'm going to introduce our next speaker, which is Eric Kirk. And Eric is going to talk now, move us from the beautiful world of songbirds to humans. And you're going to still hear about motor control, but in this case, he's going to talk about the structural properties of and how they can, let's say, impact the motor unit firing rates among muscles. And Eric is a PhD student in Charles Rice Laboratory in the Faculty of Health Sciences in Western University, Canada. Stage is yours, Eric.
Yeah, thanks. Oh, yes. All right. Just give me a second. Okay, sweet.
No problem.
So here we go. So we take
yeah,
it's better now. Yeah. So we take a muscle to brain approach in the Rice lab. So what we do is we study the activity of muscle at the single resolution of motor units during the full spectrum of voluntary contraction. So from small efforts up to as hard as you can contract the muscle, we can put a needle in the muscle and we can do electrophysiological recordings to sample the firing rate activity at the single motor unit level. So it's a spinal motor neuron to muscle fiber relationship here, where each motor unit comprises a single lower motor neuron, an axon, and many muscle fibers, and that's the needle. So we have an approximation here where we have activity in the spinal cord that relates to activity in the muscle fiber. And here's a recording in a hand muscle where the person is keeping a steady contractile effort. And then we get the firing rate from multiple motor units here that are then decomposed into single motor unit activity. And from that, we get a mean value, and that's the overlay there. So that's what we can do. So what we see when we compare different muscles is that muscles have different firing rate ranges that are independent from contraction intensity. So they can receive similar relative inputs, but the firing rate outputs are different. And in some muscles, it's up to a 3 to 4-fold difference. So the talk here is about why is this difference occurring? So we were able to aggregate a data set from 18 published studies and 3 unpublished data sets over the last 20 years from human intramuscular work from 15 muscles. And what we did is we did post hoc muscle ranking to see which hypothesis may be most likely to be true based on the way we did it. And then we, I'll show them in the next few slides. So just to give you an idea of how firing rates, mean firing rates increase the contraction effort, this is a scatter plot of contraction effort on the x-axis and mean firing rates on the y-axis, and every dot is a different motor unit sampling. So you can see that as contraction intensity increases, firing rates go up and variability increases. So here's the same 3D plot rotated. And now we see each of those contraction effort firing rate relationships grouped by muscle. So you can see that there are shifts depending on what muscle you have with variability and how the line is. So from here, we ranked muscles based on 4 common ideas in the literature. One was innervation differences, so brainstem versus spinal may affect firing rates. Axon length, so how long the distance needs to be traveled from the lower motor neuron to the muscle fibers. Proximal versus distal, so something in a hand versus something in the arm. And then fiber type, so slow fiber type muscles versus fast fiber type muscles in humans. So the technique, again, we sampled motor unit activity, we took a mean value, we then took all of the data and we put it into a regression model where we could account for things like the person, the age, contraction intensity, there were many factors. And then what we did is we created a null distribution based on the residuals of the model. And we fit the model and we bootstrap the residuals, the residual bootstrapping, to then have an association to our hypothesis of interest. So the relationship here was null. And what we did is we created a null distribution, and then we tested each alternative hypothesis. And that is the blue dashed lines here you can see. So the blue lines acted as our alpha. And this is an example of one hypothesis test. So the result from this was the 4 models were tested, and we iterated the models to get 100 hypothesis tests for each model type. And using the full data set, the model most likely to reject the null distribution was axon length and fiber type. And then we did a second series of tests with a subset data set that comprised more factors to explain the variability. And again, we found that the axon length was most likely to reject the null. So that gave us some confidence that this may be an actual relationship that determines or affects firing rates. So the main result was that the axon length model had the highest probability to reject the null. And then for open discussion, it'd be neat to learn about people's ideas with how they think axon length or other things related to our analysis may affect or limit the firing rate. So thanks.
Thank you very much, Eric, also with very good timing. So let's see if we have questions. We have the first question from Conrad, and he is asking, what does it mean for a model to have low p-value?
Yeah, so it mainly meant that the dashed blue lines were farther away from our null distribution estimate. So that would indicate there would be less area underneath it then. So they would just be farther away, and the result would be more likely to reject the null in that case.
And so have you also, how much do we know actually about the correlation of these different variables? Let's say you're testing different parameters, and do we know if some of them are correlated with each other? And have you also, for example, tried like a generalized linear model where you could have the interaction of parameters, or at least like all of them in the same model, and then see which one would explain more, like the variance in your data, in the firing rate and so on? Have you tried anything in those lines?
Yeah, example would be contraction effort and firing rates are correlated differently for each muscle. Altogether, it's like a 0.4 relationship, so high, I guess. Yeah, right now it's just an additive model, but that's a good idea to apply different interactions.
And we have one more question from Gregory Pursey, if I pronounced it correctly.
Yeah, Greg. Yeah.
Well, he's doing nice work, Eric. Did you folks, by any chance, looked into extension, flexion to explain the models? And this is one of the most basic differences that has been observed in the intrinsic properties of the motor neurons.
Yeah, the Wilson paper, I think, and Heckman's lab looked at extension flexion. We did, and it didn't really have a big effect as a model. Like an example would be we had some brainstem muscles, like the frontalis muscle, or a partially innervated brainstem muscle that had the highest firing rates. But then again, the axon length was really short. And then we had some muscles, we tested the FDI also, and that had high firing rates. And it was higher than maybe an axon length that was ordered the same as something in the lower limb. So maybe at like lower contraction intensities, flexion, extension, differentiations may hold out, but maybe not when you get higher, as we saw with the brainstem muscles and shorter axon lengths.
Great. We have one more question from Kelvin Jones. And Kelvin is asking if this prediction generalized independent of handedness. And in other words, are the models similar in right versus left-handed preference?
Yeah, maybe we didn't have access to that type of data to compare them. That's an interesting question. It could be looked at.
It's interesting too. I mean, coming from a very totally different field, looking now again at in this like motor units and something that I was, maybe learning about many years ago, but absolutely interesting looking at these correlations. And so we have follow-up questions from Gregory, and he's asking, would you speculate that the models would generalized to rhythmic activity?
I'm not sure. I made some extra slides for fun, but some of the things I thought about was when you think about max firing rates, it may have to do with the action potential wave or the latency and how long it could take maximally. So I think this may be an idea about kind of what the max limits are constraining firing rate ranges. I think rhythmic activity, which may be at lower levels, perhaps may not be constrained at all by these types of ideas. But maybe, yeah, like it's an interesting question. I don't.
Could you a little bit more elaborate on this suggestion of what you have? Like here, I'm not sure I got the idea.
Yeah, so some of the faster firing rates that we see maybe like if you think the axon length holds out, it may be due to more serial components of nodes and the waveform having to travel through it in time, which may limit firing rates. So it's kind of the maximum, or not a maximum, but one of the limits that we would think about that may constrain firing ranges. So rhythmic activity would be under the limit probably always for lower level rhythmic activity. So I don't know if it would be affected by axon length in that way. But
interesting, I think.
Yeah.
Great. Thank you very much, Eric. If there's no more questions, I would like to ask all the audience and myself for another round of applauses for Eric and all our speakers of today, Javier and Cecilia as well. Thank you very much for great talks. We learned a lot about motor control. And well, I think we have one more question, Eric, if you want to take that. So it's also from Gregory. And I think he's asking if you think that the synaptic inputs from CST CPG may retrain firing rates in various ways. We have obviously another 2-3 minutes to go through this question if you have any ideas.
Yeah, cool. Like you would think like I guess I can't ask him back, but I guess CPGs would kind of be like they would kind of set each cycle. They would be setting the firing rate rhythmic cycle, I guess is the way the way we would think about that maybe. I don't know. Like the stuff we looked at here was all steady state stuff. So we weren't doing any type of rhythmic up and down. But that'd be interesting because it may be like the onset phase and how the firing rates increase with the onset phase of the contraction. May be different that way.
Great. So thank you very much again, Eric, and thank you, Javier and Cecilia for beautiful talks. And thank you very much, the audience, for being very active and asking constructive questions. Hopefully they have been useful for also our speakers. And thank you again, speakers. So with that, I hope you will enjoy the rest of the NeuroMatch conference and all the other sessions. And I thank also Ishant for the back end for technical help of sharing this session with me. Thank you very much, everybody.
Thank you.
This from the University of Bern to talk about sleep circuits. Please take it away.
Okay, can you hear me? Okay, welcome everyone. So thank you for the introduction invitation. What I want to do today is to tell you a little bit about the structure and function of sleep circuit in the brain, the mammalian brain, in the mouse in particular. That's what my lab is interested in. And I will focus today on really brain oscillation during sleep that occurs in the thalamus, in thalamocortical loop, in particular spindle, and their relationship with REM sleep. So the reason what my lab is interested in is to better understand the basic mechanism of sleep. This is important not only to understand the function of sleep, but also to design new diagnostic therapeutics that are important for sleep disorder, insomnia, RBD, narcolepsy, but also subtle change of sleep that are associated with early onset of neurology and neuropsychiatric disorder. For instance, schizophrenia is associated with change in spindle very early on. So we do that by studying the connectivity, dynamics, and plasticity of sleep-wake circuit using different tools that I will present today. So short note, all the work I'm doing and I will be presenting today is done in mouse, in the mouse brain. And this is very convenient because sleep-wake circuits are conserved in the brain, in particular the subcortical structure, but also thalamus and neocortex. And we have this advantage of using a mouse model for which the genetics is quite well described, physiology as well, and we can use exactly similar tools that we use in human to study sleep, including EEG, EMG, EOG. So quick looking on the right side, you can see an EEG in blue, EMG in red. And you don't need to be an expert to see the difference between EEG here during wakefulness and then during non-REM sleep, which is the first sleep states, and REM sleep, which is the deep sleep states or dreaming states in human. So you see that during the first sleep, there is this slow, high amplitude oscillation that are called classically delta or slow wave here. And you can see some spindle here, for instance, that occurs during non-REM sleep. During REM sleep, it's mainly theta rhythm with high gamma synchronized coming from the hippocampus. So this is sleep in a mouse brain, and the cycle is very similar as the human brain in this case, where after prolonged wakefulness, human enter into stage 1, 2, 3, and then REM sleep as dream state. So you see that we have very similar EEG signature of sleep, including slow wave, spindle, and theta rhythm during REM. So interestingly, decades of work have brought different schools of thought on where sleep comes from, and I just want to highlight here that it's basically two current views. One is a circuit views where that basically relies on sleep and wake promoting circuit. When you are awake, wake system are active and they inhibit sleep system, while sleep system are active and they inhibit wakefulness, right? So there's a switch between sleep wake promoting circuit, and those exert strong mutual inhibition on each other. There's another view that is basically oscillation view that comes from the fact that sleep can be locally regulated. I'm showing you here slow wave in a nap that follow motor task. You can see that the amplitude of those slow wave is larger in the motor cortex. And so that refers to the local sleep and the fact that organism sleep can occur as a consequence of spontaneous state synchronization of multiple local networks. So in other words, sleep can start in one place or several places in the brain and can reach the global network synchrony. And this is quite important since with the use of high density EEG that highlight the importance of local network synchrony and how they could lead to global sleep. Both mechanisms for both views are still under a lot of work, and regarding the circuit view, this is a summary of how the sleep-wake cycle could occur in human, rat, and mice with all the possible transition between wake, non-REM sleep, and REM sleep. And I highlighted here for each state neurons that are active during those states. So they are basically the activity correlates with wakefulness is the case for locus coeruleus norepinephrine neurons or hypocretin-orexin neurons. There's the same for non-REM sleep, in which anterior hypothalamus contain inhibitory cells that are strongly active during non-REM, and for the collection of nuclei from the brainstem and hypothalamus for REM sleep. So one thing that I highlight here is the color one as the one for which there have been a lot of replication of the data, not only with correlative approach, but also with causal approach, and I'll get back to this in a moment. So as you can see, both wake and sleep comes from a coordination of complex circuit in the brain and have obviously multiple origin. How those one communicate to each other is still not clear, and that's something that we are interested in. So on the other side, on the oscillation view, what I want to show you today is our recent work where we found that some oscillation during non-REM sleep, and in particular spindle, seems to be associated with the onset of REM sleep. Okay, so slow wave and spindle come from thalamocortical loop, and this is summarized on that scheme and comes from the original description from Steriad and other groups, where 3 different cell types from the thalamus, reticular thalamus, and cortex are connected to each other, and the dynamic between those 3 different cell types generates these up and down states here, as you can see on this figure here, that are reflected as the slow oscillation here on the right. And the spindle oscillation here is thought to be generated by the reticular thalamus here that exerts a strong inhibition on thalamocortical circuit. So we started to study the slow wave using different approach, both correlational and interventional. I won't go into too many details here, but we'll focus mainly on in vivo electrophysiology and single multi-unit recording. And that gives us an important information of about the single cell dynamics among sleep circuits that we can then use to feed into our optogenetic model to manipulate circuit, sleep oscillation, and induce and study causative relationship. And so that's what we did here. But we were interested in the medial dorsal thalamus, that project to restricted part of the cortex, including the cingulate cortex and the insula. And again, I won't go into too many details, but the important finding of that initial paper is that by studying a different part of the thalamus and the cortex, and namely the medial dorsal thalamus, the anterior cingulate in the cortex, and then the high order anterior dorsal thalamus, and then barrel cortex V2 in the cortex, we're able to follow slow waves. This is known from human study that slow waves are predominantly generated in the frontal cortex and then travel to the rest of the brain. So in this study, we found that first, the medial dorsal thalamus is able to pace slow wave in the cortex. That's what you're seeing here. Those optogenetic modulation of CMT cells travel in the cingulate and then in AD and then in the cortex. Interestingly, you can see that the barrel cortex seems to be quite isolated from that thalamocortical circuit. And then we found that this higher order thalamus kind of relay what's happening in the prefrontal cortex to the rest of the brain. And we found that by, at the same time, we activate the CMT, silence those AD cells. And when we silence these cells, you can see that in the visual cortex, those optogenetically induced slow waves are much more slower. So that was one important study that really helped us to understand how slow wave travel. And then also in that study, we showed that the traveling of those waves is important for sleep recovery. So if you activate those circuits, it promotes sleep recovery faster. If you silence it, it delays it. So then we move on to spindle, which is another part of oscillation that are associated with non-REM sleep. In a recent study, we aim at quantifying them in an unsupervised manner. So we design an algorithm to detect those spindle first in human, and then we adapt it to mice and detect spindle in all those recording spots, in particular the reticular thalamus, the sensory thalamus, barrel cortex, but also different part of the cortex. EG1, EG are basically frontal and parietal. And so what we found that was surprising is that, of course, we detect spindle during non-REM. That is what is shown here. But interestingly, we could classify non-REM into non-REM going to transitioning to wakefulness, or non-REM transitioning to REM sleep. So this is the black and the pink one here, or the red one. And what we found is that there are higher number of spindle in the non-REM that transition to REM sleep than the non-REM that transition to wake, suggesting that maybe non-REM are important for REM transition. And so this is what we basically demonstrate using optogenetic, where we activate reticular thalamic neurons in the mouse brain during sleep. And this is to basically show that when we do that, we increase the spiking of reticular thalamic cell and increase the burstness of those spikes. But interestingly, we found that when we do that, there's an increase in the transition from non-REM to wake, really establishing a causal link between spindle occurrence and REM transition. We're not sure about the mechanism yet, but there's definitely a causal link here. And so I will finish with the conclusion where I show you that the medial dorsal thalamus contribute to the generation of slow wave, and is an important part of the circuit for sleep homeostasis. And that in terms of the recent spindle study that we published, we believe that extend the existence of spindle beyond the classical reticular thalamocortical pathway and include non-sensory thalamic nucleus, including the medial dorsal, and that those spindles are in particular causally involved in the onset of REM sleep. And so I'll thank you for your attention. Thanks people who work with us, funding agency, and I'll be happy to take any question.
So that's the applause you hear on my from me on behalf of all the attendees.
Thank you.
So we have one question right now from Andre. His question is, or sorry, I do not know. Does all part of the brain need the same amount of sleep, or are there parts of brain that can live with less?
That's a good question. So I cannot answer the question about the need of sleep, but I can definitely tell you that different part of the brain sleep differently at different time of the day. That means that some part of the brain are more sensitive to sleep pressure, in particular medial dorsal thalamus or prefrontal cortex are area that start to switch into the sleep mode very quickly as compared to the other sensory area. Whether they need to sleep more or less, that we don't know, but definitely there are studies that show that some cells are differently sensitive to the deprivation of sleep. So that means that some might be more resistant to sleep deprivation than others, meaning that some of cells degenerate if you have chronic sleep perturbation. So yes, definitely sleep is not homogeneous. It's different in different part of the brain, at different time of the day, depending on the sleep pressure, whether there is a need or what are the mechanisms, this is still unknown.
Got it, thanks. So we have about a minute left and we have quite a few questions. So I'm just going to pick the first one. Like, have you observed spindle modulated neurons in the anterodorsal nucleus? And do these neurons burst?
Yes, yes, and yes to the two questions. You can go to the Bandarabadi paper in NatureCom that was out a few weeks ago. We described this. So yes, we could find some.
Got it. And the next one comes from Aishwarya. What algorithm or method did you use to classify spindles back to wakefulness and toward onset of non-REM?
Wavelet analysis. B-spline morlet to be precise. This algorithm is free available. You can contact me if you want to use it. We've been testing it in human, transferring it to mouse recording, and it is all reported in the Bandarabadi paper as well.
Thank you. The last one is, do these brain regions contribute to generation of slow waves and sleep homeostasis in babies versus adults?
Yes, most likely. We have done that in adult mouse, so our conclusions are limited, but it is likely that those brain regions are also involved in generation of slow wave in infants. Yes.
All right. Thank you for the questions, and I think we are running perfectly on time. So,
thank you.
Thanking again. Next up, we have Agustin from University of Buenos Aires. To talk about sleep spindle and slow oscillation coupling.
Thank you. Well, well, my name is Agustin Solano. I work in the Physiology of Action Lab in Buenos Aires. My work is titled Local Coupling Between Sleep Spindle and Slow Oscillation Supports Consolidation of Visual-Motor Adaptation. So when we talk about motor learning, we can talk about motor skill learning that implies the acquisition of the new motor policy, the novel controller. For example, when you learn to ride a bike for the first time. Or you can talk about motor adaptation learning. That is when you adjust well-practiced action to maintain the performance when the environment is rotating. Like here, when the dog is trying to walk on ice, so he had to adjust his movement to try to be successful. So in our lab, we're interested in both kinds of motor learning. So how we study them in the lab. For motor skill learning, we can use motor sequence learning tasks when participant has to learn to perform a sequence, tapping in the keyboard or in a response box. And we measure how many successful sequences they can do it for during time, for unit of time. On the other hand, we can participant can learn a motor adaptation task. For example, here we can see a participant trying to move a cursor on a computer screen using a joystick manipulated with the thumb and the index. And we have to reach different targets with the cursor. So in some trials, the movement of the cursor is going along with the movement of the hand. But sometimes you can introduce an angular perturbation. So when you move a cursor, you move your hand, the cursor will move apart from your hand about 30 or 40 degrees. And you have different trajectories. And eventually we can remove the perturbation and see the after effect. This is a motor adaptation task. So in particular in our lab, we're interested in memory consolidation. And we have done some previous work on memory consolidation, diurnal consolidation during the day. So here in this work, we using a anterograde interference protocol, we can study a consolidation. So participant here adapted to one perturbation, a clockwise perturbation, and then after a period of time, they face the opposite perturbation. So here we have a 4 group in which this time interval was about 5 minutes, 1 hour, 6 hours, 24 hours, and a control group that only did the second perturbation. And we measure the rate of learning, the velocity of learning and retention. And we saw that both the capacity of learning and retention was released for interference after a period of 6 hours. So it's according to the literature of our work, our lab and previous other labs that confirm that this period of diurnal consolidation is about 6 hours. But we're also interested in sleep-dependent consolidation of motor memory, because we know that sleep benefits motor skill learning, but there is some controversy about the role of sleep for visomotor adaptation learning. We also know that for declarative memories, another kind of memory, the precise coupling between a slow oscillation and sleep spindle is critical for the consolidation, for the sleep-dependent consolidation of declarative memories. Finally, we ask about if this association between a slow oscillation and spindle also operates in the stabilization of human motor adaptation memories, specifically during non-REM sleep. So we hypothesize that sleep plays a role in the consolidation of motor adaptation memory, which may reflect an enhanced memory retention, or maybe less forgetting, or memory persistence, or both. And then we think about the coupling between sleep spindle and slow oscillation is also important for sleep-dependent consolidation of motor adaptation memory. So here we designed an experiment to look for the benefit of sleep on BMA memories. So here we have an experiment with 2 groups in which we manipulated the proximity between learning and sleep. So one group learned at night, and immediately after learning, they went to sleep. So here I have a BMA session. And then they went to sleep where we recorded brain signals. And then we tested memory, obsessive memory, 24 hours after learning and 2 weeks after learning. And we have another group in which the distance between learning and sleep is about 14 hours. They learn in the morning and they come back at night to sleep. And then we have the two tests for memory assessment. Just for the record, during the night in the polysomnography or sleep study, they slept all night, and we recorded EEG signals, EOG signals for eye movement, and MEG in their chin. So with this experiment, we saw that during adaptation, in the first session of adaptation, both groups adapted to the counterclockwise perturbation. But when we measure the memory retention in test 1 and test 2, we saw that the group that slept immediately after learning, the 10-minute group, benefited more from sleep, about 34%. So they show a higher retention, but both groups have a similar rate of forgetting between the 2-week interval. So we see that the retention is benefited, but not the memory persistence. So we can say that the proximity between learning and sleep is benefiting memory retention. So we also are interested about what is happening during night. So according to previous literature, we look for sleep spindle, specifically density of sleep spindle after learning. So we ran another experiment with another group of people that they participate in 2 sessions, a BMA session and a control session. So they have a training session before sleep, they went to sleep, and then we have a testing session in the morning. In the BMA session, they adapted to a perturbation, and in the control session, they just adapt to the movement, but without perturbation. So we're interested here, we're replicating the result of the PLS 10-minute group of the previous experiment. And we're interested in looking for what's happening here during the night. So this different sleep measure, we compare the BMA session with the control session. It's a % change between the BMA relative to the control session. Okay, first of all, we look for sleep spindle density, the density of sleep spindle. We separated fast spindle that they have an inherent frequency between 12 and 16 Hertz, and slow spindle that they have a frequency between 9 and 12 Hertz. And we saw that the density of fast spindle was increased in only during NOREM 3. So in the BMA session is higher than the control session. We see an increased % change, specifically during NOREM 3. So we also looked for the topography of this fast spindle aspect. We see that the fast spindle are occurring mostly in the centroparietal region of the brain. As we look for the % change between BMA and control, we see this interhemispheric modulation. So we see that the left hemisphere has an increased density of fast spindle. Specifically, remember that the left hemisphere is the contralateral hemisphere to the training hand. They train with their right hand. And we can see that there's a difference, significant difference between the left and the right hemisphere. On the other hand, just for knowing, for slow spindle, we know that the topographic distribution is in frontal region. We don't see any modulation of the density between the BMA and the control session. Didn't see any difference. So we see that only fast spindle are increased mostly in the left hemisphere, the contralateral hemisphere. So according to what is seen for declarative memory, we look for the coupling between fast spindle, because it's where we see the change, and slow oscillation. First of all, we look for a slow oscillation, and we saw that the density of slow oscillation is not modulated between BMA and control session. We don't see any difference, but we see a difference in slow oscillation coupled with fast spindle. Also, we see that the topographic distribution is in centroparietal region, and we see that the density of slow oscillation coupled with fast spindle is increased in the left hemisphere, in the contralateral hemisphere to the trained hand. is in agreement with the previous result about fast spindle. And we see a significant difference between the left and the right hemisphere. Well, we see this modulation, and we ask about the memory. So is any relation between this change in density of low oscillation and fast spindle and memory retention, for example? Well, we correlated this change for the left hemisphere. Remember that this is during NORREN 3, and memory retention, and found that there is a significant correlation between the change in density of low oscillation coupled with fast spindle and memory retention. And this correlation is specific for coupled spindle, because if we look for uncoupled fast spindle, we don't see any correlation. So we can say that the increase in the coupling, or the density of coupling between slow oscillation and fast spindle, is benefiting the memory retention. Finally, we look for the phase relationship between those oscillations. So we see that slow oscillation are coupled with fast spindle. So you look for where is the spindle occurring during the slow oscillation. Here we can
sorry
see
to interrupt you. We just have about a minute left. So can you quickly wrap up the presentation?
Yes, I'm finished.
Thank you.
So the fast spindle is occurring around the peak of the slow oscillation. We can see a polar plot and see that for all subset, the spindle is occurring around the peak of slow oscillation, and is similar in the control condition. We don't see any difference in this modulation. So to wrap up, we can say that sleep benefit and even BMA, the proximity between sleep and learning, modulate the memory consolidation. We can say that BMA increases the density of fast spindle and also increases the coupling between fast spindle and slow oscillation. And finally, only the fast spindle coupled with slow oscillation predict long-term memory, suggesting that this synchrony maybe is relevant for the consolidation of VMR memories. So thank you for the opportunity, and thank you for my lab, for Valeria, my PI. And here is the information if you want to reach out and keep talking. Thank you.
Again, thank you. You can hear my applause on behalf of the attendees. Thanks for the excellent talk. While we are waiting for questions, I have a question about like one of your findings, that visual motor adaptation that led to the increase in fast spindles. Do you think is that because there is a learning component in there that's related to this increase in fast spindles, or is that anything specific to like the motor activity that's why you see that the increase in spindles? I'm just wondering, like, if I have a different learning task, would you still see an increase in spindles?
Yes, we know that, for example, for MSL or motor sequence learning, there is reported some increase in spindle, and specifically for fast spindle. So we think that it is about the learning component and not just the motor. We have a motor control, so we're comparing both conditions, and we see that specific for the session that they learn to adapt to an actual perturbation. So we think it's about the learning and not just the motor.
Got it. Thank you. So we still don't have any questions. So if other panelists have a question, I can you can unmute yourself and ask questions. If not, like, I just want to know, like, what are your next steps with this? Where do you think you're going to take this result forward?
Well, we're interested in know more about the mechanisms, about the consolidation. We know about we EEG, we only see the EEG, the oscillation and spindle. What we're interested about what's happening in subcortical structures. We know about the clarity memory, that the hippocampus is maybe involved. We also know about our recent publication of our lab that for another kind of learning, the motor skill learning, the hippocampus is also involved in the acquisition of the memory, of the ability. So we're interested in go further about the role and the mechanisms that are in the subcortical structure.
Great. Thank you. Thanks for the wonderful talk.
Thank you.
All right. Next, we have Miguel Navarrete, who I believe is in Bogota now. I'm like the affiliation says Cardiff University, so I'll just let him take it away.
Yeah. Good day, everyone. Yeah, I'm currently in Bogota, but this is a work that was developed basically in Cardiff University in the lab of Penelope Lewis in Kubrick. So basically, previous panelists, they already give a nice explanation of what is sleep and basically non-REM sleep. Just to summarize, we know that non-REM sleep have the maximal activity of a slow EEG frequencies. So look at this hypnogram. We have the on non-REM have this nice detection of spindles and slow oscillations, and we know that they are critical for memory consolidation and neuroclean of toxins. Now about REM sleep, is characterized by the rapid movement of the eyes and is characterized also by the low muscle tone, a mixed EEG frequency. But we don't really know what's happening during REM sleep and basically what is the function of REM sleep in memory consolidation. That's what we are very interested in. So the problem is that evidence of REM sleep and memory is very inconsistent in human research. There are some previous works that say that memory, basically from procedural memory components are related to increasing of time in REM or correlated with the eye movements, but it's not really clear and how what is happening there. There are also different hypotheses. One of the hypotheses that the interleaving of REM and REM sleep may promote complex and analogous problem solving. But as I said, there is not really any clue what's happening there. So we wonder whether studying the memory function of REM sleep is problematic because it's very hard to detect any patterns from the REM sleep as compared to non-REM sleep. So we were thinking maybe if we want to study the memory function of REM sleep, we want to control the REM period without breaking the sleep continuity. But this is something that is not new. Basically, from the 80s until the years 2000, there were several studies looking at the stimulation during REM sleep. Basically, they showed that by applying very short click sounds during REM, there were some changes in the time of sleep, of REM sleep, changes in the density of saccades, and also changes in the phasic and tonic sleep. But there is also very interesting finding here is that with the sound stimulation during REM, there was an increasing of the spikes of PGO waves densities in the bones. This is in cat studies. Nevertheless, since the years 2000, there were not much studies about that. It was completely forgotten about this kind of stimulations. So we wanted to know with the new findings of memory consolidation, how can we use this technique of using sounds to control REM sleep. So what we knew from these studies is that stimulating REM sleep modifies sleep patterns from humans, cats, and rats, that the number of REM events decreases with the stimulation. The REM stage duration was increased in some of those studies, and the REM stage was also a little bit more segmented. But now we wanted to know whether applying the same technique, we could also have any changes in the cognitive performance after sleep. So what we did was applying a counterbalance protocol in 20 people. And basically, in one of the nights, we don't do any kind of stimulation. One of the other nights, do the stimulation, REM sleeps. We take all the REM sleep periods, and we only detect the eye movements, and we apply very short sounds, look to the saccades of the eyes. Also, we provide two tasks before and after the sleep, and for 3 different tasks. The first is a physical psychomotor vigilant task, which is very common in sleep studies. Secondly, a visual discrimination task and a mirror tracing task. these two tasks, the screen task and the mirror tracing task, had been previously related to the in any way to REM sleep. So what we found is that the stimulation during REM decreases the number of saccades, as the previous studies showed before, but we didn't find any changes in the time in REM sleep. We found a very short increase of efficiency of sleep, but I mean, this is still very, it's very close to not being significant, so we have to be attention of that. And okay, so for the EEG, of course, we found ERP. Basically, the stimulation decreases and the activity of the EEG basically in parietemporal areas. And we look at the frequencies, we found main factors. We found that there is increasing of beta activity after the stimulation, around 200 milliseconds after stimulation, and also a decrease of theta activity in after around 1 second after the stimulation. This increase of the beta activity is maximal in the central areas, whereas the decrease of theta activity is maximal also in frontal central areas. But what's happening with the behavior, right? Okay, so for the PBT, we didn't find like a very huge changes in the PBT, which is quite good. And also at mirror tracing task, we found that the stimulation during REM saccades decreased the change of overnight performance of the task. Basically, people who were not stimulated were better in the mornings after the stimulation, after non-stimulation, whereas people who, the nights that were not stimulated, the changes were not significant, were not that great. Now, we look at the sleep architecture and the behavior of the mirror tracing task. We found that when we don't make any stimulation, there's a great relationship with the NREM and REM sleep stages, whereas the stimulation disturbs completely the relationship between the behavior and the EEG. When we look at the VDT, the visual discrimination task, we found that the stimulation increased the overnight performance of the task. That is to say that the people were doing better the task. This task is very interesting. Just to those who are not very related to this task, people need to identify like text patterns in a screen, and this is getting faster and faster and faster. So when they're very good, they can really identify these patterns in a very fast way. So we found that the stimulation increased the performance of this task. Now, what happened with the EEG and the task? First, we found that the proportion of phasic REM, that's mean the proportion of REM, the time in REM where people is having like high density of saccades, is directly related to the performance of the task, the change of the performance of the task. And when we look at the EEG activity, the ERSP, we found that the increasing of beta activity was also related to the overnight change of the performance of the task. So that increasing of this beta activity here and was correlated to the task, whereas the decrease of activity didn't have like large effects on the performance. So what can we conclude from here? First, with our experiments, we corroborate that we could make any kind of acoustic stimulation to modulate REM sleep, not maybe in the time of REM, the time spent in REM, but mostly in the underlying mechanisms going there. We interestingly found that there were divergent outcomes of the cognitive performance. So basically, we have an increase of one of the tasks, the visual discrimination task, where a decrease of the mirror tracing task. And also, we believe that the decreasing of eye movement could serve as a mechanism of protection to prevent awakens, but this is still very, very speculative. We need more experiments to try this. And finally, this is a very broad talk. we want to know what's happening with the BDT. And just take from the literature, we know that PGO waves are concurrent with saccades during REM. Then also the PGO waves have been proposed to be related to synaptic plasticities. And also we know that the stimulation during REM increases the density of auditory stimulation. Sorry, of the PGO wave density. So basically, and this is a very new paper. The Tameki et al. Demonstrated that REM sleep has more complementary patterns with non-REM sleep, where basically non-REM sleep was increasing plasticity, whereas REM sleep was kind of stabilizing learning. So it could be that with REM sound stimulation during REM, it could help to stabilize the visual memories, possibly by increasing the PGO density. But this is a very speculative hypothesis that we propose, and it needs to be further research. I would like to thank all people in NAPS Lab and in Kubrick. Thank you very much, and happy to hear about your thoughts.
Thanks for the excellent talk, Miguel. We have some questions. Let me just read them out. So the first one is from Karen. Isn't there literature that actually eye movements increase arousal threshold and it is decreased in tonic? Also, would this mean that increased PGO leads to fewer eye movements?
Yeah, I mean, we don't know yet. Yeah, basically, we know actually that in a spontaneous way, PGO waves are very related to saccades, eye saccades, and I'm not quite sure whether they're related to the arousals. I know that PGO waves could be also related to the as a response of any kind of visual stimuli, but not necessarily this stimuli leads to an arousal. Yeah.
The next question is from Aishwarya. What type of auditory stimulation was used? And are you going to perform more cognitive studies with varied stimulation types?
Yeah, I mean, this is we use a very simple protocol. Basically, it's a very short click of 50 milliseconds of peak noise. Yeah, that's the idea. We would like to know what's happening with other type of stimulations. But looking at previous literature with stimulation during sleep, we believe that auditory stimulation could be the best way to do it, because it's kind of not invasive, and also with also previous studies with non-REM sleep, the sounds are loud enough to not get the people awake, but to have a huge cortical response.
We have one more question from Karen. Do you think it could be that stimulations increased tonic REM and decreased phasic? So maybe the mirror tracing depends on the phasic, but the visual discrimination depends on the tonic.
Yeah, at the beginning I thought that, to be honest, but when with the stats, we found no difference on time spent on phases of tonic sleep. So basically, no, that's the answer for that.
Got it, thanks. So if we have no more questions, and I think we're right on time, I'd like to thank the panelists again, and also for attending, and all the attendees for sitting through the session. That's about it. And please feel free to share information about this conference through your social media channels. And we'll see you again. Thank you.
Thank you.
Thank you.
Bye.
Bye-bye.
See you guys.
For which I have two images here of EMs that are pseudo-colored in blue and red to represent temperatures, to understand that. And we use a behavior that has been understood now for like 30 or 40 years. It's called thermotaxis. And thermotaxis refers to the capacity of these animals to move to a preferred temperature. So it turns out that C. elegans, unlike us, does not have an innate preferred temperature. Instead, as poikilotherms, it can be taught a temperature by being fed or being put on favorable conditions at that temperature. And so between 15 degrees Celsius and 25 degrees Celsius, it can learn to prefer one of those temperatures. So for instance, if you take animals and you rear them at 15 degrees Celsius in the presence of food, and then you put them in a temperature gradient without food, you're gonna see, and these are worm tracks, that they're all moving towards the left side of the plate here, which represents or corresponds to the colder temperatures, because the animals are seeking that temperature at which they were last fed. Now you can take these same animals and retrain them. It takes them about 4 hours to reset their preference, and retrain them to now in the presence of food at 25 degrees Celsius, and you put them in the same experiment, in the same gradient. I have pseudo-colored the tracks here in red just to represent the previous experience, but you can see the animals are now moving towards the right side of the gradient. So what I'm fascinated by is what changed in the brain of these animals, that they're isogenic animals. Second, they're seeing exactly the same experimental conditions in these two experiments. The only variable here is the temperature, yet something changed in their brains that enabled them to respond differently based on their previous preference, which was learned. So in other words, how do animals remember? How do these animals remember the temperature that they want to go to? And what are the cellular and molecular substrates of memory? So my aspiration is to cut across scales and bring this behavioral understanding to the level of the cell biology of the synapse. And I'm gonna be telling you today 2 short stories that were both done by a postdoc in the lab, Josh Huck. He was the primary driver of these studies. The first one has to do with what is temperature? I mean, we know it's a physical stimuli, but what does that represent to the worm? Like how is it encoded in the nervous system into interpretable neuronal signals? How does the worm sense it, and how is it represented in the nervous system of the animal? And second, what is the cultivation temperature memory? How is this memory encoded? How is it retrieved? How is the memory compared? So the animal has to be comparing the temperature that it's at with the temperature that it wants to go towards. So how does it do that? And I'm going to be showing you data that contributes to our understanding of these 2 questions. We haven't answered all these questions that I'm presenting here, but we have some of them answered. And I'm going to be showing you that we identified plasticity mechanisms that act in vivo and at a cellular level, actually at a synaptic level, to transform complex sensory information like temperature into a behavioral preference. That's what I'm going to be talking to you about. So let me start by introducing the neurons that are actually responsible for this behavior. So we have the connectome in C. elegans, and by beautiful studies done by primarily the Mori group in Japan, but also a number of other groups here in the States, they identified the circuitry that is important for this thermotaxis behavior. And one of the protagonists of this behavior is this neuron that I have here represented with this image. This is the cell soma of the neuron. This is the axon, and this is the dendrite. This is an image that we're taking in a live animal. It's immobilized, but it's a live animal. And I have it here as a placeholder to explain an experiment that we're gonna do in this rig, where we can see with GCAM6 the responses of that neuron to temperature. This is the main sensory cell for temperature in C. elegans. It's called AFD. And with the help of Aravi Samuel at Harvard University, we developed this rig in which it's very simple. It's a PLTA that allows us to arbitrarily give a temperature stimuli. Now the animals were held at a given temperature, and you're gonna see that holding temperature is gonna be very important. This is a placeholder. I'll show you the data in a second. And we give them these sinusoidal waves of temperature. So here we're seeing a live animal is gonna be moving, so the nose is gonna be moving, but I want you to pay attention to this area. Boom, there's the calcium signal. And every time that now the temperature increases, you're gonna see an increase in calcium. I'm gonna play this movie one more time. And I wanna draw your attention to the fact that there are no responses below this threshold, which is the temperature that the animal was at before. Once it hits it, it's like a sing-along. Every time the temperature increases, the calcium responses in this neuron are seen. So doing this type of experiments, we determined, and these were observations also made by other colleagues that I'm listed here, that sensory calcium signals mirror changes in temperature. But they only do so above a threshold or holding temperature. So the temperature that the animal was at right before the experiment. And one of the initial questions that we had was, does this threshold change? Is this static at 20 degrees, or can we move it based on the previous experience of the animal? So here's another experiment, another way of doing the experiment that I just showed you. Instead of the sinusoidal waves, we're gonna do a linear ramp. And then when it hits the temperature that the animal was at, boom, it lights up the neuron. Again, it lights up the neuron because we have a calcium sensor there. So it's sensing neuronal activity. And if you do this experiment for many animals and you rear the animals at 20 degrees Celsius, you're gonna see that most animals respond around that temperature. If you then change the rearing temperature, say to 15, you're gonna see that the animals respond to colder temperatures, not quite 15, but colder temperatures. And if you now rear the animals at 25 degrees, you're gonna see that the animals are gonna respond to warmer temperatures. Now, this type of experiments, this is our data, but this type of experiments were done before by colleagues in the field, and it led to a hypothesis because these type of responses correlate very well with the memory of the animal, essentially with the preference of the animal, meaning that the animals that are reared to respond that are reared at 25 degrees and move towards the warmer temperatures, they tend to respond to warmer temperatures. The ones that are reared to go towards colder temperatures, they tend to respond to colder temperatures. So this led to a hypothesis that these responses correlate with the temperature memory. Now, when we started working on these studies, this was the prevailing framework of thought in the field. But we did a number of experiments that helped reframe what these responses are, because it turns out that they don't correspond to the memory. They correspond to something else, as I'm gonna be showing you. So the way that these experiments were done by us and by others was that we will train the animals for several hours, up to 4 hours, and then we will do these two experiments simultaneously. We will look at their preference in terms of the behavior, and we will look at the responses of the sensory cell, like at what threshold the sensory cell is responding. But note that the animals were held at the same temperature for 4 hours. So these responses could represent the memory, which is the temperature at which the animals were trained 4 hours before, or the temperature that the animals saw right before the experiment. And we couldn't deconvolve those two possibilities from this experiment. So we decided to do a simple experiment in which we trained the animals for 4 hours to prefer a temperature, but then we took all those animals and we held them at the same temperature for 30 minutes. Now, it had been known since 1975 that it took 4 hours for the animals to retrain. So if you only held them for 30 minutes at the same temperature, the animals will still prefer perform the preference that they learned 4 hours before. But the question that we had, so this was known, but the question that we had was the sensory cell. Is it responding now to the memory, meaning this preference, or is it responding to the temperature it saw right before the experiment? And it turns out that the sensory cell, the threshold, is responding to the temperature that it saw right before the experiment, not to the preference that was learned 4 hours before, which leads to this behavior. If these animals were actually resetting the memory based on these responses, they will all be behaving the same because they're all responding to the same temperature. So the sensory threshold of the sensory cell cannot represent the memory. So this reframed how we were thinking about the sensory threshold in the sensory cell. It correlates with if you do this type of experiment, it correlates with this training temperature because you haven't changed it. So right before the experiment, you still have the same temperature, so that's why you see these responses. But if you actually change the temperature right before the experiment, you get different responses. So what I'm telling you is the sensory cell is responding to the temperature that the animals saw right before they were tested, while the memory, it's encoded somewhere else. So then the first question that we had is, what do these sensory responses represent? Because this is the primary sensory cell that is telling the animal what temperature is sensing. So what do they represent? So we decided to do the following experiment. So you can think of the data I just shared with you. Another way of representing it is like this. If you train animals at 15 degrees and you don't shift them from that temperature and you just examine the threshold, you're gonna see that they're responding around 15 degrees or 20 to 20 degrees, or if you train them at 25 to 25 degrees, because there's no shift. So the temperature they see right before the experiment is gonna be the temperature at which they were trained. However, if you also train them at these same temperatures, but then right before the experiment, you shift them to a neutral temperature for 30 minutes, you're gonna see that they're gonna be responding to a neutral temperature of 20 degrees, right? So then the question is, how fast can this sensory reshifting occur? So let's just look at the conditions in which the animals were trained at 15 degrees and look at how long it takes the animals to change from here to here. So if you plot these two data points, now I'm plotting them like this. So 0 minute shift, they respond around here, 30 around here. So we repeated the same experiment, but with different shift times, many times, and we created a curve. And this is how the curve looks. And here, once you have that curve, you can generate a tau, which is essentially gives you a sense of how fast the sensory cell is adapting to the temperature that it's facing. And you can see that it's 45 seconds. So it's actually much faster than the memory. It's not the memory, it's something else. It's encoding another aspect of temperature, and it happens very fast. So here I'm gonna summarize some of this data and other data I haven't shown you. So the sensory cell adapts its threshold very rapidly within seconds. It detects not absolute temperatures, but changes of temperature. So it's a temperature, it's a contrast detector of temperature. It tells the animal you're going warmer or you're going colder. It's not responding or encoding absolute temperatures. It's really a contrast detector. So then we have actually now, this is kind of like an aside very quickly to tell you about a system that I'm very excited about in my lab. We have integrated both the ability to do calcium imaging and the ability to see the animals performing behavior by using machine learning and some microscopy that we have adapted for this problem to be able to image the animals as they're thinking essentially and performing the behavior. And what you can do is you can essentially track the animal. This is the worm as it's moving. And you can see, this is another neuron. This is an interneuron that we have been tracking. You can actually visualize calcium responses in different subcellular regions. I'm very excited about that part. But importantly, because you know where the animal is, you know what it's experiencing, you can kind of create a track of what the animal had experienced. And once you have that track of what the animal has experienced, you can also correlate that with the calcium responses that animal has had, in this case, in the sensory cell. So as the animal is moving through the gradient, all of a sudden it hits a temperature that makes that hits the threshold, and you see a big increase in the temperature responses of the sensory cell. But importantly, and this is the part that I'm really excited about, you can take this experience and now replay it. So this is similar to like those experiments that you have two animals yoked, where one experience is being replayed to another animal, kind of similar, but across time, because you're doing it with animals that were tested at different times. You can take this experience and replay it to an immobilized animal and see how it's actually responding. And regardless of what the cultivation temperature was, these immobilized animals always respond very similarly to the freely moving animals or to siblings that have different cultivation temperatures. The importance of that result is that what that is telling us is that these responses in the immobilized animals of the sensory cell, they're actually representing what the animal is experiencing, not the previous training temperature, consistent with the results that I had already shown you. So how are these animals sensing temperature? So it turns out, I told you that this sensory cell is a fast adapting contrast detector, and this was actually really neat. So this is a summary of a cartoon diagram of molecules that are important for this ability of these animals to sense temperature. There are a number of labs that have contributed molecular genetic studies that have resulted in the elucidation of these mechanisms. Now, if you look at these mechanisms, it's essentially a receptor that senses temperature with a cyclic GMP mechanism that gates a calcium channel that's a cyclic GMP activated channel. Now, what other system looks similar to this in nature? So it turns out our photoreceptors used almost identical molecular machinery to encode for also contrast. Our photoreceptors are also contrast detectors, but they're not contrast detectors of temperature like the sensory cell. They're contrast detectors of photons. So in our photoreceptors, we have photon sensors, but so that's different, photon sensor from a temperature sensor. But if you look downstream, it's essentially you have the cyclic GMP dependent mechanisms. And what this is, think of it like a nonlinear amplifier that enables the animal to very quickly sense contrast. And in the context of the fast adapting abilities that I shared with you earlier, what this animal is essentially doing is that it's moving through the gradient and is quickly adapting its perception to be able to sense changes in temperature. So I like to tell the students that This reminds me of those games of Marco Polo that kids play, for example, in a pool, where the kid doesn't really know where they are in the absolute context of the pool. They just know where they are with regards to a target. Here is not the target so much as it is where they were right before they sense the temperature. So what does this temperature changes, what does temperature changes signal mean to a worm? So this is how we think it's working. So we think that temperature in the context of this neuron is represented again as a change because it's sensing contrast and changes through time. So the sensory neuron, if the animal is moving towards the warmth, we are representing the sensory neuron here, then the sensory cell is going to respond. We're representing that as an increasing green signal here when calcium rises. And if the animal is moving towards the cold, then the calcium levels are going to fall. And so the responses of the cell, and the cell is constantly adapting again that response as it's moving through the gradient through time. So what this is essentially telling the animal is in which direction it's moving in the gradient with respect to the direction that it was at right before it sensed that temperature. So the sensory cell is rapidly adapting thermal change detector, and these changes indicate the direction of migration in a thermal gradient. And we're actually now working to model this, to really to understand this very thoroughly, to model this to see how these animals are actually representing that sensory space. But this was a neat finding, but it doesn't answer the question of what is the memory, like how is the behavioral preference encoded. So we moved on to understand that by looking at the connectome. So let me first introduce you to the type of data I'm going to be showing you. So the sensory cell, which I've been talking about, I'm going to present it here. Again, what you can do is this kind of like increasing temperature, and then you get a response. And so you see the increasing temperature, you get a response around here, around 20 degrees. And then you can represent that response in calcium as a heat map. So when it's cold or when it's blue, it's essentially the animals haven't responded. And then when it gets red, it means that the animals responded. And you can do this for many animals. Each of these rows represents a single animal. And you can see you get very stereotyped responses around a given temperature. If you align these to the slope of the increasing temperature, you can see that they correspond nicely to 20 degrees, which is the holding temperature. And you can also have an aggregate like this with a distribution. So this is how I'm going to be representing the calcium responses from here on. And if again, like if you do it for the sensory cell, you get very stereotyped responses. And our question was, the first question that we asked was, okay, so what is this cell? Who is this cell talking to? So it's encoding now a change in temperature. It's adapting, so it's keeping a dynamic range as the animal is moving through the gradient. But then who is it talking to, and where could the memory live? So we were lucky that, of course, we know the connectome, and the main postsynaptic partner for this presynaptic sensory cell, it's known, and it's this cell here. The name is AIY. You don't have to remember the name, but I tell you the name just to communicate to you that we know specifically what cell we're talking about. And we had tools, or we developed tools to be able to also visualize the calcium response in this cell. So again, the sensory cell, you get very excellent reproducible responses around the temperature, around the threshold, as I have been showing you. What happens with this postsynaptic cell? And I want to point out a few things here. The first one is that the responses are not identical to the sensory cell. So you can see that the sensory cell kind of holds the calcium levels. This postsynaptic cell has a different type of kinetics in terms of the calcium patterns. But the other thing that we observed, and I use this as a teaching moment for the students, because we observed this and we ignored it for about a year and a half. These animals here do not respond. So we spent a lot of time trying to understand the memory in the context of the animals that were responding, and we got nowhere. Because it turned out that the important part for the paradigm of how this memory is encoded is in these animals that do not respond. I'm gonna pivot and present it in a different way in the interest of time, but I wanted to make a point out of that. Like, be careful with your assumptions, because what's really dangerous in these experiments is not what you know that you don't know, but what you don't know that you don't know. So that is if you remember one thing, remember that. So we know these responses are coming from the sensory cell because we can do in genetic ablations. And if we get rid of that sensory cell, we get rid of those responses in the postsynaptic cell. So again, these are the responses in the postsynaptic cell. So to summarize the type of responses that we're seeing in this postsynaptic neuron in response to temperature, I'm just going to very quickly present them in these bullets. This postsynaptic neuron has multiple temperature responses. I'm not going to discuss all of them, but I'm going to discuss the one that is dependent on the sensory cell, which is due to increases above cultivation temperature, which is this graph that we see here. It's similar in terms of timing. These responses are similar in terms of timing to these ones here, and they are dependent on this one. If you get rid of this cell, you don't get them. But you get fewer, we're interpreting this as fewer response frequency. Although these are different animals, we're interpreting it as fewer response frequency than the sensory cell in the interneuron as compared to this cell. So eventually we came up with this hypothesis, it wasn't the hypothesis that we had at the beginning, but eventually we came up with this hypothesis, which is that this sensory cell always responds to increases in temperature, but then sometimes that, for example, in these animals, that is transduced to the postsynaptic neuron. So when temperature increases, this response and it gets transduced. But there's also a population in which you get these responses to increases in temperature, but that doesn't get transduced to the postsynaptic neuron, which are these animals here. And then the question that we had was, does this matter? And does this probability of response that we see here in the postsynaptic neuron in response to temperature varies under different conditions like cultivation temperature, like the previous memory of what the animal wanted to do. So we examined this, and we examined this by training the animals to like different temperatures and then looking at the calcium responses. And indeed, when we trained the animals to want to move towards 15 degrees Celsius, we got fewer responses in the postsynaptic cell. Although, again, the sensory cell is responding every time we increase the temperature, it responds. But if the animals want to go to the cold, when we increase the temperature, what happens is that the sensory cell responds, but that doesn't get transduced to the postsynaptic neuron. While if the animals want to get moved to the warmth, when the sensory cell responds to warmth, that gets transduced with a higher probability to the postsynaptic neuron. So this is a quantification of those responses. So we interpreted this to mean that the probability of responses were changing depending on the previous experience, and we asked, does this matter? Does this really represent the behavioral preference? And this is a hard question to answer, but we were fortunate that in our system, we have mutants that we could use to tackle this question. And these mutants are actually exceedingly cool. Like these were part of the work that inspired me to examine these questions that I'm presenting today. And they were originally reported by Ikue Mori in this paper, this beautiful paper, Okoshi et al. And essentially it's a mutation in a protein that is conserved. It's called PKC. In vertebrates, it's also involved in presynaptic plasticity and memory. In C. elegans, when you get rid of PKC, the animals, regardless of what temperature you train them at, that's what I'm representing them here in gray, these mutants always want to move to the warmth, and they do so to their death. So they will continue to go to the warmth until they die. But the incredible thing is that if you put a wild-type copy of this gene in a single sensory, in a single cell, which is the sensory cell, you don't affect the sensory responses. They actually look fine in these mutants, but you do affect the preference. So meaning that now these animals, when you put the wild-type copy of this gene, although this gene is lacking in the rest of the animal, is only present the wild-type copy in the sensory cell, now these animals respond just like wild-type animals. And moreover, this kinase has a really interesting feature, which is that it has a autoinhibitory domain that closes it upon itself like a Pac-Man. And what you can do is that you can introduce point mutations to leave it open. So now the kinase domain is always open, phosphorylating whatever molecule it encounters. And in that gain of function mutants, the animals do exactly the opposite of the loss of function ones. So now they always want to go towards the cold. So what I'm telling you is that in C. elegans, we have a single molecule that acts in a single sensory cell, but it doesn't affect the sensory ability of the cell. And that single molecule actually affects the preference of the animal independent of the previous experience. So besides the fact that it's mind-blowing and fascinating, this was a really useful reagent, because if you think about it conceptually, what this reagent, what these mutants allow us to do, it's separate experience, which is usually what drives the behavior, from preference, because these animals have the same preference regardless of the experience. They're always are going to want to go to the cold or the warmth regardless of the previous experience. So we can disentangle the experience from the preference. So we could ask, is this postsynaptic cell responding to the behavioral preference or to the experience? So we did the following experiment. We kept the experience the same, but we have different preferences because of the different genetic backgrounds. And then we ask, how is this cell responding now to temperature? And we observed that actually in this gain of function mutations of PKC, in which the animals want to go towards the cold temperature regardless of the previous experience, they actually show very few responses. And in these ones that they want to go to the warmth temperature, they show many more responses. This is the quantification. So what that told us is that this type of responses in the postsynaptic cell, whether the sensory cell transduces to the postsynaptic cell or not, correlates with the preference of the animal, not with the experience, because we're getting the same thing with the same experience in these mutants. So it turns out that I'm not gonna show it here in the interest of time, but this conserved kinase actually functions within the sensory cell, but in gating presynaptic plasticity. We did electrophysiological studies to show that. But the concept that I want to leave you with is that this started to show to us that within this single cell in C. elegans, we had two plasticity mechanisms. We had sensory adaptation, which was the first part of the talk, that allows the animals to keep a dynamic range and sense contrast and tell the animal, okay, you're going warmer, you're going colder. And that ability of the animal to know if it's going warmer or colder, the adaptability as the animal is moving through the gradient is critical for the animal to be able to keep a dynamic range and know where it is. But then in the same cell, so we're kind of getting to a cell biological level of resolution here. At the presynaptic site, you have a gating mechanism that decides whether that signal gets transduced to the postsynaptic cell or not. So this conserved kinase is mediating presynaptic plasticity. And if the animals are moving to the warmth and the system wants the animals to continue to move to the warmth based on a previous experience, then that signal gets transduced to the postsynaptic cell and the animals keep on going to the warmth. The converse is also true. If they are moving to the warmth, but they shouldn't be because they want to move to the cold, that signal gets blocked. So there are two plasticity mechanisms acting within a single neuron to essentially both encode and interpret this temperature signal based on a previous experience. So again, this is like this sensory cell responds all the time, but sometimes it gets blocked. And we came up with a hypothesis here, and this was a crazy experiment, but it worked, and I'm really proud of it. So we thought, if this is true, if we were to sidestep that presynaptic gate, if we were to short circuit and somehow couple these two neurons, we could make some predictions. So the first one would be, of course, if you couple them, then every time this responds, which is every time the animal is going to go to the warmth, this cell is going to respond. The second prediction is going to be that I'm telling you that if this cell responds and this cell responds, that the system interprets that to mean we're going warm and we want to go warm because we learned that warmth is good based on a previous experience. So even if we had trained the animals to prefer the cold, if we had this type of manipulation, then we will re-encode the preference over the memory, re-encode the preference, and the animals will always want to go to the warmth even when they were trained to want to go to the cold. So again, these are the predictions. The responses in the postsynaptic cell will not depend on the cultivation temperature experience. This cell is always going to fire when this one fires. These increased responses should alter the preference. We should be able to recode the preference with this type of manipulation, this short circuit, and we should be able to suppress the gain of function phenotype of PKC that actually blocks this communication. So how can you do this experiment? Well, we were lucky that around that same time, Bill Schaefer over at the MRC had published a paper in which he used vertebrate connexins to rewire circuits in C. elegans. And we saw that paper and we were like, this is what we need. So to understand this experiment, it's important to understand. So you have, of course, chemical synapses, which I've been talking about, but there are also electrical synapses, which are facilitated by gap junctions. These electrical synapses in vertebrates are encoded by proteins called connexins. Now, in invertebrates like C. elegans, there are also gap junctions, they're also electrical synapses, but they're not encoded by connexins. C. elegans does not have connexins. So vertebrate connexins are inert to the proteins that form the gap junctions in C. elegans, which are called inexins. So you can take the connexins from vertebrate, put them in C. elegans, they're inert, and you can re-encode or re-engineer electrical synapses to bypass the regulatory mechanisms of the chemical synapses, which is what we did. So again, if you train animals to want to go to the cold, you get very few, you get excellent responses in the presynaptic, in the sensory cell, but you get, those responses are actually blocked. They don't make it to the postsynaptic cell. So if you look at the postsynaptic cell, you get very few calcium responses. And the animals go to the cold. If you engineer this gap junction, now if you look at the postsynaptic cell, boom, you get every time that this cell responds, this is going to respond. So this block no longer exists as we predicted. Actually, if you look, I don't know who was paying attention to the kinetics when I presented it of this one, but if you look at the kinetics, these kinetics are much more similar to these responses to temperature than they were to these responses to temperature. And it is because we have engineered this electrical gap junction. So what do these animals do? All these experiments are done in vivo. So what is the preference? And it turns out that you can train those animals to want to go to the cold, but if you engineer this gap junction as we predicted, now these animals, you have rewired the preference, and these animals want to go towards the warm side. So it is this simple. It is this simple in terms of this block. You have a gate here, which is probably encoded by it's encoded by the memory of the animal. We don't fully understand how that memory is encoded, but we know that it's expressed through this blocking that is mediated through this protein, this kinase called PKC. So if you put it in the context of the cell biology, which is our aspiration, essentially you have a contrast detector. This is the cell soma, axon, dendrite. You have a contrast detector here that's sensing temperature changes and then gating the communication with the postsynaptic cell via a PKC-mediated presynaptic plasticity. So we're very excited that we have now a molecular foothold to understand this. And I wanted to, I just wanted to, these are my last couple of slides. I just wanted to kind of present the concepts that we have learned and present it in the context of the behavior of the animal. So if these animals, take these animals, for example, they're moving towards the colder side of the gradient. But if you actually take one of those tracks and you look at it, I mean, we can also look at the responses in the presynaptic cell and the postsynaptic cell. But if you take one of these tracks and you look at it, this is one track, and then you break it down in the decisions that the animal is making. What I want to highlight here is that the animal is not just beelining it towards the cold side. Sometimes it goes towards the warm, sometimes it goes towards the cold. So there's a probabilistic element to it that where you have a convolution of a number of decisions that the animal is making, which include travel direction of the animal and duration of time of travel. So if you take then these sub-decisions and you plot them, they end up going to the cold because most of the time they spend them moving towards the cold. But there was some time that they were spending and moving towards the warmth. And we believe that these decisions are actually mapping onto the postsynaptic cell in these calcium responses, or this probability of calcium responses, in which it's telling the system, yes, keep on going that way, or no, turn around, you're going in the wrong direction. And what we want to do is, across scales, map these decisions into the cell biological components of the synapse and understand how these single cell activity patterns and molecular signals map onto the decisions, like these high level decisions that the animal is making, like the direction of travel and the duration of travel that we're observing in the tracks that the animal is making. And I don't want to make it much more complicated, but I wanted to mention that the animals not only move in the temperature towards the temperature that they want, which is what I show you, but once they find that temperature, they actually track it. They form isotherms. So they track it with exquisite precision. And we have additional mutants that we have identified in which the animals abnormally track temperatures that they're not supposed to like. And what's fascinating to us is that those mutants are actually mutants in electrical synapses. So now we're starting to understand not only the connectivity at the level of the chemical synapse and how the plasticity is encoded in that synapse, but then how another layer that we understand very little about, electrical synapses, how they encode changes in the locomotory strategies of these animals, like changes in single cells in the circuit that then transform themselves into changes in the locomotory strategies of these animals as they switch from going towards the temperature that they like to preferring that temperature. So this has to do with this concept of kind of like preference of behavior, like performing the right behavior at the right time. And it turns out that in these mutants that is off, and I'm very excited about examining that now. So with that, I'll finish by thanking you for listening to this talk and thanking the people in my lab that did this work. I want to again underscore a lot of this work was done by Josh Hawk. The freely moving rigs were built by Ernesto Cabezas Bo. The work on the isothermal movement and in axons, which I presented at the end, is led by Agustina Moril Porras and the rest of my lab and funding agencies and collaborators. Thank you.
Thank you very much. That was fantastic. As someone who studies humans, it's always astounding to me the precision with which you can study neurobiological mechanisms in these specimens. It's just such a great thing to see, the incredibly fine detailed work. If any guests would like to ask questions, please feel free to type them in the chat or enter them into the Q&A. I guess the question that I have right off the bat is, and maybe you touched on this and I kind of missed it, but how do you have a sensory neuron that is basically sensing contrast, but also allows the system to converge on a specific temperature? I can't in my head get it, right?
I think that is, I did not touch on it, and that is, there are a few questions that are the questions, and that's one of them. So the other one is, where's the memory? Because I think we have bypassed the memory, but we don't know where the memory is. So let me start with the question you're asking me. So yes, so so I told you two things that are that cannot, that at the level that we understand it cannot both be true. Like our understanding is incomplete. I told you that the way that these animals are moving towards their preferred temperature is that they're sensing contrast. They're sensing if they're moving colder, they're moving towards the cold or towards the warmth. But then you need to have another system that compares with the temperature that they prefer, number one. And number two, you need to have a system that tells you that you have arrived, because otherwise you'll always be moving towards the cold or always be moving towards the warmth, which happens. It happens in the mutants, the PKC mutants, that's how they are, right? So how do the animals derive an absolute temperature value out of a relative temperature sensor? And we don't know the answer to that question. This is, I find that incredibly exciting to think about it. And if people have ideas, I'd love to hear ways of testing that, but we don't know. I have some ideas, like maybe there's another encoder that works more like, like other temperature encoders like trip channels, they respond to temperature ranges. So it's possible that there are trip channels in C. elegans there are definitely trip channels in C. elegans, but it's possible that those trip channels are feeding absolute temperature ranges to this circuit to tell the animal, okay, you're in the correct range, and that's how that information is stored. That's one possibility that I can think of. Another possibility is that the animal somehow is, there's another aspect of the encoding that we're not fully capturing in our calcium dynamics that is encoding for absolute temperature. And I'm fascinated by that question.
Yeah, I really can't wrap my head around it. I mean, like humans are also notoriously bad at pinpointing exact references. So it's not something that we're great at either. But it's possible because we have more different kinds of sensors, they can sort of play off of each other.
Right.
Whereas if you only have one, I can't even.
No, so in humans, so yes, in humans, you have this kind of contrast happening at a circuit level, as far as I understand, right? There might be cells that also send, like these receptors that I presented today that sense contrast of temperature, they're actually conserved, they're present in mice, and in mice, they're also sensing temperature. So it's likely that those same receptors are conserved and work even in humans, but a lot of our temperature perception is derived at a level of this circuit. So here, what I found fascinating was the ability of a single cell to be able to create these derivatives, right? And compute a big aspect of the actuation of the memory. And now you can look at this, if you're a human researcher, you can look at this and be like, wow, it's fascinating that evolution will do that and maybe make sense for a compact nervous system like C. elegans. But on the other hand, it tells you what evolution is capable of. And with the right evolutionary pressures, I doubt that this is specific to C. elegans. You know what I mean? This is telling us what a level of computation at a cell biological level. If you tell this to a cell biologist, they won't find it surprising at all that cells can act as computers. But it's important for us as neuroscientists to keep that in mind, right? That you can have a lot of computation happening within a single cell. And it's possible that this is exaggerated for organisms with compact nervous systems, but it's also likely that our own cells are capable of such complex computations like this by integrating what we now, at least I thought of, plasticity mechanisms that usually live in different circuits or different components of these circuits. These are all living within the same cell.
Mm-hmm. Yeah, I mean, I guess I would say that the photoreceptors in our eye sort of are some combination of both. They've got like a sort of steady state, but they also have some amount of gain adaptation that works online due to just, you know, bleaching of photoreceptors. But there's also the like multiple levels of contrast adaptation at the circuit level as well.
Right.
And it's hard to like tease them all apart as well.
Yeah, we've drawn a lot of inspiration out of the photoreceptor work for trying to think about the encoding of information in the system. So I think that it's possible that absolute temperature values could be similarly derived. You know, there's some experiments that we could do that we haven't done. Like so because everything lives in this cell, you can imagine like a way of testing the hypothesis that I told you that it comes from a different circuit would be just to isolate the cell, which we can do, put a sensor for synaptic vesicle release, and then see if we can encode if we can alter the memory of this cell, but like by the readout will be not the behavior of the animal, but the ability, the release probability, and just do it in a dish and see if the cell has all the information to be able to derive the absolute temperature from that, right? Because our readout right now is here, but we're reading out cell biological aspects of this cell here. So if we were to move our sensors over here, which we can do that now with Glut sniffer and other sensors like that, then we could get to that question. We just haven't done the experiments yet.
Yeah, that sounds great. Yeah. I want to dip into the questions here as well to give the audience a chance to chime in. Viral Mystery writes, I'd like to quickly clarify something you said with the temperature-directed movements, cold directions and warm directions near the end. Do you think you're observing population codes in C. elegans? And if so, how do you think that works in such a small nervous system? And if not, what do you make of that?
So can you read? So they're talking about this here. Sorry, let me move to they're talking about this here. Yeah. And what is the question? If we're seeing population codes?
Population codes, right.
So I don't know, what do they mean by population codes?
Yeah, it's a good question. I mean, because you're talking about single neurons here, right?
Yeah. So I think we're seeing, what we're seeing is these are decisions of a single animal, and these are responses in a single animal of a single neuron, right? Mm-hmm. And what I was trying to say by comparing this with this, which we're actually getting much more granular information because we're combining them in the machine learning system that I briefly described in the middle of the talk. But for now, what I was saying is, if you look at these animals here, I think they represent response probabilities. So these are different animals because of technical issues. But let's think of them. Our assumption is that these represent at a level of a population, maybe that's what this person is asking, at a level of a population, the response probability of this cell. Well, we think if we were able to do it for a single animal, we will get a similar profile. If that is the case, then we think that's what's happening is that this represents decisions that the animal is making as it's kind of navigating the gradient, in which in some cases it's like warmer keep on going or warmer stop. And the warmer stop will make it go colder, and the warmer keep on going will make it turn. In this case, in which the animal wants to move towards the cold. The converse is also true for animals that want to move towards the warm. So what I was trying to do was map these decisions in the context of, these cell biological responses in the context of these decisions that the animal is making.
Yeah, I like the idea of taking population more literally in the case of C. elegans, where you could have hundreds or thousands of them in a dish and observe in real time the behavior of literally the population. Yeah.
So to do that, we so we could do that. We can do that at the level of locomotion, but we cannot do that at this level. So now we can. So now we have, I mean, we don't have the capacity to do like hundreds of thousands, but we can do several worms, definitely one worm, and follow it. And with machine learning, the importance of machine learning is that the cell is getting deformed as the animal moves, but you can extract all the information of the cellular responses by training a machine to identify the cell. So it gives you a wealth of data of both what the animal is experiencing. It's like an MRI, it's like an fMRI on a freely moving animal kind of thing.
That's awesome. All right, next question is by John Oyem, who asks, interesting talk. Does this encoder for temperature encode for pressure too? Is there any, do you know anything about the pressure sensitivity?
We don't know anything about the pressure, but I was telling Brad at the beginning of the talk that this encoder for temperature is incredibly sensitive. We're trying to understand why it has to be that sensitive. It's more sensitive than this is work by Miriam Goodman at Stanford, and we have also seen it in our own rigs. It's as sensitive as a sensor that a viper uses to hunt based on, like, infrared detection. So that's how sensitive it is. And why does a worm need that? Because a viper, you understand why it needs it, but a worm, why will it need it? And I think that what I have two hypotheses. One of them is that this is really sensing something very like a heat producing event that is minuscule, like light, for example, like photons of light. So that's one. The second thing is that it could be sensing, like the animals are essentially moving their heads left and right, just the way in which they move. They're capable of tracking very, very precise gradients as they're moving. Once they find the temperature that they prefer, they're capable of beautifully tracking very precise gradients. And maybe that's how that sensitivity of moving the head is giving them the dynamic range that they need to be able to track the gradient that they need to track normally in nature. Those are my two hypotheses. They're untested.
Right. Yeah, and when you do the gradient of temperature, is that by just changing the bath temperature?
So we have two LTAs at the two ends, and then we can calibrate them, and that creates a gradient across an agar plate in which the animals move. In the microscope, we have, when we're doing the visualization, we have a single LTA we can change through time.
But it's done by conduction in the water bath, not by radiant heat, for example.
We're doing it, I mean, it's done through like a current that runs through the LTA that is then linked to a radiator. And then it's heat that is radiating through a ceramic, that goes through a ceramic plate into an agar in which the worm is held.
Okay, right.
People have done it with water baths too. We don't use water baths.
Okay. Well, yeah, it's going to be incredible what happens with, I guess, as machine learning makes it easier to decode the population activity of larger numbers. Even you could just have and you could even I just love thinking about all the amazing things that are going to happen with this preparation. We've got 0 time, but maybe we won't cut off right away. Would you say that memory is not overwritten and encoded separately is the last question that pops up here?
Yes, I think that for sure. That's exactly my interpretation. So for a long time, I was thinking, I think now incorrectly, that we were recoding the memory. I don't think so. I think the memory is there, and what we're doing is bypassing the actuation of the memory. So if we could have a readout for the memory, which we don't, I think we'll see the memory just correctly encoded, but we are bypassing it in a way that the animal now displays a different preference, because our readout is the behavior of the animal. So the animal is displaying a different preference because it cannot actuate the memory the way that it's encoded. But now what I'm really excited about is now we have a molecular foothold, because we know that PKC is actually actuating the memory. And the molecular mechanisms upstream of PKC are known. For the aficionados, the geneticist and the biochemist, it's a second signal called DAG, which is produced by phospholipase, which is activated by G protein coupled receptors. So now we have a direct line of molecules that we can start testing to find what is the actual encoding of the memory.
All right, well, we're going to give time for people to head off to the next thing. Thank you very much for this fantastic talk. It was really wonderful to learn about this preparation.
It's a pleasure, and thank you for the opportunity.
All right. Bye, everybody. Bye, Danielle.
Bye.
talk about the way that we study these monkeys. And the work I'm going to describe today involves the coordinated effort of an enormous number of people. Just the tip of the iceberg is shown here, but I'll just say at the beginning, this is 3 labs working together. My lab in neuroscience at Minnesota, Jan Zimmerman's lab in neuroscience, and Hyun-Soo Park's lab in computer science. And then a lot of other people working really closely and really carefully along with us. Today, I'm going to focus on the work of Praneet Bala, Hao Zhengyu, and Yuan Yao. So here's a monkey. Here's 2 monkeys, and these guys are in their somewhat natural environment. This isn't exactly where they historically lived, but this is a field site in Puerto Rico where we like to study them. Here's a picture of some monkeys moving around, and we're following them with a video camera. And here's some more ethological research. We're watching these monkeys groom and tracking them with a video camera. And so what my lab and the other labs that I mentioned at the start of this talk are interested in is relating neural activity to behavior. And this is actually something a lot of people do, but one thing that really is unique about our perspective is we're much more focused, I think, than most other people on understanding behavior in its natural, in its ethological context. And so for me, for the last 10 years, but especially for the last 5 years, that's been a focus on taking behavior and studying it in a freely moving context, as opposed to a plexiglass chair, understanding how behavior works in something like the environment that these monkeys are in. Understanding the brain activity in freely moving monkeys is very difficult, but it's much more difficult than actually behavioral tracking. This is a system that Jan Zimmerman developed, and he's, as far as I could tell, in addition to all his other talents, has a degree in engineering. He built this really amazing system. And I think what Jan would tell you is, despite the technical advances in this system, that the behavioral tracking is much more difficult than the neural tracking. So I'm just going to talk about behavior only today. We've been working for the last several years collaboratively on this 3 co-senior author paper that just came out a couple months ago, where we introduced Open Monkey Studio, which is a tool that we developed for monkey tracking. I'm just going to briefly introduce it to you because it's critical for the rest of the brief talk that I'm going to mention today. Here's a picture of our system. You can see it's pretty cool. It's pretty awesome. It's an 8 by 9 foot. For Europeans, it's about 2.5 by 3 meter cage, and it's 8 feet tall. It allows the monkeys to move around in 3 dimensions. There's barrels they can crawl on. There's ropes. There's tire swings we can put in there. There's a bunch of cool stuff. There's interactive feeders that monkeys can work with. They can press levers. They can look at screens. They can get reward. They can get food reward, juice reward, and we're going to track everything that they do with 62 high-resolution machine vision cameras. Here's another view of the same thing. You can see the whole thing is painted chroma key green, which is a special green that was invented for Hollywood that maximizes our ability to segment things that we see. So there's this rigid exoskeleton around the cage that mounts all these cameras, and on that exoskeleton, we're going to mount the cameras, look at the monkey from all directions. And so here's kind of some of the raw data that our system takes in. This is one monkey, and this is a nice video because it illustrates a few things. One is that these animals move a lot in 3 dimensions. Monkeys are arboreal creatures, and they like to climb and they like to crawl, especially the young ones. And so we're gonna need coverage of the entire situation, and that means we need that number of cameras. It also illustrates that even with 62 cameras, we only get monkeys in about 20 of the cameras at a time, so about 1/3 of them. Open Monkey Studio, I'm not going to go through the details because they're already published. I'll just say the number one question I get asked is, how much data do we generate? We generate about 3 terabytes an hour. We usually do 4 hours of recording at once. And at the heart of it is a convolutional pose machine. A CPM is basically not too different from what Deep Lab Cut is. There are several of these out there in the academic sphere, and we use CPM for a couple reasons, but ultimately it's a similar thing. What really differentiates Open Monkey Studio is all the stuff that's on top of that. And the main thing that's on top of that is solving the annotation problem, which is really the most difficult problem. So let's see, why is annotation the big bottleneck? There's another cute picture of one of our guys crawling around in the ceiling of our cage. Monkeys are, as Jan Zimmerman likes to say, big balls of fluff. They don't really have articulated joints that are easy to identify. They have hair all over their body and often very long hair. You can't put markers on them, of course, but even markerless systems can't detect their joints. They move very rapidly and much more rapidly than most other tracked species relative to the size of their body. They have many more degrees of freedom. They move in 3 dimensions. So they have very complex and rich behavior. And I don't know how much any of you have worked with deep learning, which is basically the technique that we're using to do this. Deep learning is actually really, really poor at generalization. And you might not know this if you kind of read the news articles and you read Twitter, but it's actually quite brittle. And so deep learning is really good at looking through its archives for video of frames, annotated frames that resemble things that it's seen in the past, and resemble the things that are on the screen, and relating those to each other and using that to make a small leap, but it can't really do big leaps. And so you need to have a very rich database that's going to have all the rare things in it, and monkeys have a lot of very rare activities. And so solving that problem is going to really require a huge amount of work. In humans, the problem is solved by companies just pooling their resources, and it would cost us, we estimated, somewhere between $10 and $12 million to get a database for monkeys that's equivalent to what exists for humans, and we just don't have that kind of money. So basically, most of what's in the paper is a series of tricks and insights and hacks that we use to get over this problem and deal with a database that cost us about $80,000 to $100,000 to generate, so several orders of magnitude cheaper than humans, and get it to be as accurate as the kind of human databases. And the result of it is a system that compares well with Untitled. Does that mean I have 5 minutes left? Okay, I'll go really fast here. So the result is a system that can track 13 joints, and these are joints that we chose heuristically that we think are pretty interesting. But the first thing I want to talk about is what happens if you want new joints that weren't in there. And so this is Praneet, who was the leader on this Open Monkey Studio project, is now this is his follow-up project. And basically what he's doing is he's saying, what if you really wanted the right elbow? What if you wanted the ear, the left and the right ear, which gives you a rudimentary estimate of gaze direction? We could go do the same thing we already did to build Open Monkey Studio, but that would cost us another $80,000. We have to start from the beginning and have these images re-annotated. Can we, with a minimal annotation set, expand our system and get extra annotations essentially almost for free? This is very difficult with this standard 2-dimensional approach, which a lot of markerless tracking systems are. You know, just to give you an example, the distance between the butt and the knee is 60 pixels in this one image, and in a slightly different image, it's gone down to 8 pixels. And so you can't really solve this problem on the 2D representation. So you need a system that gives you a rich 3-dimensional representation, which our system is. And then once you have that, you can do this. In the interest of time, I'm going to skip over this. I'll just say the details of what we're doing here at a high level is we're building a system that can essentially teach itself a model of the skeleton frame of the monkey. It's going to teach itself how long each of the bones are in the monkey's body, more or less. And it's going to use that information because bones don't grow or shrink during movement, to infer where the shoulders or the elbows or whatever are. And so we can actually do something really impressive with this. So Praneet sat around one afternoon and labeled 568 frames. So that's 2,000 images, just a little bit over 2,000 images. Since remember, there's 62 images for each frame. He just picked 4 at random with the monkey in them. And with those 2,000 plus images, he was able to get really nice tracking of a new joint that was not in the original annotation set. All right, so moving forward, I'm going to go very fast because I'm running out of time here. Hao Zhang's project is trying to do even more key points. And so basically what we're doing is saying, what's the limit to what we can accomplish here with the number of key points? So he's going to have a system where he's going to segment. He's going to use a kind of a very standard neural network to segment the monkey from the environment with a few extra tricks that I could go into if I had more time. And the result of that is that he's going to come up with a system that moves a mesh computationally. It's sort of like putting a robe around the monkey, a detailed 3-dimensional mesh around the monkey. And that's going to eventually result in a representation of the monkey that we can figure out where points are from time to time with high precision, in fact, arbitrarily dense precision. So this is a 40,000 point mesh that we've generated, tracking where the monkey is, using this system that I don't really have time to describe. All right, with 1 minute left, I will just I will answer the single most commonly asked question that I get, and that question is, what happens if you don't have 62 cameras? What happens if you're this guy, which is actually me 12 years ago, with one camera out in the field? How do we solve that? And really the question is, can we leverage the fact that we have a very large database now of 2D images with joints projected onto them and three-dimensional representations, and the relationships between those, which are veridical and highly validated, to start inferring from photographs and from videos exactly where on the in space, or if not that, at least in the screen, where those animals are. And so we're going to basically leverage what we've already done to start doing tracking with minimal number of cameras, even down to as low as one camera. And so this is Yuan's project. And this is kind of the first image we've shown of our new system that we're calling OpenMonkey Wild. And it's actually very powerful. It started to actually have some real ability to estimate the position of various key points on monkeys and other primates from single views, even from single shots. And just to illustrate its robustness, here's some data that we collected from some other non-monkey primates. And I'll just add in that we're looking to build a very large database test set for this, and we're looking for about 50 to 100,000 photographs of monkeys and other primates. So if you have several thousand of those sitting around, give me an email. You can find my email on the NeuroMap website. Finally, we'll just point out that we can, one of the really impressive things about this is because we have this large database of 2D, 3D correspondences, we can start doing what's called lifting, which I think one of the next talks in the session is going to be about. Lifting is basically when you start to infer 3 dimensions, you start to do the Z dimension or depth from just a 2-dimensional image. You can start to guess where things are, and that winds up being very, very valuable for a lot of purposes in neuroscience and in terms of image processing as well. So Yuan's system can do these right now. And I'll just say this is very preliminary, but we're starting to take raw video and started trying to infer skeleton key points from these videos. This is definitely a very preliminary version, but this is the direction we're going. We're trying to do real life in the wild, ideally as fast as possible, tracking. All right, I just saw a 0, so I will say thank you to the entire team at OpenMonkey Studio and to everybody else in my lab who's been extremely supportive of this sometimes very crazy, very ambitious project that we've been undertaking for the last several years.
Thank you very much, Benjamin. Very exciting talk. So we have one question from Lawrence Hunt. The question is, I think you answered a bit of it, but it gives you some time to talk more about it. So Lawrence says, extremely cool. And does the challenge of generalization mean that it will only work in your room in Minnesota, or would you be able to generalize to larger, smaller rooms and other environments? And also, do you have a sense of whether the green background is essential, or whether it might work in home cages, etc.?
That's a great question. I would say that the green background has been absolutely essential for us to build our model and to get things working. Now that it's up and working for us, I think we can start to relax a lot of those constraints. So in some ways, this is like a physics experiment where it takes you 2 years to build your thing, and then you get your measurement. And then once you have your measurement, you don't need that thing you built anymore. So we're still using that in our room, and it's going to give you higher quality data, but I think we're going to be able to start helping other people. And we've actually got several requests from other people that we're working with to start building other systems at their places. And I don't think, for example, the green walls are essential. I don't think the particular dimensions of the room matter that much at all. You're going to need a certain amount of coverage with your cameras or your one camera, and that's going to be the most important thing.
Okay, thank you, Benjamin. So we'll switch over to our second talk, and that's Adam Gostolai. He's a postdoc in Pavan Ranvier's lab at the Brain Mind Institute at EPFL in Lausanne. And he will talk about Lift Pose 3D, a deep learning-based approach for transforming 2D to 3D pose in laboratory animals. Ben, could you stop sharing your screen, please? Thank you.
Sorry, just it's on the first slide.
Adam, we can still see both, like current
Okay.
and next slide.
Oh, sorry, 1 second. Okay. So good evening, everyone, or good morning if you are somewhere else in the world. And thank you, Benjamin, for leading so nicely to my talk. So whereas Benjamin talked about how to make really accurate 3D pose estimation using many cameras, he briefly also hinted on that there is an easier way using a novel method called lifting, and this will be the topic of my talk. But before, I'd like to thank a very interdisciplinary group of the Ranvier Lab and my funders for support. So very briefly, in the Ranvier Lab, we are interested in studying behavior from neuromechanical principles. So our favorite experiment is to pick a command neuron, which is a neuron that creates behaviors, then study it, the behavior generated by making that command neuron generate the behavior using statistical methods. And then if we are really interested, we can look at the neuromechanical principles using calcium imaging. One thing that I hopefully don't need too much convincing of is that pose estimation is really key to understanding the neural control principles. So one key aspect to it, though, is that there is a trade-off. So although pose estimation has gone a long way from the beginning, so it probably started by Bernstein, who essentially took intermittent photographs to study essentially the atomic composition of behavior. We've gone a long way from that. So novel methods using 2D markerless pose estimation methods can have a really give a really accurate quantification of behavior, and is really widely used. However, 2D poses suffer inherently from a loss of information. So you can have multiple pose configurations in 3D that will still lead to the same 2D projection. And therefore, a second big leap would be going to 3D. And there are many different methods. One of them, of course, is what has just been presented. However, there is a challenge here, which is that many of the experimental systems look like that. There are heavy space constraints, and this is something that we face in the Ranvier Lab too. So one of my challenges was in my larger scale project to solve this. And essentially, I like to give you our contribution already. What we do is essentially take single camera images, and we try to generate 3D positions. So on the top video, what we can do is to essentially consider a situation when some poses will be occluded by, for example, the animal turning and not being visible in the camera. Or perhaps you only have one view at all. And in that case, you still want 3D information to generate, for example, joint forces or for more advanced kinematic analysis. Let me give you a brief background, is essentially what the challenge is. So traditional methods rely on typically something called triangulation. In triangulation, you essentially take a point in 3D space, and those points through the focal points of the camera will create projections on your two cameras here too, or you can have more cameras as well to increase accuracy. And what you do during triangulation is to imagine that there is now a point estimate which will project onto your camera frames, and then you optimize for these projections so that they're as close to your observed points as possible. This is right now the state of the art in obtaining 3D. However, it's quite complicated because it needs multiple cameras. An alternative, though, is through lifting, or if you think of it as a glorified regression. Essentially, what you do now is to, rather than individually think of the points, you think of a collection of points. So lifting doesn't make sense for only one point. You need a point arrangement or a pose, as we say. In that case, you take all of the points in a two-dimensional camera projection and then try to infer the three-dimensional pose using this function f. This function f is typically approximated by a deep neural net with certain parameters to minimize, again, this projection error. This is by, well, I have to say that you need one camera, of course, but also it's an ill-posed problem, simply because, again, what I've said, there can be many different three-dimensional poses that lead the same projection. So this challenge has nevertheless been solved. So for human poses, at least, or at least to a large extent. In human poses, they have a large number of poses. So there's a huge data set that contains in the order of millions of poses of people doing, dancing, yoga, or whatever you like. However, for animal pose estimation, there are particular challenges. So for example, we can have occluded key points, possibly no ground truth at all, and much less data. So these are the challenges that we like to address by taking a well-known network that is quite simple. It doesn't require us to use temporal information, and we can modify its training and data augmentation methods to actually cope with these challenges. So what we do is we take our simple setup, we take 6 cameras, we record our fly because we do flies in our lab. And then suppose there's a case when we only have 2 cameras. So each camera is recording 3 limbs on one side of the fly. One thing what we can do then is to feed in both of these images on either side into our network. The network will have no idea which side the image is coming from, so it will learn both pose configurations against 3-dimensional ground truth data. We typically rotate this so that, as Benjamin said, it is advantageous so that if the network only learns the depth of the image as opposed to trying to predict all 3 coordinates. And what we notice is that by choosing the behavior sufficiently diversely, we can get really remarkable accuracy. So if we trained on different behaviors, we notice that, so the little golden dot shows you what behavior has been trained on. So MD and ADN are 2 optogenetically driven behaviors through command neurons. And the control behavior is just a spontaneous behavior when the fly can do whatever she wants. So basically what we notice is that if we train only on optogenetically driven behaviors, so the first 2 columns, then other behaviors are typically quite badly predicted. But if we train on spontaneous behaviors, which supposedly explore the kinematic space quite well, we actually get quite a good prediction for even optogenetically driven, highly stereotypical behaviors too. We also compared it to triangulation, and we noticed is that if we have 4 cameras, we basically get the same accuracy by using only 2 via lifting. And at least for the camera angles we tested, we get actually quite a good accuracy from each angle. However, there are much more complex cases that one can consider in experiments. One is the one that showed where I actually have occluded key points, especially now that the animal is turning. In this case, from the side view that gives you the triangulated ground truth, only 1/2 of the points are visible at any given time. So if you want to now train such a lifting network, you actually face a disaster simply because the other 1/2 of the points that are not shown in red, the network is free to predict since you don't have ground truth for the unseen points. So what you observe when you're trying to train the network is that the network has very completely chaotic predictions. So what do we do to fix that? We noticed that one thing that we can do is to exploit the symmetry of animal movements, so the left flight or bilateral symmetry. And this applies for both of these cases that you can see now. So we take, again, 2 viewing angles. So we like this prism system, which is very simple. Using only 1 camera, you get 2 angles. And we try to align the images so that the fly is aligned with respect to the same reference frame. In this case, through training, we feed the network with either the left or the right-hand side. However, all of these have been aligned against the same reference frame. So this way, the network is quite remarkably able to essentially delay triangulation towards a training stage, effectively filling in information that you were not given it. So you get a 3D information. So when you're actually trying to predict, you see that it can give a very good accuracy for unseen points. And moreover, for experiments that have occasionally have corrupted points, our network can actually, from the trends of the data, predict those points correctly that have been mislabeled, essentially. So these are shown by these little white triangles. Those are points for which the input was good. So the ventral view, which we are lifting, is good, but the side view has been incorrect, and the network corrects it. And this is the result. Essentially, triangulation and lifting gives you similar results. However, what happens when you have simply no 3D information whatsoever? This is quite frequently encountered, especially in simple systems where you require high throughput. And this is very commonly employed in Drosophila, but other systems as well. Here is one example where you essentially study social behavior of flies with one camera from the bottom. Crucially, you cannot study physical degrees of freedom here. So you have a projection and you're observing changes in length from this projected view. So what you would like is to recover joint angles, for example. One thing that we notice is that we can transfer the network that we had previously obtained from a same angle and study this problem, except that there is a domain adaptation step that's needed. So if you have different resolutions, you can imagine that if you have now a lower resolution system where you have multiple flies, hence you share resolution, in that case, the input can, the high resolution input can essentially be varied to many different pixels and still be indistinguishable from the point of view of your test data set. So therefore, in training, one has to account for that by essentially adding a noise in proportion to the resolution difference of data sets. And therefore, you can generate now data that has been previously unseen. And this is what I show you here. These are two data sets that one was taken from a previous study, and the other one is from what we collected. And the essentially the red dots show, the red boxes show information that has previously not been seen at all in these experiments. And what you also see is that in the projected view, so if you look at the ZX projection, those are actually progressively smaller circles that also people see in experiments in terms of lag amplitude motion. So to sum up, we have created a framework that can really make 3D pose estimation simple from one camera. It can use corrupted data and also occluded points, and is able to essentially perform domain transfer where you get 3D information for free using one of our trained networks. So I invite you to look at our GitHub page for the code, because it's already been on bioRxiv for a few days now. And welcome your questions now.
Thank you, Adam. So unfortunately, due to these technical issues we had, we're a little bit behind on time. So we'll just move on to our next speaker. And our next speaker is David Mason. He's a PhD student in, well, Ben Hayden's lab, who gave the first talk in the session, and also with Jan Zimmerman at the University of Minnesota. And David will talk about a functional hierarchy for choice in medial prefrontal cortex. So if you try to share your screen, please, David.
Okay.
Okay, great. Go ahead.
There we go. Okay. Hello, everybody. I'm going to hopefully do this fairly quickly because this paper actually is on bioRxiv. So if there's questions, hopefully we can either get to them at the end or get them resolved by checking that paper out. So yeah, I'm going to talk about a functional hierarchy for choice in the medial prefrontal cortex. And hopefully, so this is a little bit of a change of gear due to a schedule change from the previous talks in terms of topic, but hopefully it's still interesting for everyone who's here. So hopefully if I've done my job correctly, then by the end of this, I will have at least started to convince you that value-based representations or value encoding within the medial wall of the prefrontal cortex is distributed functionally rather than modular and is organized hierarchically. So first things first, just to thank all the members past and present of the lab, our funding, and of course our wonderful participants, our subjects, who are fantastic. Okay, so I think the first order of business is to kind of establish some key terms that are necessary for understanding what we're trying to get across here. And the first is to say that what we're going to be dealing with is the sort of functional computations that are involved in value-based decision-making. One of them being to encode magnitude. So here we're kind of showing, you know, if you want to, which one do you want more, assuming that the neighborhoods are identical and the commuting time is identical and you like bigger houses. I mean, maybe there's a subjective thing here, but, you know, objectively we'll say the magnitude of value of the house on the left is greater than the magnitude of the value of the house on the right. And so that kind of suggests that you're able to compute a sort of representation. You're able to encode some sort of value magnitude. But if we were to then say, okay, which one do you want more, given the chance that you only have a 1% chance of actually getting the thing on the left versus getting the thing on the right as a 90% chance, then now you're starting to integrate the information of probability of success as well. And so these are the types of computations that we're going to be talking about. And sort of these economic choice computations, what I'm hoping to show is that in the medial prefrontal cortex, they're distributed across a number of areas, and that the difference between those areas is quantitative rather than qualitative. But they're still organized, and this is kind of a representation of what I'm talking about, this modular organization of function versus distributed. But they're still kind of organized in this functional hierarchy. Now, this paper doesn't exactly talk about functional hierarchy, but these are the areas we're kind of referring to here, namely the prefrontal ventral medial prefrontal cortex, excuse me, the SG-ACC subgenual anterior cingulate, the pregenual anterior cingulate, and the dorsal anterior cingulate. So next, there we go here. Oh, there we go. So this is the task that we used. It goes back a number of years. So this is our standard risk-based gambling task. The order of operations here from left to right is that the monkeys are presented with some combination of probability and magnitude represented by the size and the color of the bar. Then there's a delay period after which they're presented with another offer. Okay, so this allows us to kind of segment the trial into epochs. After another delay period, a fixation is reacquired. Both of the same offers are then presented simultaneously, and the monkey saccades to a choice that he prefers over the other. And so the question is, what's going on in each of these four different areas that I'm going to talk about in terms of representing the computations involved in kind of representing these offers and coding the value of these offers and eventually making a choice. So these are the areas that we recorded from: the ventral medial prefrontal cortex, the subgenual anterior cingulate, pregenual anterior cingulate, and the dorsal anterior cingulate. And here we have the behavior across all the monkeys across all the sessions, just kind of suggesting that the probability of choosing the first offer is very low when the value of the second offer is dramatically larger than the first. Whereas over here, when the value of the first offer is significantly larger, they're obviously more likely to choose that offer. So it's kind of showing us that their behavior is pretty normal, pretty expected, and sane. So in terms of defining these computations that are involved in representing and encoding value, what I'm going to try to show you is that there's an expected shape to the way that they're encoding. So if we imagine that what we did is we regressed the mean firing rate on the expected value of offer 1 during epoch 1, and we did the same thing for EV2 during 2, then what you can expect is that assuming that one is encoded in the same format as the other, you would expect that there's a sort of positive correlation between the two. But if they're encoded in opposite formats, then as the encoding magnitude of expected value 1 increases, then you would expect the encoding of expected value 2 to decrease, and we call that mutual inhibition. And so that's kind of what I'm going to show in these next graphs here. So hopefully that kind of becomes clear from what I'm talking about. So in this first row, there's all the different areas, okay? And we have the encoding of, here we're talking about integration. So we have the firing rate regressed against the probability of offer 1 and the magnitude of offer 1, and they're positively correlated, suggesting that the format in which each of them is encoded is integrated into a single expected value code. Now, this is our first indication that these functions are distributed rather than modular, because you can see that same positive correlation across all 4 areas. Whereas if it were modular, perhaps you would expect it to only be here and not anywhere else, where that doesn't appear to be what's happening. In the next row, we have a similar sort of representation, but here instead we're calling this attentional alignment, okay? Because in this case, we're looking at the expected value of offer 2 during epoch 2, during its own epoch, and the expected value of offer 1 during its own epoch. And again, we see that the computation is the same across all structures. And I kind of alluded to the mutual inhibition of 2 offers. And here again, you find that there's a negative correlation in all 4 structures. So the fact that it's the same format for encoding for all 4 different structures begins to tell us that this function is not modular, but in fact distributed across this network. And so the next question is there a functional hierarchy? I won't spend too much time on this, in fact, because I'm running out of time. I'll just skip over it. We can come back to it if there are questions. But in order to get at functional hierarchy, what we decided to do was run a linear SVM to basically decode different types of information depending on the time period that we're collecting the mean firing rates from. And so the moral of the story here again is to show us that function is distributed because in all cases we're above the shuffle value across all 4 structures. The other really important thing is that with few exceptions here, for example, and here, by and large, the extent to which information can be decoded from the firing rates during a given epoch, information about that type of value tends to increase as you go down, as you go up this functional hierarchy. And that seems to be the case across the board. And so I guess our sort of takeaway message from all of this is again to say that these sorts of economic choice computations are distributed across the network in a way that the distinctions between the functional contributions are quantitative rather than qualitative, where there are sort of meaningful qualitative distinctions between strict anatomical boundaries, and that this sort of functional organization appears to happen hierarchically, moving from the VMPFC through the SGACC to ultimately to the DACC, at least for this medial prefrontal network. And so I did that kind of quickly, but I'm hoping that that was a good thing and that this leaves a lot of time. I know we were kind of back against the wall here. So with that, I'll take any questions. And if there was anything that was super unclear that I can't answer, then hopefully you can reference the bioRxiv paper.
Okay, thank you, David. So if anyone wants to pose a question, just pose it in the Q&A window. Maybe I'll start with one. So you showed this hierarchy from ventromedial prefrontal cortex up to DACC, where you basically accumulate more information. And does this information, where does it come from? So are there other inputs to this network where you can obtain the information somehow?
Yeah, I think that's going to be a really important follow-up question. We don't have the neural data to show that at the moment, but I would imagine that there's kind of a long chain leading from sensory input through all the different sensory and association processing areas until it eventually gets here. But we kind of approach this question of, okay, so once all of that sensory information has been integrated and is kind of formulated into something that might represent the value of an offer, once it gets to this medial prefrontal network, what then does the network do with it? But I suspect you're probably right. That information must be coming from somewhere. And I imagine that, again, in those cases, you find a sort of gradual transformation of information rather than stark, clear modular boundaries between anatomical areas.
And we have another question from the audience. So again, Lawrence Hunt asks, with the decoder, are you decoding left versus right action choice, or choose option 1 versus choose option 2? And if the former, might you have a hierarchy that increases towards action in the MPFC to the ACC, but it also moves away from 1 versus 2 comparison?
Yeah, so actually we have both pieces of information here. So we have the offer side during epoch 1, that's left versus right. Here we have choice, as in choice 1, option 1 or option 2, but that's during the choice epoch. And again, we have choice side during the choice epoch. So we have in all, we have three cases in which that trend kind of stays the same regardless of whether it's position or offer number. I hope that answered the question.
Okay. Yeah, thank you. So, yeah, so we don't have any further questions, and we're already a bit over time. So thanks again to all three speakers, Ben, David, Adam. Thanks for the questions from the audience, and enjoy the rest of the conference.
Our results on 4 very different data sets to give you a sense of the capabilities of the framework. So in recent years, many new technologies have emerged that are giving us new ways to study the brain. Technologies like neuropixels allow monitoring of hundreds to thousands of neurons in a wide variety of brain areas. Compact and robust recording technologies are also allowing us to capture data during increasingly complex, even natural behaviors. On the modeling side, we have new ways of uncovering single trial neural activity and understanding population level dynamics. However, the sheer volume and dynamical complexity of this data is a critical barrier to uncovering and interpreting these dynamics. Thus, we need a more general, flexible, and efficient modeling framework to complement these technologies. So latent factors analysis via dynamical systems, or LFADS for short, is a very effective deep learning model that represents neural activity as the output of a latent dynamical system, which we call the generator. According to the dynamical systems framework, the generator's state is determined by its previous state and any time varying inputs. LFADS takes segments of spiking data as inputs and produces estimates of the Poisson rates underlying the observed spikes. It infers latent population dynamics, estimates single trial firing rates, and models large numbers of neurons, and requires no behavioral data. As you might have noticed, LFADS already has many characteristics of a generalized neural dynamics model. So what's missing? The problem is that it's in its original form, LFADS is not easily generalized across data sets. Like most deep learning models, LFADS has many regularization hyperparameters whose optimal values are impossible to know a priori and may be drastically different for different data sets. For most other models, this would be addressed by training on the order of 100 models with random hyperparameter values and then choosing the one that achieved the lowest validation loss. However, using synthetic data with known firing rates, we've shown that LFADS models with the best validation loss are actually overfit to spikes, resulting in noisy representations that don't reflect the underlying firing rates. So in practice, we don't have true firing rates. So identifying high-performing models on real data requires following the random search with supervised evaluation using indirect measures of model quality. For example, PSTH reconstruction or behavioral decoding. This not only requires additional behavioral data, but also biases model selection towards the metric rather than the true objective of modeling neural activity. Thus, the key problems with applying the original version of LFADS to a variety of datasets are the computational expense of the random search and the unreliability of the negative log likelihood as a measure of model quality. So AutoLFADS solves these key problems with two complementary innovations. The first is a novel regularization technique called coordinated dropout, which prevents overfitting to spikes, allowing us to simply select high-performing models by negative log likelihood rather than behaviorally biased supervision metrics. The second is our use of an efficient hyperparameter search algorithm called population-based training to discover optimal hyperparameter schedules. So in the rest of this talk, I'll illustrate how AutoLFADS surpasses LFADS in 4 key ways. First, it infers higher quality representations of neural activity. Second, it finds high-performing models much more efficiently. Third, it is unsupervised with respect to behavioral data and brain area. And finally, it produces generalizable dynamics models on condition subsets and small data sets. In the subsequent slides, I'll indicate which of these categories each experiment falls under in the bottom left corner. So in our analyses, we applied AutoLFADS out of the box to 4 non-human primate data sets, which I'll describe briefly here. The first data set was the maze data set used in the original LFADS paper, which included intracortical recordings from M1 and hand kinematics during reaches through 108 different maze configurations. The second data set, the random target task, had similar recordings during a self-paced sequential reaching task between random elements in a grid. The third data set included recordings from somatosensory area 2 and hand kinematics during either mechanical perturbations or voluntary movements of the hand. The final data set was recorded from the dorsomedial frontal cortex, a cognitive area, as the monkey was presented with two stimuli separated by a time interval, and then attempted to reproduce that interval by waiting before initiating a movement. On subsequent slides, I'll use the task names and icons to indicate the data sets corresponding to each result. So we trained AutoLFADS out of the box along with about 100 LFADS models with random hyperparameters across a reasonable range for each data set. We then calculated PSTHs for smooth spikes, AutoLFADS rates, and LFADS rates by averaging across trials, and we calculated the coefficient of determination between empirical and modeled PSTHs. These plots show that AutoLFADS retains features of empirical PSTHs better than any random search model for all stereotype tasks in these 3 very different brain areas, from a motor area on the left to a sensory area in the middle and a cognitive area on the right. So in addition to the performance gain, it's important to note that these random searches used 3 times the computational resources for training and required supervised evaluation in order to select the best model. So these results demonstrate the accuracy, efficiency, and unsupervised nature of the AutoLFADS framework. Similarly, when we evaluated models based on linear decoding of hand velocity for these 3 hand velocity tasks, we found that AutoLFADS consistently outperformed the best random search model for all 3 kinematic tasks in motor and sensory areas, without manual adjustment for individual tasks. Again, note that AutoLFADS used 3 times less computation for training and found a high-performing model without a supervision signal. One challenge in working with neuroscientific data is that large data sets are hard to come by. Most data sets are fairly small by machine learning standards. To evaluate the performance of AutoLFADS on small data sets, we divided the maze data sets, almost 2,300 trials, into smaller subsets with 20, 10, and 5% of the original data. We trained AutoLFADS models along with manually tuned LFADS models on these subsets and evaluated linear cursor velocity decoding performance from their resulting representations. We found that even with only 10% of the data, AutoLFADS models were able to maintain high decoding performance, while performance dropped significantly for manually tuned models. Given that LFADS models population dynamics, another reasonable question to ask is how well the inferred dynamics can explain conditions that aren't found in a given data set. To test this, we held out condition subsets in the maze task while controlling for data set size. We trained AutoLFADS and manually tuned models on the subset. then froze the generators and trained only the encoding models for the full dataset. Linear cursor velocity decoding from the resulting inferred rates showed that AutoLFADS infers generalizable dynamics even when trained on only 1 or 2 conditions, while performance of manually tuned models tends to decline more quickly. Normally, the input samples for LFADS are entire trials, but they don't necessarily need to be. Because the random target task was self-paced, the trials for this task had various lengths ranging from 1 second to almost 4 seconds. Rather than using whole trials as input samples for AutoLFADS, we generated samples by chopping the continuous data into fixed length overlapping segments, without regard for trial boundaries. After training, we reassembled the rates and broke them back up into trials, and we reduced dimensionality to the first 3 principal components. Here, we color the neural trajectories by reach angle to show that AutoLFADS discovered trajectories that contained information about reach direction, despite having no information about the notion of a trial during training. This shows that AutoLFADS requires virtually no knowledge outside of neural data to infer meaningful representations. So in sensory areas like area 2, it's common to fit behavior to neural activity using a behavioral encoding model. Here we show that the rates inferred by AutoLFADS explain the observed spikes much better than a GLM for all neurons across 2 recording sessions. This shows us that AutoLFADS is capturing a lot of information that isn't contained in the behavioral data. Again, I want to emphasize that AutoLFADS out of the box was highly successful in modeling a highly input-driven brain area with very different statistical and dynamical properties from the motor cortex. In the cognitive timing task, it's been shown that the neural speed in DMFC during the produced interval is negatively correlated with the length of the produced interval. Neural speed, analogous to kinematic speed, is defined as the norm of the change in firing rates over time. We computed neural speed for neural representations obtained by smoothing, GPFA, random search, and AutoLFADS. AutoLFADS reveals this negative correlation better than nearly all other methods, but is comparable with the best LFADS model from the random search. However, recall that finding the best LFADS model requires extra computation and supervision data. To drive this point home again, I want to emphasize that we applied AutoLFADS out of the box to a new brain area whose activity depends on the confluence of both inputs and internal dynamics, and we're able to achieve better results from the PSTH reconstruction, decoding, and decoding than any other method.
All right, 1 more minute.
Thanks. So to conclude, we've shown that AutoLFADS greatly enhances the accuracy, efficiency, and generalizability of LFADS, while also reducing the reliance on supervision signals and manual input. We were able to find the best performing models with high computational efficiency, perform model selection using only neural data, apply the model out of the box on motor, sensory, and cognitive areas, generalize well to unseen conditions, perform well on small data sets, and model data without regard to trial boundaries. So we believe that by freeing researchers from the often tedious problem of hyperparameter optimization, this technique will enable the study of dynamics of neural populations in a wider variety of brain areas and for a wider variety of behaviors. So I'd like to thank all my collaborators for all their hard work on this project, especially Reza Keshkaran, my co-first author, and Chetan Pendarnath, my advisor. Also, really huge thanks to the collaborators who shared their data as well as our other funding sources. Thanks.
All right, thank you, Andrew, for that excellent talk. We already have a question for you from Adam Willits. So they're wondering how specialized the innovations of AutoLFADS are specific to this technique. Do you think other models would benefit from regularization hyperparameter search, specifically the specific forms that you presented here?
Yeah, so it's an interesting question. So one of the problem with AutoLFADS, so AutoLFADS is a sequential variational autoencoder. So when the model has enough capacity, it will take these spikes that we're feeding as input and basically put those or output those same spikes or rates that look very similar to those spikes because they'll have high likelihood for the observed data. So coordinated dropout is essentially preventing spikes that were passed to the model at the input from also being back propagated, or from the loss being back propagated through those samples at the output. So I think a model that uses negative log likelihood and is based on spikes could very well benefit from coordinated dropout, especially if you're seeing this sort of overfitting that we were seeing, where the model isn't inferring rates based on this dynamical systems constraint, but it's instead just passing rates from input to output in sort of an uninformative way.
It's a great response. And we have one more question from Cole Hurwitz. Could the overfitting of LFATs be regularized by increasing the KL divergence in the elbow?
I think so. I think you're talking about the KL penalty. So, for autoencoders, variational autoencoders, there's a KL penalty, which is sort of a regularization. And yeah, that's what the hyperparameters are actually doing. So when we're adjusting these hyperparameters during PBT, it's actually adjusting those penalties in sort of an adaptive way.
All right. Thank you very much, Andrew. That was great. We are going to move on to Omid.
Cool.
Oh, okay. So there's one more question down there. Andrew, you can feel free to type your response to that answer if you'd like. But next up is Omid Sani from USC. He'll be talking about preferential subspace identification for modeling behaviorally relevant neural dynamics. Go for it.
Thank you. Hello, everyone. So thank you for the introduction. I'm Omid Sani, a postdoc at the Shinichi Lab at USC. I'm going to talk about this method, PSID, for modeling behavior relevant neural dynamics. So to give some background, a lot of studies have some recordings from the brain, and we also have some behavior that is tracked. And we're interested in studying the relation between the two. And one of the traditional approaches is based on the idea of tuning, where you have, when you observe the firing rate of some neuron goes up and down based on the behavior, like faster behavior with a higher firing rate. And this leads to a model like this, where you have neural activity as a function of behavior at each point in time. And further, you can also model the dynamics of behavior, which can help with reduction of noise, and it's largely based on physics, like the relation of velocity and position. And you can also model the residuals of these relations. And together you have, once you learn the parameters using least squares regression, for example, you have what is typically called a representational model. Now the issue is there is potentially a lot beyond the behavior going on in the brain. So we might have the same neural activity, the different neural activity for the same behavior, and the other way around. So another approach that's quite popular is to model the neural activity as a function of now a time series of latent states. Now these latent states drive the neural activity, and they have their own dynamics that you can model with a difference equation. And they also drive the behavior. This is more flexible than before because these states are latent. And once you learn their dynamics, the residual statistics and all the model parameters, this is what you could call neural dynamical modeling, or NDM. So notice that both cases you are using a linear statistics model to describe the neural activity. And the difference is really in the objective that was used to fit the model that leads to them describing potentially different parts of the neural activity. So to see that, let's consider that our brain has produced the recorded neural activity and behavior. And you can imagine the state of the brain at each point in time to be a high dimensional variable where some of its dimensions have contributed to driving the recorded neural activity, some have contributed to driving behavior, and some have contributed to both. And you might be interested in finding this shared dynamic, the behavior relevant states of the brain. But let's first see what those standard methods that we just described do. So in neural dynamical modeling, the dynamics of the model are learned purely using neural activity. So it may learn dynamics that are unrelated to the behavior of interest. And in representational modeling, the dynamics are learned purely based on behavior. So you may learn behavior dynamics that are not encoded in the recorded neural activity. So we need a new method that we've developed here, PSID. You can check out the details in the preprint. But the general formulation is a linear steady space model where you have both neural activity and behavior in terms of the latent state. And importantly, the way the model learns the parameters of the model, the way PSID learns the parameters, it dissociates the states into two parts, the behavior relevant dimensions and the other dimensions. And critically, you can only learn the behavior relevant ones, which has several benefits to touch on here. So the fitting is very efficient. It involves just a few linear algebra operations, SVD and so on. You can check out the preprint. So it's efficient in fitting. It doesn't involve like unpredictable, computationally expensive optimization, for example. It has a predictable cost of computation. So first, as general validation, when we apply this to a lot of random models with random parameters, we can show that as more training data is provided, all model parameters are learned correctly with the error converging towards 0. And we can also show with simulations that it correctly prioritizes learning of behavior relevant dynamics. How can we do that? So here we've simulated random models that have 16 latent states, but only 4 of them drive behavior. And we want to see if we identify a model with only 4 latent state dimensions, what would the method learn? Would it be the 4 behavior relevant ones or any of the other ones? So to evaluate that, we can look at the eigenvalues of the block of the A matrix that is related to behavior relevant states and see how well we can learn them. And here I'm showing the result average over the models, the random models. You can see that PSID in green as the horizontal axis is the latent state dimensions. As we increase the latent state dimension, as soon as we get to 4, which is the minimal number required to describe these 4 behavior relevant latent states, the error of describing them goes to 0. Whereas if you use neural dynamical modeling, for example, even with 16 dimensions of latent states, you still have more error than with PSID. So PSID is correctly prioritizing behavior relevant dynamics. Now, this has also implications in terms of the number of training samples that you need. So now I'm showing the same error as a function of the number of training samples that we used. And you can see that PSID, the green, is going to very small errors, while neural dynamical modeling with a much higher latency dimension of 16 requires orders of magnitude larger training samples to get to similar errors to PSID. And if you have lower dimensional states, it wouldn't get there even with more training samples. So PSID, we can confirm with simulations, correctly learn all model parameters, learns them with lower dimensional latent states and fewer training samples. Now let's see what we can see in the real data. Now here we are analyzing data collected by our collaborator, Bijan Besaran's lab, where monkeys are performing reaches to diverse 3D locations with their hand, holding the target, letting go, and returning to the resting position. And we are tracking the angle of all joints in their fingers, arms, elbow, and so on, so all degrees of freedom. And now in the real data, we don't know what the true eigenvalues are to evaluate whether we've learned behavioral dynamics correctly or not, but we can decode behavior. And that's our measure of how well we've learned behavioral dynamics. And just to give you an idea, the decoder consists of just the neural activity goes into a Kalman filter, and then that gives you the latent states that describe the neural activity, and then a linear combination of those latent states gives the behavior. So this decoder structure is the same in all methods, but they've learned different parameters for this decoder. So here I'm showing as a function of the latent state dimension what the cross-validated decoding accuracy was for across all joints. And you can see that PSID, the green, converges to its peak decoding performance with very few latent state dimensions, whereas the other methods require more latent state dimensions to reach to their peak performance, which is lower than PSID. So now to go deeper into this a little bit. So across all data sets, this is the distribution of the dimension at which each method reaches its peak performance. So for PSID4, much higher for the other methods. And at that dimension of 4, PSID is significantly more predictive of behavior than neurodynamical modeling. And even with a much higher dimension, the other methods still are significantly less predictive than the low dimensional model that PSID finds. So PSID is finding a low dimensional representation of behavioral element dynamics in neural activity, this real data. And it may help characterize or quantify the dimensionality of behavioral element dynamics here. So next, we repeated this for every recording channel and every joint separately. Similar results for almost every recording channel and joint. PSID learns significantly more accurate models of behavioral element dynamics. And next, we asked whether this low dimension that PSID finds is low because behavior or neural activity are as low dimensional. But we found that when we modeled neural activity on its own as a linear status model with different latency dimensions, or behavior on its own as similar linear status model with different latency dimensions, the dimensions that you need to reach the peak prediction of each signal using its own path is significantly higher than the dimension that PSID reveals for the joint dynamics. So there are predictable dynamics in both signals, much higher dimensional, but the shared dynamics is low dimensional, and PSID allows us to uncover that. And then another interesting thing in this data that we found is now that we have a tool to do dimension reduction while preserving behavioral element dynamics, we wanted to visualize what's happening in the population activity during the task. So we fitted models with two-dimensional latent states, both with NDM and PSID, and then plotted these two dimensions during reach and return. So we found that, interestingly, there is rotational dynamics both during reach and return in our task, which is kind of a complex task. It's interesting that here in such a complex task, we still find rotational dynamics. But then more interesting than that, when we apply PSID, we find rotational dynamics that reverse direction between reach and return periods. So note that only neural activity has gone into the models that extract these latent states. So it doesn't know that the arm movement is roughly reversing direction during reach and return, but it happens that the latent states that PSID extract are intuitively more congruent with the reversal of the hand direction in the task. But quantitatively, they also lead to significantly more accurate prediction. So this shows that both kind of dynamics existed in the neural activity, but PSID allowed us to extract the ones that are more behavior relevant that might otherwise have been missed. And with that, I would thank the lab and all the funding sources. And here is the link to our preprint. And the Python and MATLAB code for PSID will be online soon. So this is a general method. You can apply it to any two signals, like two different neural activity from two different regions of the brain to find the shared dynamics, or neural activity, or even two different signals that are not neural activity. Basically, the math, the simulations don't care about the signals. So I'm excited to see what people might be able to find using the method. With that, I'm happy to take any questions.
Excellent. Thank you for that great talk, Omid. We do have a couple questions already. So Cole Hurwitz wonders, have you tried using PSID on one behavior and then seeing how well the learned dynamics explain another behavior? He followed up with some held out behavior you would think shares the same neural dynamics as the other first behavior.
That's a very good question. We've actually done this. Hopefully in the paper that comes out, you would see. We tried leaving out different dimensions of behavior from our training data, like train a random subset of joints and then try to predict the other one. And indeed, even in that case, when PSID hasn't seen the behavior, because all those joints are involved in the same task, there are correlations between them. The model that PSID learns was more predictive of unseen joints than the other methods.
It's a very illuminating answer. And we have a second question from Matthias Hennig, wondering whether you checked the variance associated with the relevant dimensions and compared them with others.
Variance associated. So, okay. You mean the variance in neural activity that was behavior relevant versus the rest. So we have also done that. We've swept the latent state dimensions and then looked at how much variance in neural activity they predict versus how much variance in the behavior they predict. And with PSID, you can explain the behavior variance very quickly. And there is a lot of neural variance that is not still required to explain the behavior dynamics. And PSID has this optional second stage that continues to also learn those neural dynamics. And you can see, I wish I had the plot here. You can see that, yeah, there is a lot of behavior irrelevant variance in neural activity. The question is which ones you learn first, and PSID tends to learn the behavior relevant variance in neural activity first.
I think you did a great job illustrating it with your hands.
Oh, you could see it?
Yes. All right, and there's a follow-up comment, and then I think we'll have to switch over. Matthias says, at least in the mouse M1, the neural variance associated with the task could be very small.
Oh,
I think that's just that's a follow-up comment in the chat.
Did you check the variance associated with the relevant dimensions?
Oh, I think Matthias was just making that comment that in mouse M1, it might be expected that the variance associated with task could be very small.
Yeah, and that's one of the motivations of PSID, because if you blindly try to model all of neural activity, you're likely to focus on just the most variant parts of it that are likely not related to behavior. So the motivation is to try to not miss those potentially small variances in neural activity that are behavior relevant.
Great points. All right, thank you, Omid. I think we're going to have to switch gears to Joel. All right, so next up is Joel Yee from Georgia Institute of Technology talking about representation learning for neural population activity with neural data transformers. So go for it, Joel.
Thank you. Yeah, great. Okay, so I'll be talking about my work with neural data transformers. I apologize if some of the terms are overloaded from the previous presentations. They may have slightly different meanings here. So I'll start by talking about how recurrent neural networks, like the LFADS model you heard about earlier, can model neural population states sequentially, and a little more on what that means. And then I'll discuss how sequential modeling can be a problem and how this transformer architecture we're introducing enables parallel modeling. And finally, we'll show that this neural data transformer NDT architecture we're introducing matches recurrent neural networks in modeling autonomous systems and offline analyses. Okay, great. So we've seen that neural activity predicts behavior, which is great, but unfortunately, it's noisy. So if we're trying to train behavioral decoders on top of these, they won't perform very well. Fortunately for us, neural activity reflects an underlying state, which is more stable. We hope that we can train better behavioral decoders on this state, and we can approximately represent this state by referring to the firing rates of the individual neurons instead of the spiking activity itself. This state is consistent across trials of the same behavior, which will be important for if we want to decode behavior. And now we ask, how can we model this state? So we consider a deep learning approach to this problem, where we're feeding in the raw neural activity and we're asking the deep neural network to predict firing rates. Specifically, our input spiking activity is first binned off into T time steps, and these time steps correspond to the initial population representations that can be fed into our deep neural network. The deep neural network processes these features and finally maps them into firing rates. And again, these rates are a view of our neural population state, and they can be useful for predicting behavior. So the state of the art for this problem in offline analysis is LFADS, as introduced earlier. And LFADS trains a deep neural network to reconstruct neural activity, as like discussed. But the problem is that LFADS can be slow, and that can be bad for real-time applications. And to understand why it can be slow, we'll need to break it open a little bit. So LFADS is a sequential encoder-decoder architecture. So inside our deep neural network black box, we have an encoder, which is responsible for summarizing the input spiking activity into a single code. And then a decoder is responsible for extracting each time step's rate predictions from that code. And more specifically, this decoder is a recurrent neural network. And recurrent neural networks operate by sequentially updating an internal state. So the decoder takes what the encoder has outputted, and it will use that to initialize its state. And then using learned rules, it will update its internals t times, and then it will output t firing rate predictions. And this is well motivated by the dynamical systems view of neural populations, which posits that neural population states themselves evolve as a state that evolves over time according to a fixed set of rules. So in online decoding, our models will receive windows of recent activity. So for example, let's say we receive 30 milliseconds, 3 bins of 10 milliseconds each. And to minimize latency, we'll be asking the model to predict firing rates on overlapping windows. So in the next 10 milliseconds, we'll feed in another 30 millisecond snippet, but our decoder will need to iterate through all 3 of those bins, even though it's only interested in the last bin's new firing rate predictions. And so this is our slow bottleneck. And RNNs could theoretically be trained to work around this issue. We asked instead, what if we could find representations without any sequential processing? What if we could transform our representations to find our firing rate predictions in parallel? And this is exactly what the neural data transformer is doing. The neural data transformer essentially has these transformer blocks stacked on top of each other for L layers. And inside each of these transformer blocks, our fed-in features are extracting the information it needs from every other feature. And it learns how to do this through something called a self-attention mechanism, but the details of which I won't go into here. So in contrast with a recurrent neural network, which is learning how to update a system that corresponds to our neural population activity, then transformer is an entirely statistical approach to this instead, which just learns the different input dependencies between different firing rate, spiking activity inputs. And because we have parallel processing now, this neural data transformer can predict rates in 4 milliseconds. And this is independent of our input bin length. This could enable a much smoother real-time application because on a typical sequence length that we would feed in during real-time application, like 50 bins, LFADS takes upwards of 20 milliseconds. And we care about transformers for more than just this speed up, because transformers have outpaced recurrent neural networks in modeling representations in both natural language processing and vision domains. So on the right-hand side, you can see that a transformer can split up different patches of an image, feed them into the same type of architecture we just discussed, and train the output representations to do object detection or classification. And similarly, in natural language processing, we can learn representations of words that can be fed into a spam classifier. But we face 2 potential challenges when transferring or translating to the neural domain. Specifically, dynamical systems, the theory that we mentioned, matches recurrent neural networks much better, right? And secondly, neural data sets are much smaller. We're looking at megabytes of data instead of gigabytes. So we verified this first problem directly by assessing whether the neural data transformer can model autonomous dynamical systems. We do this by making synthetic data sets, the Lorenz system and the chaotic RNN system discussed in the LFADS paper. We can see how the Lorenz system is built out in this series of figures. We have 3 dimensions of variation forming the butterfly figure in the Lorenz equations. And these 3 variables can be projected upwards from figure B to figure C into a firing rate matrix of neurons against time. And then this firing rate matrix can simply be sampled according to, for example, a Poisson distribution to make our actual synthetic spiking activity. Now that we have these ground truth rates, we can compare our actual model's predictions against this ground truth. And this is something we can only do in synthetic systems. But we see that in this case, our neural data transformer does make predictions that are very closely aligned with the ground truth firing rates. And this is quantified by the R² between the ground truth and the neural data transformer, which we average across channels and times of neurons. We see that the neural data transformer recovers rates that are essentially as good as the LFADS model in both these data sets. Similarly, we can model neural activity recorded from the macaque M1. And this data was recorded during reaching task, where the monkey reached one of 108 different kinematic trajectories. And the plots on the right are color-coded by the different reaching trajectories. So we see on different trials of the same reaching condition, we are recovering qualitatively similar rates for a given neuron's activity. And again, as mentioned previously, when we don't have ground truth rates, we can use behavior to evaluate our model quality. So we do least squares regression to predict the monkey's arms kinematics that were simultaneously recorded. And we see that when we do this, we can report the R² again between the predicted and ground truth velocities for this data set. And here, the neural data transformer and the LFADS do, again, roughly on par and do very well. Whereas the smoothing baseline that we present which would be analogous to trying to build a decoder on top of the raw neural activity, only gets 0.6 R². And we verified the second challenge, that the neural data transformer can scale to smaller data sets, which may be of more interest in other neural domains. Our data set has 2,300 trials, which is fairly large. So we restrict to smaller subsets. And we see that when we train our representations using only 90 training trials, our neural data transformer and LFADS do outperform the smoothing baseline still. So in summary, we've introduced the neural data transformer, which is a transformer for neural population activity. And offline, this transformer models autonomous dynamical systems and aligned M1 activity, as well as a recurrent neural network, and it runs 5 times faster. So currently it does lag on small and non-autonomous data sets, but it still improves on smoothing. And so we've introduced a drop-in alternative for LFADS, which is independent of any dynamical systems theory. And we're hoping that this can provide a different lens for neural computation and could scale to different brain areas or scale to better performances with more data. Thank you.
All right, thank you for your great talk, Joel. We really appreciate it. I'm looking for some questions, so feel free to submit those in the Q&A, and I'm also monitoring the chat box for that.
Mm-hmm. We have some additional results that maybe would be not prudent to go through if there's only a minute, but we could maybe talk about it if there are no questions.
Yeah, definitely. We have a question here, but let's keep that on the back burner because it could be fun to see some additional results. So Cole Hurwitz is wondering, on the second to last slide, it seems as though the LFADs still outperform NDT. Given that the transformers outperform recurrent neural nets in NLP, do you have any intuition why this could be the case?
Mm-hmm. I am fairly confident that this is just because transformers are typically trained on much larger data sets. And so typically it's already a challenge to get it working on a data set of 2,300 trials. We expect that, for example, if we increase the number of trials, the transformers could potentially even overtake LFADs, but in the domain or the data ranges that we care about, we do expect to see some lag because the recurrent neural network is just much more suited for the type of data we're working for.
Yeah, that definitely makes sense. And we have another one. So from Mustafa Abku, I'm sorry if I've mispronounced anything there. Is this a trial-based model like LFADS? And if so, is it feasible to use this in a real-time application?
There are still many things to prove out before it could be used for real-time application. But yes, this is also a trial-based method. So it can directly be used anywhere LFADS is used.
Good to know.
Mm-hmm.
Awesome. So if you feel like a burning desire to show some of the additional results, we do have
It's
a little
okay.
bit of time. Okay. Yeah. So now I'll say, everybody, you should feel free to answer to address any questions to any of the panelists right now. I'm going to hang on for a little bit longer. Technically, we're at the end of the session, but we do have a few minutes before we'll be kicked out. So I'll just hang out here and see if there are any further questions. All right. So already we have Chad Boulay is curious, has a question for Joel. Said he never really tried to learn about transformers because they didn't seem to have applications in his domain. But this seems to change that for them. Do you have any tutorials or codes for understanding transformers in this context that you'd like to share?
So yes, it hasn't been applied to this domain before. So I think this is the first work to show that. I would say that the existing transformers resources are quite good for understanding it in a general sense. There are a lot of resources for understanding in natural language processing. There aren't any resources for understanding it in the neural domain. We're hoping that this can be extended into a full work, but that should still take at least a few months more time. So I would suggest if you feel a burning desire, you should go ahead and check out those other resources.
So this publication will be that resource that he's asking for. Excellent.
Hopefully.
All right. Yeah, I'm going to hang out here and just keep an eye out for any other questions. But again, we are at the end of this session. And Nicola sharing a resource in the chat for kind of introduction to transformers. And I'd just like to say, while we potentially wait for any questions, I'd just like to thank all the speakers. These were fantastic talks, and I really appreciate all the wonderful stuff that we learned today. So thanks. All right, so then at this point, I think we will wrap up and we'll close out. Thank you all for attending. I really appreciate you joining us today.
Bye-bye.
Bye.
Okay, I'll kind of jump right in there and talk about development of order in the visual system, which we are interested in. We use the visual system as a model to try to get at the mechanisms that establish order in your brain. It's been a really great system over the years. And one of the things that we know is that connections are established in a activity-dependent and activity-independent manner that interacts during what we call field development in humans, and then they're refined, elaborated, and modified by experience. So here's a classic study by Horkin and Hocking. We have an adult macaque, and we have these famous ocular dominance columns shown. And these are modified by sensory experience, but they're present at birth. And they're organized through these kind of interactions between guidance cues and activity. One of the major organizing features in the visual system is spontaneous activity in the retina that is produced as waves by a specialized circuit, a number of specialized circuits in the retina. And these waves then provide positional information to the downstream structures. Here is a famous picture. So this was done back in the 90s by Rachel Wong and Marla Feller, amongst many others. These waves correlate nearby ganglion cells, and they are required to establish some features of order in the visual system. So here we have a paper by Andy Huberman and Barbara Chapman's lab then, who showed that blocking retinal waves eliminates these ocular dominance columns in ferrets. I'll show you one other picture. There's a lot of different studies on this nature, but this is a paper by Michael Carrer's lab showing that in the thalamus, where you have the segregation of the two eyes into ipsilateral and contralateral projections, blocking retinal waves, shown here, or even eliminating them from a subset of ganglion cells, perturbs this segregation and causes the projections to become overlapped. So these waves are critically important for the establishment of structure organization in the visual system. Now, what is less considered and what concerns me, or what motivates our lab, is we know that there are these structures, these unique circuitries in the retina that drive the retinal waves, but what's less considered is, are there circuit specializations in the network centrally that are dedicated to the transmission of these waves and that may interact with and influence their downstream roles. It's generally assumed that kind of like the developing brain just sort of acts like the adult brain, or is a kind of gross primitive of the adult brain, and the retinal waves come in, they're driven into the thalamus and cortex, and just drive activity sort of globally. But are there specializations within these circuitries that help transmit and organize the waves? And I'm not coming at this just randomly. We have a number of reasons to believe this. So a number of years ago, I published this paper showing the activity that's present in the visual cortex during retinal waves. And we showed, this is when I was a postdoc with Rustem Kazapov in Marseille. The retinal waves drive what we call spindle burst oscillations. And these spindle burst oscillations look like this. We have a depth recording through the visual cortex of a P10 mouse during a retinal wave. And what you see are these oscillations, these rapid oscillations around 20 hertz, 10 to 20 hertz, that synchronize the firing of the neurons within, in particular, the superficial layers. So here's a kind of depth profile of one of these oscillations. You see they're maximal right in the superficial layers and look like a thalamic input. And they synchronize the firing of the neurons in the cortex. More interesting, perhaps, these spindle burst oscillations spread as a standing wave, so that as neurons nearby become active, they actually oscillate together. And then you come over here and the next group starts oscillating. And as a result of this, neurons, in particular in the superficial layers, are very synchronized with each other. So here I've kind of quantified layer 2, 3, and 4 neurons versus layer 5 and 6, and looked at the correlations horizontally, so in the same layer versus vertically. And what you see, the horizontal correlations are very high, regardless of distance, much more so than depth, although there is some correlation by depth. And the retinal waves, they synchronize activity in layer 5, 6, but maybe not as much. So what does this all mean? It means somewhere in the nervous system, you're generating rapid oscillations at a very young age that are synchronizing the activity at a very high rate. That should be very good for linking neurons that are co-active together, meaning from the same, representing the same part of visual space, and allow them to form a network. So that's what we think. We think that there's some value added, and we think this occurs in the central nervous system, not in the retina, for a number of reasons, one of which I'll tell you about here. So this is here's a mouse. These are recent recordings, but we did the same thing in rats many years ago. This is a 6-day-old animal. So during this period of retinal waves, spontaneous activity in the cortex, what you see again are these spindle oscillations here shown in low resolution. This is 100 seconds. So you see 2 retinal waves with these oscillations. And as a result of these oscillations, your spontaneous activity, this is the spectra, a normalized spectra, it's whitened, but you can see this really high peak right at this sort of spindle frequency, in this case 10 hertz. If we remove the eye, we give it a nucleation, let the animal recover from anesthesia. I should mention, actually, I glossed over this, I was supposed to put this in. All of what I'm going to tell you about are head-fixed, unanesthetized neonatal pups. So we anesthetize the animals, put a head post on them, keep them warm, keep them head restricted, and let them wake up. This lack of anesthesia is critically important because almost all the activity goes away if they're anesthetized, in order to get a kind of naturalistic view of what's going on in the animal. And we use multi-electrode, usually silicon probes, now 36 and 64 channels through the depth of the cortex. So when you nucleate the animal, all of this oscillatory activity goes away, and you're left with just a little bit of spiking and a few bursts. And we can see this really clearly here, that these 10 to 20 hertz oscillations are just eliminated. So that's one of the pieces of evidence we think that they're driven by retinal activity, and most of the retinal activity drive at this age. We know from work by Michael Crer's lab and me as well, and a number of other people, Christian Lohmann, are driven by the retinal waves. Okay. So I don't want to belabor that point. But interestingly enough, if you leave the animal for even an hour or 2 hours, these oscillations start to come back. You get some kind of compensation. And while activity is still suppressed after 2 hours, it becomes oscillatory. And here you see this green line. You start to specifically recover these rapid oscillations. So you don't need you need the retina's driving the activity normally. In its absence, when you take it away, you basically lose activity and you get a few primitive bursts. But over time, the compensation in the thalamocortical circuit brings back oscillations, and we think that's good evidence that they're central, not sort of retinal. And other piece of evidence for that is that if you look at the autocorrelation of activity in visual cortex, we've now done this for thalamus, versus retina, you only see this oscillatory structure in V1. You don't see it in the retina. Okay. If anyone wants to argue about that at a later point, please email me. We can talk about it. But we're going to take that as a given that, yes, there is a kind of specialized central circuitry that turns retinal waves into these rapid spindle burst oscillations. And we'd like to know how they're generated. So I'm going to tell you about one recent story first. And this was done by Yasunobu Murata, who is an advanced postdoc in the lab. And the question we had is, do interneurons in the visual cortex contribute to the synchronization and generation of these oscillations? And he modified the sort of standard, what are now sort of standard techniques for manipulating activity, but got them to work really early in the brain. And so we take GAD2 Cree mice and express a floxed virus. And put the virus in the visual cortex locally at P0, and we can actually manipulate activity as early as 2-3 days later. And so put an electrode in, give the drug, you do baseline activity, give an injection of the drug, and we can look at how interneuron activity is affecting cortical activity. So here, some of the controls, we can express them. The virus expresses in GABA neurons. It expresses in 90% of the GABAergic neurons, and 90% of the expressed neurons are GABAergic. And even at P3, so here is the inhibitory dread that we're using, and here's excitatory dread we can use. Both of these have effects at P3 that are equivalent to P10. So this is the change in millivolts. This is done in a slice when you give drug. Okay, so remarkably, we can do this quite early. What did we see? Well, so there's been a lot of discussion for many years, which is, are interneurons and GABA excitatory or inhibitory to the cortex? And a number of studies have recently suggested that actually in vivo, although in slices, GABA is excitatory, GABA is actually inhibitory in vivo, and we found the same thing in visual cortex. So at P3, when you suppress the activity of GABAergic neurons, you increase firing in the cortex. So this is, we have an example shown here. Here's the LFP. Here's the firing, mostly around layer 4 and superficial layers. You give injection of the drug, you suppress GABAergic interneuron activity, and you increase the firing rates. We've blown this up here. So interestingly, these oscillations seem to be untouched. So you increase activity something like twofold, but you don't change the fundamental structure of the oscillations in the visual cortex. Now, we don't know how much, we're really affecting interneuron activity, but we think this is fairly reasonable evidence, combined with a number of other pieces of evidence throughout the years from my lab and others, that pharmacological inhibition of GABA doesn't really affect the oscillations at these ages, that the local cortical circuitry is not involved in the generation of these spindle burst oscillations. Here's some quantification of this. So control animals don't have the effect, and GABA has about the same, interneurons have about the same effect on firing throughout development from P3 to P7 to P11. And spectrally, it doesn't change the system. Okay, so I don't think they're in cortex. Could be involving the subplate somehow. Maybe we're not getting down there. Maybe we're not manipulating. But we think we have really good evidence that it's where they're actually generated is in the thalamus. So the first piece of evidence of this is if you look closely at this depth profile, right? It looks for all the world like a thalamic input. It mimics a thalamic input in terms of where it's coming in layer 4 and then moving up through layer 2, 3. So Rustam Kazapov's lab and Heiko Luhmann's lab have done really great work in the somatosensory cortex showing that the thalamus is in fact the origin of these spindle bursts in somatosensory cortex. So we're really strongly suspecting that they are originating in the thalamus in the visual system too. Well, how do we go about looking at this? So again, Yasunobu had started recording simultaneously from LGN and visual cortex in these baby mice. Here's a picture of a electrode into the LGN. We published a couple of papers a few years ago that I'll tell you which this data is drawn from. So here we have a line recording, at least temporally, maybe not spatially, of the spontaneous activity in the LGN and visual cortex. So these recordings, of course, are not exactly aligned spatially, so we don't necessarily expect activity in LGN and cortex at the same time. We think this one was slightly aligned, so we show it. But what you can see is that activity in the LGN during this time looks a lot like visual cortex. It's denser, but it has lots of quiet periods and then these long duration periods of activity. And here's the visual cortex. Here's our spectrogram. If we do a spike spectrogram, we see that the spikes have oscillatory structure. In this animal, it's about 30 hertz, a little less than 30 hertz. But on the whole, if we average a number of animals, we see that the LGN spikes are at around P10, P9 to P11 are 20 hertz, 25 hertz, and the LFP in the visual cortex, which would be reflecting this input, is around the same frequency. So we think the thalamus is producing these spindle burst oscillations and it's transmitting it to the visual cortex. And we've blocked LGN and it blocks activity in cortex. We blocked retina and it blocks activity in the LGN, as you would expect from what I've said. But we found something really surprising in these studies and trying to figure out how these oscillations are generated. Are they generated in the thalamocortical loop or just in the thalamus? And what we found is that the cortex is really contributing not only to the oscillations, but to the general drive of activity in the thalamus during this early period. So here is an example. We take an animal, and this is the LGN, spontaneous activity. We get these oscillations and a lot of firing. And you silence visual cortex. In this case, this is a rat that we've put glutamate blockers on the cortex, and you knock down activity by something like 80% in the thalamus, and you eliminate the oscillations. This is really surprising. I don't know if I discussed. This is really surprising from a number of perspectives. One is that in adults or even P13 animals, this just doesn't happen. So you don't need the visual cortex to drive thalamic activity. It's much more modulatory. So we have a unique period of development that is the same time when you have these retinal waves, when the cortex is amplifying activity in the thalamus by something like 5-fold, at least. So knock it down by 80%, it's a 5-fold increase. So we don't really think activity is sort of generated in the cortex, but it's coming up because it's generated initially in the retina, goes up to the thalamus, down to the cortex, and makes an excitatory loop that amplifies the activity. And you can go through those papers if you don't agree with that or argue with me. But what I want to do for the rest of the talk is look at unpublished data that we're exploring what is the role of these oscillations and the cortical feedback in activity and generation of form in the visual system. Okay, so we're doing mice, and we're starting to spike sort the neurons as opposed to multi-unit activity in the thalamus. And if we do this, we can get, I think, it's much harder to sort spikes in development. I'm a little bit nervous. I wouldn't make too hard a claim. We're enriching for single units, let's say. We can get, if we have high firing neurons, we can get nice autocorrelations from those neurons. But a lot of the neurons fire at such a low level, we can't get accurate information one way or the other from the autocorrelations. But when we do this, we can see the spread of actually retinal waves across the thalamus. You can see kind of a wave kind of come up here and come back down, one come up, one come back down, as you might expect if we're picking up the waves. If we take a number of high firing neurons that are close to each other and take their spectra, they are oscillating, as you might expect from the multi-unit activity, along the spindle frequencies. We can further explore this and try to get at a little bit of the function of these oscillations. What we do is, if we're lucky, we stick an electrode through the ipsilateral and contralateral projections in the LGN, and we can define the neurons, and we give light flashes to each eye, and we can define which of them are ipsilateral responsive and which of them are contralateral responsive. We don't see very many pure ipsilateral responsive neurons. You can see these strongly respond to ipsilateral, but they also have contralateral responses, whereas you don't see a lot of ipsilateral responses in the contra-responding neurons, as we might expect. So if we do this, we take the neurons and we sort them by distance across the electrode, on the same electrode here, on electrode that's 50 microns away, and we sort them by whether we take the pairwise correlation coefficients of the firing, contra versus contra-responsive neurons, IPSI versus IPSI, or contra versus IPSI. And we sort them on we do this correlation at a very fine time scale, so 20 millisecond windows, where we subtract out an 80 millisecond window. So we can really see not the sort of general increase and decrease in firing that happens during retinal waves, but if they're correlated on a very fine time scale, as you would expect if they're synchronized by these 20 hertz oscillations. And in fact, what we find is that contralateral versus contralateral neurons, or ipsilateral and ipsilateral neurons, are synchronized on this fine time scale. But this drops off with space. Now, even at a 50 micron distance, contralateral versus contralateral neurons tend to be correlated to each other, but contralateral versus ipsilateral neurons are not correlated, as you would expect if you're being driven by one eye or the other. So this is not too surprising, but it's nice we can sort of see both, that these retinal waves are decorrelating activity in the LGN, which has actually been shown in ferrets by Willicke back in 1999, but also that they're synchronized on this very rapid time scale. And we can show this a little bit further by doing the spike coherence. So correlated pairs, they're actually coherent at this spindle frequency, whereas uncorrelated pairs aren't. And if we take a spike-triggered population mean, you can see structure. So just every spike, we look at the population firing, you see this oscillatory structure in that. So we think that these oscillations, these spindle burst oscillations arising in the thalamus, are synchronizing, sort of do what oscillations doing. They're making a small time window for the neurons to fire so they can better drive the downstream cortical network in order to stabilize their synapses. Okay, but what is this all? Does this have any real effect on development? So to get at this, meaning Yasunobu, using the mice, as I told you about, used EMX Cree and injected the dread cord at P0 into the cortex in order to silence cortical neurons, and then look at the effects on the LGN. So here's electrically what we see, very similar to these pharmacological studies I showed you in the rat. When you silence the cortex, you knock down, of course, activity in the cortex. We don't completely eliminate it. And activity in the LGN is massively silenced. In this case, around 50% reduction in the firing. This is the P10 animal. And perhaps more interestingly, this silencing of the cortex eliminates these the correlated activity in the LGN. So now at baseline, spike autocorrelation is around 20 hertz. When you block cortical activity, you eliminate that autocorrelation. And more interestingly and surprisingly, blocking cortical activity removes that correlation of like neurons. So normally in animals, right, contra versus contra neurons these are animals. So this is the average correlation between contra neurons is significantly above zero, let's say, and it's eliminated when you block the cortex. Whereas contra versus ipsi neurons are not significantly correlated. So basically you turn
So I'm sorry, Matt. We lost sound from you about 20 seconds ago.
Oh,
Oh, now you're back. Great, thank you.
Okay.
Sorry for that.
Okay, thank you. So I hear I lost sound. So I'll repeat this. So when we block the visual cortex, you eliminate synchronization of local LGN interneurons, making them unable to. They're not more significantly correlated with each other than they would be to an ipsilateral neuron. So we want to know the significance of this at a structural level, developmental level, and we really would actually expect this to have a strong effect on the thalamic projection to the cortex, because these correlations in the thalamus to be really important to drive it. But that's hard to get at, and we're working on it. But in the meantime, we've looked at this segregation in the LGN. Is this amplification from cortex critical to do what retinal waves do, which is to refine topography in the LGM. So to look at this, Yasunobu again did the same thing where he expressed the cord virus in EMX Cree mice, and then from P7, which is when we think that we could probably do it earlier, but we know things are expressed, and this is when the cortical feedback effect is strongest, until P12. We gave 2 daily injections of the cord to suppress the activity, and then labeled the 2 eyes to see if it affected the refinement. And most of this refinement in the 2 eyes occurs during this period, but you do have another reduction by half in overlap during this period, driven by late retinal waves. So we would expect to block that. And that's actually what we've seen. This is still fairly preliminary data, but here you can see what you'd expect at a P12, P13 animal. You get the contra and ipsilateral projections. They are not very well overlapped. Here you show the overlap in white. You get a little bit of tip here. Whereas these animals that have had their cortex blocked for a number of days show overlap in the 2 projections. And here's the quantification of overlap versus the percent overlap versus the percent occupied by the ipsilateral projection. Instead of being down in the 15 percentile range, you're up in the 40 percentile range, which is this is a number you would expect if you stopped segregation around P7 when we started giving the injections. So they've refined again down to around 40% by that time. This is by Bill Guido and others show this. Okay, so we know, so then this ipsilateral, this projection back is critically important for even this sort of low resolution refinement. We predict it would be very important, for thalamocortical projections, and we're looking at that. The last thing we want to ask is, so what is to stop it? Is this really a specialization of this period? Of the retinal wave period, and it is. So I'll show you this evidence. So P9-11, when late period retinal waves, you block the cortex. This is back rats again, and you silence the LGN. So knock activity down by 80%, remove oscillations. Earlier, you also reduce activity in the LGN. It's not as critical. So it's around 50% reduction, and you don't change the oscillations. We don't know about the synchronization. But at P13 and 14 after the waves and around the time of eye opening, when you silence the cortex, you actually increase activity in the LGN. And we think that this is due to the development of functional feed-forward inhibition from cortex to thalamus driven by the thalamic reticular nucleus for the following reason. We express opsins in the cortex. They make, you can see, this is a general AAV chronos into the visual cortex. We express in all layers. You can see terminals in the thalamus even at P6 in rats. And you can drive firing in the LGN even at P6, 7. So we fire activity in the visual cortex, we drive it in LGN. Same at P10. But interestingly enough, at P13, we start seeing a functional inhibition of baseline firing at this time, suggesting that we've activated. I mean, this is characteristic of TRN neurons driven from the cortex, and we don't see it at these ages. So we think that this period of cortical feedback amplification is mediated by a lack of inhibition in the network, allowing for a feedback excitation, and it's ended by the functional development of this inhibitory circuitry. So I'll repeat that sort of what I said. Like, early development is characterized by no feedback amplification, allowing for this synchronization. And that this selective amplification, maybe it's selective, maybe it's general, but it synchronizes topographically matched LGN neurons to rapid timescale. So we're really interested in computationally trying to figure out, why do you need to synchronize local LGN neurons in order to segregate the eyes? You shouldn't really need that. I mean, I have some ideas, but we'd like to look at this kind of computationally. I forgot to throw out there, in general, the lab is recruiting postdocs, so please get in touch with me if it's interesting. I will skip through the remainder of that since I am out of time. But we're looking at how the thalamus changes spontaneous activity in the cortex. So in conclusion, there's a corticothalamic booster circuit that's specialized for the transmission of retinal waves. I mean, it actually occurs throughout all of cortex, but so in visual cortex is specialized for retinal waves. Somatosensory cortex is probably specialized for twitching feedback, and auditory cortex for spontaneous auditory waves. And this booster circuit specifically synchronizes local thalamic neurons in fast oscillations to drive circuit refinements, would be our hypothesis. I didn't really get to talk about this, but we think that as a result of the specialized circuit, the brain is actually maintained in a kind of vegetative state during this early thalamic circuitry. And that means you can't process information, but it allows for this really strong drive of activity to drive refinement. So thank you all for coming. Thank you to the members of my lab. I mentioned them as we went along, mostly Yasunobu Murata, and I would love to take questions.
Great, fantastic. So let's I will clap on behalf of the whole audience, who unfortunately can't hear you here. I have a whole piece of paper full of questions, but unfortunately the public is also asking lots of questions. No, I'm just kidding. Like there's lots of great questions. I will start asking a question by Maya Wild, who asked, what is the effect of blocking the cortical activity during development on visual behavior post-eye opening?
Yeah,
and it's so interesting. No, like it matters, the behavioral the developmental programs.
I think so. It's a long way off. We would love to know that. I think we'll characterize all of the electrophysiological and anatomical differences, and then make some predictions about behavior. I tend to stay away from behavior with like a 10-foot pole. But you're right, that's the ultimate output of the nervous system, and we should get onto it, but we don't know at the moment. Thanks for the question.
Great question by Maya Wild again here. Let me ask a question by Matthias Hennig, who asks, the acute experiment blocking cortical activity was done at P10, if I saw correctly, where the retinal waves are quite small and less correlated. Earlier waves supply stronger correlations. Could the lack of correlations from waves lead to cortex taking over in driving correlated activity?
Does the change in, so they're referencing differences between different stages of waves. So there are early waves, stage 2 or cholinergic, and late waves. And the Crare Lab has shown that in vivo these late stage waves they correlate a smaller region. So for us, we would still pick that up because we only see one small piece of space at the moment. It's a great question. So does this lesser drive, is that what allows the cortex to kind of take over? I don't know. It's a great idea. I mean, it doesn't look like the cortex is taking over. It actually looks like it's being less driven, though, because it's quite strong early and becomes less strong later. But it may be allowing for something like the TRN to get in there and start suppressing its activity. That's a great question. Thanks.
Great, thanks. So let's take a question by John Lamoute, who asks if you've seen Rolf Hassler's work on the reticular nucleus. I'm not sure if okay. Let's.
I'm sorry, I'm not immediately familiar with it.
Great. Let's move on to a question by Silvia Pitolo, who asks, thanks, beautiful work. What do you think is the contribution of the developmental GABA switch that you mentioned and the lack of inhibition that you see before eye opening?
So I don't think it's involved. So we have shown, and then this confirms other groups, that GABA is inhibitory at P3, P7, and P11. So the switch is not regulated by that. I didn't go into it, and we've shown that in hippocampus, GABA is actually excitatory at P3 and becomes inhibitory at P7. So changes in the hippocampal activity may be dependent on this switch, but we don't think it's dependent in the cortex. What I think is happening is circuit based, right? So it's not necessarily polarity, but you have neurons that are coming online. So I showed in 2014 that you don't get feed-forward inhibition in the cortex until this switch. You get sort of feedback inhibition, but not feed-forward. And we think that this is Simon. I'm blanking. Simon has shown that there's a reorganization of cortical circuitry and inhibitory circuitry. So we think it's a circuit-based effect, and not one of polarity.
Great. So here's a question that I wanted to ask, which is, you analyze burst and synchronicity, and with the strong assumption that this drives development, and it's very convincing because when you causally perturb the system, you show the predicted effects. But what can you say about the path? Like how is it that the burst or the correlation ultimately translates into a different brain?
Yeah, that's great. That's why I mentioned we need to do some computational modeling of this, because it's not immediately apparent. So the first thing I'll point out is that this blockade of the cortex, it knocks activity down massively. So you could simply be reducing activity, and that itself is what causes this this effect. So it's going to be, the dream experiment is to remove the correlations, but keep activity basically intact. And that has been fiendishly difficult to do. We can't do that. So, we're desperately trying to remove the oscillations, but every idea that we've had so far hasn't really worked out. So I think at the moment we're left with trying to explore what it would mean, these correlations. Like how could you how do you get to where you're going with that? I guess that's,
yeah, and in part, it's just might be that like the system you look at is just so complicated. I remember very dimly an old Gilles Laurent paper where they actually managed to affect synchronization, if I remember that right, without affecting firing rates. But that's only possible because like the grasshopper is just such a weird animal system. So
yeah,
that's but that's, as a computational scientist, I'd be super interested in that.
Yeah, and so I'm very interested in, why do you need local you don't really need local LGN neurons to be correlated to each other to drive refinement of the retinal terminals. Like they should be on a single cell level, but maybe, I think about the LGN, it's not a structure that's attached to each other. And so maybe you need some kind of global imposition of correlation in order that you don't get local minima of like an ipsilateral neuron in the middle of a contralateral section, where it's refined onto a single cell, just in the wrong place. So that's the kind of ideas we're looking at.
Yeah, great. So I have a question by Jimmy Dooley here, who asked, when you remove retinal inputs and see a decrease in activity, spindle bursts, followed by a rebound, why do you think the rebound occurs?
Hey, Jim, thanks for the question. Another great question. We are submitting a paper soon, actually, that extends that finding out. So at P8, at P10, firing rates are totally normal in the LGN, in the cortex, and they develop normally. So there's this like central I think there's basically a homeostatic mechanism that drives compensations. What I will say is, we don't think it's like sprouting from another sensory region into the thalamus, because we did these lesions at P6, which is after the time that really happens. So in our hands and others, it just looks like, I think what happens is that the retinal waves are coming in consistently, and they're suppressing, in a way, the activation, the intrinsic excitability of some group of cells in the thalamocortical system, because they're what's driving things. As soon as you take that drive away, you're going to get this sort of more graded ramp of excitability in some group of neurons, probably layer 5, cortex, that becomes more excitable, starts driving bursts, and then that's what starts triggering the system. But we have no real evidence for that. But thanks for the question.
Great, cool. One more question, and forgive me if it's a naive question, but in a way, you said that inhibition kind of ends the state where the system is basically operating early on. I was curious, if you block inhibition, could you give the system the ability to do things at a later point of time that it could otherwise do at an early period of time? I'm not sure what's the ideal perturbation there, but I think it would just be so nice to be able to kind of see that you can extend that capability through blocking inhibition. Or like,
it would be great. I mean, one of the justifications we give for this work is, the early period of development is very, very plastic. I want to give a number, but let's say 100 times that of ocular dominance column plasticity, or the critical period. So, if you want to regrow retinal axons, or you want to grow thalamic axons, you don't want to play around with inhibitory, like perineuronal nets or some subtle balance. Like maybe what you really want to do is get rid of thalamic inhibition. You want to generate massive bursts in the system that would probably cause epilepsy and be totally clinically unusable. But anyway, that's the general idea, right? Yeah. Can you drive really strong sprouting and reorganization of the system when you want to regrow, say, axons or something?
Great. Awesome. Well, this was a super inspiring and real fun talk, certainly for me, given how much I'm interested in that for historical reasons. And like, it's just such beautiful work. So with that, I hope I express what everyone else in the audience feels. Let's give the speaker one more clap. And thanks so much for the great talk. And thanks everyone for tuning in.
Thank you so much for coming. Thanks, Conrad, for organizing. Are you in Europe, or are you in the US?
I'm in Philadelphia.
Okay, so you're not staying up too late. If anyone tuned in at 3 a.m., thanks so much.
It could be any time of the day. If you look at the time zones of the
at
people at NeoMatch conference, there's some distribution, but it never reaches zero. So you can be anywhere on the planet, and there's people who you could meet for lunch realistically, which is pretty crazy if you think about it. In any case, thanks everyone.
For organizing. Thanks,
and
for
have a good day. Bye-bye.
Bye-bye.
At the beginning, and we will go ahead and go live. Great. So as people filter into the room, we will just introduce our panel here. So hi, everybody. My name is Megan Peters, and I am one of the co-organizers of NeuroMatch Academy, or NeuroMatch and NeuroMatch Academy. And today we have a great panel of 3 talks, all on various aspects of, well, 2 of them are kind of on various aspects of consciousness, and then we've got kind of real world predictability here. So there's going to be a great talk and discussion going on here. Just to orient you all who are filtering into the room now, we have a Q&A button at the bottom of your screen. So you can use that to ask questions to the panelists. You can also use that to reply to each other's questions in comment threading. So please go ahead and use the Q&A box as we get into the talks. And we will also be dropping a few related links into the chat. So be sure to pay attention to that. And now that it is 6, we will wait just a few more seconds before introducing our first speaker. So why don't we have the other 2 panelists turn off your cameras, please, and then we will go ahead and introduce our first speaker. And our first talk is from Regan Gallagher from Monash University, and he will be talking about the massive report paradigm, assessing the richness of conscious visual experience. So Regan, go ahead and share your screen. Perfect. And we can see what we're looking at here. Looks fabulous. Okay, so why don't you go ahead and take it away, and I will hold up some fingers when we are approaching the end of the talk. Thanks.
All right, so mic is working, yes? Okay, first up, what I'm about to talk about is a large project that involves a lot of researchers. So I've got listed here all of the other people who've been instrumental in getting this work up and running. So I'll be presenting the research that I've done, the experiments and analyses on, but it is part of a large project. So it's worth knowing that a lot of other people have gone into developing this to the state it is at. Okay, and the project really deals with an age-old question of how much do we see? And both philosophers and neuroscientists for as long as we've had them both have really delighted in showing us how much of our experience is illusory, so that when we look at the world, it appears as though we have a really rich experience. We take in so much, but when we actually analyze the information content of our visual experiences, it appears as though there is not that much information that we are able to report. So one of the classic paradigms that has demonstrated this is the Sperling paradigm, in which participants are presented with an array of letters, as you see here, for a very short period of time. And even though subjects feel as though they see everything, when they're asked to report on any of the letters, usually they only are able to accurately report 3 or 4 letters. So the implication here is that the information that we have available to our conscious perception is actually really limited, even though the subjective experience is the opposite. Of course, this is more complicated because if you present the same stimulus for that brief duration, and then after the stimulus has disappeared, you cue one of those rows, let's say the middle row, participants then go from very poor reporting to almost perfect reporting. So they can report any one of those rows after they've seen it. And so it appears as though what we are seeing is greater than what we can report in any particular instance. If we loosen the constraints on the Sperling paradigm a little bit further, we can see that people don't just report, let's say, the letter A or Y or a Q. They're able to report a lot more than that. So they can report that they saw letters, that they were from the alphabet, that they were white or uppercase or on a black background. So again, what is being experienced is far more informative than what a lot of these experiments are actually tapping into. So the point of the talk today is to examine that problem of deriving the information content of a conscious visual experience. So the problem, to put it very simply, is that previous tests of visual experience leave out important information, and they're underestimating, we hypothesize, that the relevant information content of our conscious experiences. And so the solution of the massive report paradigm is to allow people to report more and report more freely. And importantly, for understanding the informational content, we need to test what they can identify, as in what's present in that image, versus what is absent. So what they can differentiate one stimulus from another. And I'll explain a little bit more about the importance of that. So today I'll talk about the massive report paradigm, in which we're extending some typical psychophysical parameters. We use massive samples, massive responses, and we massively improve, or at least increase, the informational estimate of conscious visual experience. Okay, so what does that look like? So here, hopefully the frame rate of your screen is adequate, but you should be able to see a variety of images flash relatively briefly on the screen. And I want you to notice two things. The first is that of any particular image, there are probably many things that you could report. More importantly, for the informational content of consciousness, is that each of those images can be easily discriminated from every other image. Okay, so those are two important aspects of understanding the informational content of conscious experience. How rich an experience is and how differentiable it is from other experiences. Okay, so here's what a trial sequence in our paradigms would look like. We have a fixation point, an image flash briefly, and then a mask. And in one aspect of the paradigm, we had people just freely report what they saw, as well as give a confidence level. In a follow-up experiment, we took all of those words that people reported, and then we played them back to a new set of people in which they reported their whether they saw that word in the image and whether they were confident. From 1 to 4, low confidence being 1 and high confidence being 4, and yes or no, they saw this particular word. So fountain or various or park, etc. For each image, we had obtained a large set of words. We have a large set of images, over 420 actually. And importantly, what we can do is for each image, we take this set of words that is present for that image, and we can take the word set from every other image as being absent from this image. Obviously, we have to exclude the words that overlap, but we now have a massive set of words that we can play back to people and see whether they can respond with yes or no and their confidence in this new paradigm. Okay, we then estimate the information transfer rate. So what this basically is based on Shannon's information theory, in which the number of binary decisions accurately decoded through a physical medium disturbed by noise, that is the definition of information. And so we have a simple formula of the number of responses people are able to give, yes or no, so it's a binary response. And because it's a time value, we can take that bits estimate and put it per presentation time. So we get a bits estimate per second. Okay, and we also have to measure accuracy. And we do this using signal detection. We take accuracy as the area under the curve of this rock curve. If we have time at the end, I can explain a little bit more how that's relevant, but just the takeaway here is that we use signal detection theory as our accuracy measure here. Okay, what does that look like in an example? Well, imagine people saw this image of the president, prior President Obama. They see these present words, President Barack Obama, etc., etc. And then we select randomly from this set of absent words and people discriminate between them. Then we can look how accurately people classify the present words as being present. So yes with high confidence, all the way down to no with high confidence for the present words, and do the same for the absent words. And we get this pattern where yes words are responded to, or present words are responded to more accurately and confidently, and absent words are responded to as no accurately and confidently. Okay, and then we can derive our AUC measure from this reporting method, and we can use that to develop our information bits per second estimate. Okay, so that's just an example. Here are the pilot results where we do the exact same thing. We start with these very simple images with only a couple of words: heart, love, outline, black, red, color, etc., and we present it for a longer period. And then people also see a subset of these absent words. We get this same basic graph. We derive our AUC. So this is real pilot data for this cartoon image. And we can use that formula to derive a bits per second estimate. So this simple image, based on these responses, we get a 223 bits per second estimate. This image, you see the same pattern of results, similar pattern of results for the AUC, and an increased bits per second estimate. And again, for the final pilot image, we get a similar answer. Okay, and then here are the test trials. So we used a real world image taken from a, thank you, Megan, from a subset of a larger image database. And you can see, using the same formula, the same word analysis that we did in the previous ones, we get a massively increased bits per second estimate for the information transfer rate from a single image. And the point is that people are able to differentiate between this huge number of absent words as well as a relatively limited but still large set of present words. And so we can derive a huge amount of information from these images. I'll just flick through the next few images so you get an idea of how consistent these responses are. So here we have another just natural scene. We have a very high bits per second estimate. Same again, and still very, very high estimates. Okay, so I'll just say that that's our pilot data. This is going to be a registered report. So we're still yet to submit that as a registered report. But to just wrap up, and I'm happy to answer questions about any of the specifics that I've talked about, but basically what we've tried to do is loosen some of the typical constraints on a psychophysical paradigm. So we've massively increased the number of responses people can give. We have massively increased the number of participants. So we've put these out on MTurk, allowing us to gather huge amounts of information, all using either the same image or a variety of images, a variety of present words, a variety of absent words. And by doing this, we see a massive increase in what participants can report about their conscious visual experiences, over and above things like the spelling paradigm and other similar paradigms. A really important aspect of the information theory basis of this paradigm is that information depends not only on what is present, it also depends on what is discriminated from that experience. And so when we take into account all of the absent words, all of the things that each stimulus can be discriminated from other experiences, then we start to see this massive increase in information. So it depends on what is present, it depends on what is experienced, but equally important is that the information content is also dependent on what is excluded by observing a particular experience. Okay, information theory also provides a principled way to estimate, thank you, Megan, the informational richness of a conscious experience. Previous estimates are that the human visual system or vision comprises only about 40 bits per second in terms of informational content. And obviously, our estimates drastically revise this estimate. But of course, the upper limit really depends on what can be excluded, and the upper bound could be much, much larger. So just to conclude, a moment of a conscious experience is potentially richer than philosophers and neuroscientists might like to lead us to believe. Thank you.
Fantastic. Thank you so much, Regan. We can't do clapping in Zoom webinar, but if you enjoyed the talk, please drop something in the chat to say, yay, that was a great talk. I think this was a great talk. So if there are questions from the audience, we have a few minutes for questions. So please use the Q&A box at the bottom of your screen to submit questions. And while here we go. Okay. Oh, great talk. Thank you, Enrique. All right. So while we're waiting for some other questions to roll in, I wondered if I might ask a very brief question, which is, this seems like a really cool way to look at the interaction between the bit rate of perception or the informational content in perception and the informational content in a visual stimulus itself, or the spatial distribution of stuff on the screen in the visual stimulus. So I'm just wondering, do you have any plans to look at that?
Yes, so we have looked at, say, pure noise stimuli, Gabor stimuli, and what is very interesting is that while the information that's present in those stimuli is very difficult to describe by your novice observer, most people don't know what a Gabor patch is. When you factor in the discriminability of a Gabor patch from any one of the insanely large number of conscious experiences, you can still derive a large estimate. Now, that is important as a critique of the paradigm as well, because you could say even the simplest experience in our paradigm appears to have a very large, informational content. And so some people might say, well, a Gabor stimulus is not informationally that rich, but because it excludes, our brain is able to have a tremendous number of experiences. So having this one experience excludes all of those. So in that way, it is highly informative.
Cool. Thanks.
Does that answer your question?
Yeah, I think it does. And I'd love to talk to you more afterwards. And unfortunately, we're now out of time, but just very quickly, there's some questions about dependencies between words and also like the length of time of the stimuli. So maybe you could just say like very briefly and then answer more in the chat or something.
Okay, unfortunately, I can't see the Q&A, so I just have to guess.
Okay, it's really, do you plan to change the duration of the stimuli? And also, do you look at dependencies between words when you are computing this measure?
Yeah, so quickly for the first one, yes, we have a variety of time estimates, both for the word generation and for the playback scenario. I could talk about that with an interested individual. But for the other one, correlations between words is a big factor that we're exploring right now. So, if you see a cow in a paddock, then words like grass and sky and fence, etc., all can perhaps come along for free, I suppose. So you get this correlation that can bump up the information estimate. So what we're trying to do is use artificial intelligence algorithms that, smart photo analysis software, use the gold standards and compare a conscious observer. And the image analyses do okay. They do well because they can see the correlations in the image and the correlations in the word. But human conscious observers, as far as we've tested, do much, much better.
Great. Awesome. Thank you. And we can continue to go. If you click on the Q&A at the bottom, you can continue to answer some of these questions as well. So fantastic. Thank you again, Regan. So we will now move on to our next question, or our next speaker, which is Olenka. And Olenka Graham-Castaneda is a graduate student in my lab here at UC Irvine. And today she is going to be talking about the metaperceptual function, exploring dissociations between confidence and task performance with type 2 psychometric curves. So please take it away, Olenka.
Hi. Okay, I'm unmuted. Cool. So my name's Olenka. I'm a graduate student at UCI in Megan's lab, as she mentioned, and I'll be talking to you about dissociations of confidence and task performance. Okay, so our goal of characterizing dissociations between confidence and task performance really fits under a bigger goal of improving the approach in researching metacognition and consciousness, specifically in minimizing confounding variables. So quickly, I'll just talk to you a little bit about consciousness. So under the consciousness umbrella, we have state consciousness and content consciousness. In state consciousness, we're looking at the difference between a state of sleep, wake, and being anesthetized, while in content consciousness, we're more concerned about what you perceive, why, and then how that influences the decisions you make. And we call our qualitative experience of sensory events phenomenal awareness or just awareness. Okay, so like I mentioned, our goal is in minimizing confounding variables, and the variable I'm talking about today is task performance. Okay, so in a given task, high levels of confidence are typically accompanied by high levels of task performance. Like in this graph here, we have as you perform better in a task, you're more likely to say you're more confident in that task. But that's not necessarily happens all the time. So in different either experimental manipulations or because of brain trauma or even because of just spontaneous fluctuations in neural signals, there can be a dissociation between performance and confidence. And I didn't say, but confidence here we're using to just track the general level of awareness of our subjects. Okay, so what do we do? So when measuring or when looking into metacognition and consciousness, we need to consider matching the accuracy across our conditions while leaving the confidence different. And we call this matching match performance different confidence, or MPDC for short. Okay, so because like I said, because confidence and accuracy don't always correlate, we need conditions in which these are matched so we can make meaningful observations about the change in confidence. The takeaway with the dissociation is that because these two processes dissociate, there could be different underlying cognitive processes that contribute to them. And we don't want to be making conclusions about the wrong cognitive process. So in working to account for this confound, we are looking at two specific questions about the MPDC effect, and that is its characterization over different levels of D prime or task difficulty, and also its dependence on temporal organization of trials. Okay, so in our behavioral paradigm, participants viewed full screen displays of moving dots, and they had to judge whether there was downward coherent motion on the left or the right of their screen in one of these two circles. They also had to report how confident they were in that judgment on a scale of 1 through 4. Right, and then what is important to note here is that we're controlling the MPDC effect using dot density in general. So all the results I'm about to show you are coming from our new preprint, which you can find right now. Okay, so first question, characterization of this effect over different levels of D prime. So in this first set of analysis looks at the examined results of fitting type 2 psychometric curves to the confidence in D prime data using a logistic function. And these plots show curve fits to the group average confidence and D prime data. So here we're looking at confidence on the y-axis and D prime on the x-axis. You can think of D prime as task performance, with 4 being subjects found the task easier, and then getting lower D prime values, we can think of the task as harder. These 3 different lines each stand for a different density, dot density. So basically a higher dot density, we're expecting to come with higher reports of confidence. And so the main takeaway with this question is that the MPDC effects are more pronounced at higher levels of D prime or task performance, as you can see the lines separating here, which implies that future experiments using MPDC should seek to use D prime in the range of 2.5 to 3, or otherwise 89% to 93% correct or accurate. Our second question was concerning the temporal organization of the trials. So we looked at blocked versus interleaved trial design. In the block design, you have the different conditions of density all within their own block, and the interleaved, their trials are interleaved together so that you can't predict what comes next. And with this, the main thing we see is that MPDC effects are stronger under a blocked under an interleaved experimental design rather than a blocked one. So in summary, we're looking at the effect of match performance different confidence in order to improve methods for studying metacognition and consciousness. And we found that MPDC effects are more pronounced at higher levels of D' or task performance, meaning that future experiments should implement MPDC at D' levels around 2.5 to 3. And we also found that MPDC effects are stronger when the manipulations are under an interleaved trial design rather than a block trial design. But with this, it's important to note that although the effects were stronger in the interleaved condition, there is still some evidence for MPDC effects in the block condition, although there were smaller. So with that, I'd just like to thank everyone else involved in the project, as well as the funding sources and institutions, and Isaac, our postdoc, who also helped with this project. Thank you.
Great. Thanks, Olenka. And if you liked the talk, please drop some praise in the chat, because we don't have any clapping that we can do here. So we have one question in the chat so far, and we have quite a bit of time for questions, actually. So this is great. So we'll do the first question. And if you do have other questions, drop them in the chat or use the Q&A box at the bottom of your screen. Okay, so the first question comes from Regan, and the question is, would interleaved trials induce sensory adaptation?
I think with the interleaved versus blocked, what we're thinking happens is that the interleaved kind of messes with your ability to change your decision criteria. So it makes it harder to adapt to the new dot density level, which makes our effect better.
Great, thanks. And do we have any other questions rolling in? While we wait for them to roll in, I can ask a couple questions. So the first question that I'd like to ask is, what are the next steps, the next plan for this project?
So we're going to take this project into the scanner now. Isaac is actually going to be doing that part. And then hopefully after that, we're going to be using it to answer another question about phenomenal awareness, and that has to do with its role in action initiation. So our original thinking was looking into whether being aware of the stimulus in front of you would prompt you to interact with it more than if it weren't. And if that was the case, then that would be a really good reason for evolutionarily for us to have phenomenal awareness.
Okay, great. Are there any other questions? I can just sit here and ask all of the questions, but I think that we might want to take a few from the audience too. Anybody? All right, well, I can continue asking the questions if you would like to do that, just for a few more minutes until we reach the next tick on the clock. So I was wondering if you, oh, here we go. We have one question, maybe? No raised hand? I saw one. Okay. Feel free to use the Q&A. Okay, we've got another one. So Regan is asking now, have you examined the psychometric function rather than D'?
Yes, I think we did. Wait, I don't know if I understand the question. So D' not being included in the examination of the psychometric function? We did a lot of different analysis in a lot of different ways in order to try to figure out how to best characterize all these different parameters and variables. But the answer is probably yes, and you can find it in our preprint.
Okay, I will look there. But maybe it is easier if I ask the question verbally. Is that, we have time?
Yeah, sure.
Okay, then,
we do. We've got a few minutes.
The reason I ask is that D' being a measure of task difficulty may not necessarily match onto perceptual appearance. And so by looking at the psychometric function, you are looking at how people perceive that, how rather than accuracy per se, because sensory adaptation could make a leftward stimulus look stationary or stationary look rightward moving. And so you get an estimate of how much perception differs from the objective truth.
So I don't know, so this might answer your question. We did look at dot density over D', or as a function of D', vice versa, I think. Also at, so that's traditional psychometric curves. And then we looked at confidence sorry, hold on. Confidence over, well, I'm gonna have to look back before I mess this up.
That's okay. I can go to the preprint. I just have not, I've recently published some work similar to what you're doing. So maybe if my question isn't answered in the preprint, I can send you something that I have for you.
Yes,
not to
please.
shamelessly plug, but might be helpful.
Yeah, that sounds great.
And I'll chime in here too. We'd love to see it. So yeah, let's talk afterwards. That'd be great. Thanks.
No problem.
Fantastic. Okay, well, we have a couple more minutes here. If anybody has any other last lingering questions for Olenka. But if not, maybe we can move on to the next talk. Okay, well, if you do have any left over, then you can drop them in the Q&A and we can answer them in a threaded chat, or you can just drop them in the chat. Okay, thanks, Olenka, for a great talk. And we will now move on to our final speaker of the event. So we will now hear from Lena. And Lena Peroni is going to tell us about online assessments of world predictability, a feasible alternative to in-lab research, which is not the title of the slide on the screen, but it's pretty close.
Okay. Hello,
Great.
my name is,
please.
yeah, okay. So my name is Lena, and I'm glad to be presenting this work with you here today. This work on predictability during reading is a part of my graduate thesis, and it's been done on the Laboratorio de Inteligencia Artificial Aplicada at the University of Buenos Aires. So, in everyday tasks, our brain performs predictions about future events, anticipating possible future actions and building an internal model of the environment that is not sensed directly. For example, we could be walking down the street as we see a car speed up at us. In that case, we would have to predict its velocity in case we need to move. Less life-threatening, but not for that less interesting, we can consider the case of reading. Reading requires language, reasoning, and memory processes. So we want to understand how internal processes of the mind and external stimuli play together in the generation of complex action. Reading might serve as the ideal case, since despite its complexity, it occurs in very amenable experimental situations. Moreover, it is a trait shared by a large percentage of the population. So when we read printed text, we are continuously predicting upcoming words to integrate information and to guide future eye movements. These predictions can be operationalized as a predictability and can be estimated with a closed task experiment. The experiment consists of a survey in which subjects read incomplete text and guess missing words according to a belief on the most probable word given the preceding context. So participants guess the words, and then predictability is defined as the proportion of correct answers for each word across participants. So I have already explained to you what a closed task is, and now I want to tell you what we use it for. We have three corpora, that is a large data set of text, each containing different types of sentences. For each corpus, we perform two independent closed task experiments. We wanted to see whether the online results could replicate the in-lab ones and if those results were robust. The first corpus we work with is made up of common and memory-encoded sentences. We perform two independent experiments, the first one done in-lab and the second one done online. In-lab closed tasks tend to be a time-consuming and expensive endeavor. A large and in practice unattainable number of participants would be required to complete the experiment in order to obtain results that are generalizable and reproducible. Also, in-lab experiments tend to be largely dependent on the people that are readily available to you, so you end up repeating the same population for several experiments. The use of web platforms for experiments is fast increasing, since they allow for a large number of participants to partake in the experiment, attaining a large definition of the results. Also, using social media platforms, one is able to reach people from several demographics, making the results more generalizable. With this corpus, we wanted to see if online could replicate in-lab results. The first thing we wanted to see is how predictability behaved within each experiment. This is a 2D histogram of predictability for both experiments. Each pixel's density reflects the number of words that have a certain value of predictability for both in-lab and online experiments. If every word had the same value of predictability for both experiments, all the points would be in line with the diagonal. Even though these results seem to be scattered along the figure, we can see that predictability behaved more or less the same for both experiments. We now wanted to see whether the type of sentence being tested made a difference. We can now again see that there was not a large difference in behavior for both experiments, although there is a large noise due to the fact that we had a small number of participants for both experiments. Lastly, we want to see how predictability behaved within each sentence. So we analyzed the relationship between word predictability and word position. As you can see, predictability increases with word position, and that for both experiments, it behaved in a similar manner. So we can say that online can replicate in lab results. The second corpus we work with is also made up of common and memory encoded sentences. Even though corpus 1 and corpus 2 share the same type of sentence, the particular sentences that make up each corpus were not the same. For corpus 2, 2 independent closed tasks were performed approximately 2 years apart, with approximately 300 participants participating in the experiment. With this corpus, we wanted to see if online results were robust. We can now see how data spreads along the diagonal and how predictability behaved a bit different for both common and memory encoded sentences, with higher values of predictability for memory encoded sentences. We can also see how the last word of a sentence have a larger standard deviation. That has to do with the type of sentences being tested. Common sentences will continue to have low values of predictability for each word along word position, and memory encoded sentences will have higher values of predictability for the last word within a sentence. So with the results, we can say that online experiments are robust. The last corpus we work with is made up of Spanish short stories. We performed 2 independent closed task experiments. First, we tested the sentences in their context. That is, we tested the whole story. Then, in a separate experiment, we tested the sentences isolated from their context. Sentences from each story would appear interspersed, showing no apparent relationship between this sentence and the prior sentences. With this corpus, we wanted to see whether context or the lack of it had an effect on word predictability. As you can see, these two graphs are a bit noisy, and that is a direct result of the naturalness of the sentences being tested, since the stories we use were not made up for the experiment. And then predictability was not balanced within each sentence. So we checked to see how predictability would behave in this context. As you can see, there are significant differences for the first words between predictability for isolated sentences and contextualized sentences. When we read a story, we have a previous context that aids us in predicting upcoming words. If we start fresh from an isolated sentences, we lack that context, and so words from the first position of sentences for isolated sentences showed lower value of predictabilities than for contextualized sentences. As we move along a sentence, that is, as we continue reading, the value of predictability rises, and because the previous context aids us in predicting upcoming words. And with these results, we can say that close predictability on some words is dependent on context. We are especially interested in these results since we are now using it to analyze how predictability guides future movements when reading natural texts. So, in conclusions, we can say that online experiments can replicate in lab results, indicating that it is correct to use them for future experiments. Online experiments are robust, and they ease the use of large sample sizes and thus collect gross predictability with higher resolution. Close predictability on some words depends on the global context on the text being read. This difference tends to appear mostly at the beginning of sentences where predictability from previous words becomes limited to isolated sentences. Dependencies of predictability effects in position in text were previously observed in a previous work done in lab by Bianchi et al. So if you have any questions, feel free to ask them, and thank you for listening.
Great. Thank you for the fantastic talk. And if you want to drop in the chat that you appreciated it, because we can't applaud, I'm sure that we would appreciate it. So do we have any questions for Lena? All right, we have a question from Regan, who's asking, can you give us some information on the number of participants excluded online versus in lab? Are they comparable in your samples? Also, nice work, smiley face.
Thank you. I don't know if I understood the question. I can't see the chat, I'm sorry. But for example, I want to show you the results. We did an online experiment last year. That is the paper that we published last year was a direct result of that experiment, and approximately 2,500 participants were able to partake in the experiment. And in lab experiments, we usually end up using 18 participants since they need a lot of time to read the stimuli, the text, and you usually end up taking, I don't know, maybe 6 hours per participant. So it is very not practical.
Yeah, I think that the point is well taken, that we can really get a lot of volume out of online experiments. Yes. I think to follow up, Regan is asking to expand. We find good data from online samples, but we need rather strict a priori exclusion criteria. And I'll jump in and say that we've found the same thing in psychophysics studies online as well, that the quality of the data is just really much poorer when you can't control whether people have their cat walking across the keyboard or, whatever else they're doing. So are you finding, though, that with these tasks that you have to set very strict exclusion criteria, or that you end up excluding a lot higher of a percentage of your participants than you might in the lab?
No, we actually didn't have to end up excluding several participants. We had a very important restriction, and that was that participants could not partake in the experiment with their cell phones, because we wanted to avoid the use of keyboard predicting services. But with that restriction in mind, we didn't end up having to throw away any results.
That's really good. That's fantastic.
Yes. And there was also the case that you could log in and log back off in the platform whenever you wanted to, and the results were saved. So participants were able to partake in the experiment in a window of a month, maybe or so.
I see.
So that had that eliminated certain restrictions or stress that could maybe change the results.
I think that's actually a really interesting point, because usually what we're used to in the lab is bring people in and make them sit in front of a computer for an hour and do the thing and then leave. And if we can do it online and have you just log in and do like 10 minutes at a time here and there, we might actually end up with better focused participants and better data. Not a bad plan. Why would we ever collect data in the lab ever again?
Yes, and also the data collected can be thought of as more natural, since participants partook in the experiment between meetings, between Zoom calls, and then they were in their natural environment. There is the influence of the lab and of the people probing you was eliminated.
So do you have any plans to look at, or I'm sure you have data on how many sessions or how often they logged in for 5 minutes at a time. And so it might be interesting to see whether some of the divergence that you see between the lab and the online participants, whether that in any way correlates with the distribution of time spent in the online platform.
Well, we hadn't thought about that. That is very interesting. We have a large volume of data that can be used in a lot of ways. We also plan to do emotion analysis on the answers, because we want to see whether the global context affected the words that were being predicted by the participants. So we have a lot of different points, then we can explore the results.
Great. Well, it sounds like a great project. Thank you so much for sharing it with us. Do we have any other questions from the audience before we close up? Okay, well, I'd like to invite all of our speakers to turn on their cameras and wave. Once again, thank you to Regan, Lena, and Olenka for some fabulous talks. And I'd also like to send a shout out and special thanks to Amit, who is our backend tech from Neuromatch, who has made this session run very smoothly. So thank you very much. And we can follow up later. You know how to find all of the speakers, and maybe you will even match with some of them on the NeuroMatch platform. So look forward to seeing you all around NeuroMatch in the next coming days. Thanks very much for being part of this session. Take care. Bye-bye.